So as Shan said, thank you all so much for coming out here. For those who traveled near and far to be here, from all the families who drove down from around the US and from Canada, from the scientists who flew here from around the world, to all of those logging in virtually. It really means the world to us here. Let me make sure I can advance the slides here. Okay, there we go. And so one of the things that I'm most excited about over the course of this weekend is to get to hear from each of you. So everyone here has a story, and the story is for what led them to be here this weekend, whether it's a family who's been affected by these diseases or a scientist who's been working on this channel or got drawn in to help us as we navigate the waters forward. And I'd love to have the chance to learn from each of you about your journeys and your story. Each morning and each evening of the conference, we're going to have a family share their journey. So I'm going to start today by sharing my personal journey. And so as many of you will have, who I've met have already known this, but so I'm originally from Houston, Texas, where I was born and raised. And growing up there, I lived down the street from my grandmother, who was a second parent for me. And as I was getting older, she started to develop symptoms and was eventually diagnosed with Alzheimer's disease. And during that time and over the subsequent years, I saw the effect the disease had not just on her, but on my grandfather and on our entire family. And I realized to me just how devastating it was, but also I hated that feeling helpless, seeing somebody you love so much slowly get worse and not being able to do anything about it. And that's ultimately what led me into medicine, into neurology. And so after 21 years of training in school, there we go, as the many fun years, I found myself, I was finishing up my neurology training at UCSF, where I did 4 years for residency and then 2 years of behavioral neurology specialty, which is focused on dementia. So I was engaged in clinical care and then clinical and translational research focused on neurodegenerative diseases like Alzheimer's disease. And I thought we had it all lined up. So Shan and I, after 6 more years of training, Shan and I had signed contracts. We were ready to go to Zambia. There I was going to be training neurologists locally. And then I was planning to accept a faculty position at an Alzheimer's disease research center, really to be able to focus on this throughout my career. I was like, great, 25 years later, finally ready to go. And then in 2022, our daughter Cora was born. And at the time, she had some clubbed foot, she had some hip issues, and we were told these are each individually treatable, but it's uncommon to have multiple of these. Why don't we consider genetic testing, which we were of course on board for. And we were told it'll probably be negative, maybe there's a connective tissue disease, but let's just see. And so it was at that time that we, when she was 6 weeks old, that we got the diagnosis of CLIFAD, that she had an ultra-rare severe neurodevelopmental disease due to a de novo or randomly acquired mutation in the NALCN ion channel that I had never heard of, and I wouldn't expect anyone to have ever heard of unless they've been affected by this channel and this disease. And so when we met with the geneticist, what they told us was, you know, we don't know much about this. There's less than 30 published cases. Of those cases, about half are unable to walk, about 3/4 are unable to talk. There's a high risk of early childhood mortality, and many require feeding tubes and breathing tubes to live. And there's no treatments, and good luck. There's nothing, that's all I know, that's all I can help. And it was devastating. I had a 6-week-old, and I had opened a bank account for a college savings account for her a week earlier. And so we started the journey that many of the families here I'm sure can relate to, where we started frequent medical appointments, many specialists, therapies, bracing for every day for the first 3 years of her life. And then at nighttime, I'd started reading, learning everything I could read about the channel, reaching out to all of those who had published and studied on it, really trying to learn where's the field out, what can we do to help? And I want to share what we learned then in 2022. So the first was that there was minimal active research on NALCN in the US. And as a parent, that was almost equally devastating. One, being told your child has a disease with no treatments, and two, everyone you reached out to said they weren't currently studying it. They couldn't find funding, they couldn't find support, and they weren't searching for treatments. And from a clinician and scientist perspective as well, that was awful, because I know just how important having that framework and body of research is to be able to develop treatments. Second, we are incredibly fortunate that there is an international consortium led by Dr. Arnaud Montiel and others internationally who had been studying the channel and the disease and had helped to understand the underlying biology of the channel and the mechanism of the disease, and had shown in vivo and in vitro models that treatments were possible if somebody just went to search for them. And so that was the first time I remember meeting with Arnaud, not too long after we got the diagnosis, and being so thankful that there was somebody, a scientific champion, working on this. And that was the first time that I had hope after the diagnosis that we could help to make treatments. And the third thing I learned is that for ultra-rare diseases, there are many that have developed treatments, but each of those has a strong parent-run, patient-run advocacy group behind it that helped to make those treatments happen. That brought the scientific community together, the families together, and the resources and support together needed to develop the treatments and to make it possible. And that's, these are just some of the success stories on there. And then at the same time, this was the same as well for the rare disease genetic treatment that was just administered earlier this year. That behind every treatment, there's a patient advocacy organization that helped to make it happen. And so we had no prior plans to, but that's really what led us in 2023 to work with other parents, and Suzanne Engel, who's here as well, to start Channeling Hope Foundation, really over our shared hope for a world free of these diseases. And so our mission is to build a global community of families and researchers to advance care and develop treatments for these diseases. And at the same time, our vision is a future free from the complications of these diseases. And one of the parents also shared that her goal was to show the many faces of the diseases, not just the 30 that were published in the literature, but just how broad of people can be affected by this disease, and to show the many other faces and sides of it as well, and to share the stories. And so we launched Channeling Hope Foundation at the first NALCN conference in Spain in 2023. And we went around and asked everyone at the conference, what can we do to help? How can we help move this research forward faster? And there's two major goals that were identified. The first was for generating patient-derived induced pluripotent stem cells, which are valuable tools for research, both to understand these diseases and to develop and test treatments. And so we started working on that with others, many others in this room, and happy to share that now there's 3 patient-derived iPSC cells developed for NALCN channel-related diseases, at least 3. One publicly available and hopefully many more in the future. And we'll have updates on these the presentations this afternoon and tomorrow afternoon. The second was the need for drug discovery and to develop treatments, and using high-throughput screening as one strategy to identify new treatments for NALCN-related diseases. And so we started a multidisciplinary international collaboration to work on this. How can we bring together experts in drug discovery and high-throughput screening to help us to find treatments? And so we'll be excited to share an update on our efforts on this tomorrow morning. And then I wanted to come back to our journey. So in 2022, when we first got the diagnosis, in many ways it felt isolating. Nobody knew about the disease, and it felt like we were just trying to stay above water and to keep from drowning. And by no lack of, people were amazing in trying to be helpful, but there's only so much help that can be offered. When we started working together with other parents in Channeling Hope Foundation, it was really meaningful, because finally we were working together to move forward. We were rowing together to helping to drive the research forward together. And so for us, and for me, that in many ways that was therapeutic. I no longer felt helpless. I felt like there was something that we could do together. It just organized our efforts to do so. And then we were really excited over the last year to be awarded a Chan Zuckerberg Initiative Rares 1 grant and to join the Rares 1 network to work with other ultra-rare diseases due to mutations in other related channels, as well as to join Combined Brain, where we could work together with other neurodevelopmental disease organizations, channelopathy organizations, to synergize our efforts in our research to move forward together faster. And that's been incredibly valuable to learn from others many years ahead of us, to share what did they learn, what worked, what didn't work, and how can we do this as for each of our organizations. So one of our major goals here in this conference is to ask for your help to chart the path forward and help us identify what's next. So we have each day we're going to have interactive workshops to brainstorm this and to ask for everyone's input here, the families, the scientists, everyone's input to help us understand really where are we now, what are the strengths, what do we know, what are the gaps, what do we need to learn, what do we need to focus on over and then to identify the priorities over the next 2 years for what we need to focus on. So this afternoon we'll have a workshop focused on the basic sciences, tomorrow afternoon on clinical and translational research, and Saturday morning on community building. We do plan to publish this in conference proceedings afterwards to help really just set the stage for where we're at now as a community, and anyone interested in working with us on that, please let me know. Then we're also excited to have a keynote speech from Dr. Fox, representing the CACNA-1A Foundation, both as a parent and as a scientist. CACNA-1A is another ion channel, very similar to NALCN, and mutations also cause ultra-rare diseases. And so this is an organization that in many ways has been our role model in doing all the things we'd like to do, and they've been doing it for far longer. So we'd love to learn from her about what they've learned within their organization and community, so we can take those lessons going forward. I also want to share, so another question we've had is, how do we get a ride on these large pharmaceutical companies, ships? How do we get them interested in NALCN and working with us to develop treatments for these diseases? And so I want to share some things that I've learned over the last 2 years now. I've probably had 30+ conversations with pharmaceutical companies, experts, representatives, just to try and learn how do we get them interested in NALCN and in related diseases. The first is how important it is that we grow and engage our community. The first question I get asked every time I meet with one of these representatives, how big is your community? How many people are there? And how engaged are they? And so we'd love to hear from you how we can help to build our community going forward. Second, how important data is. I get told data, 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 and asked what data do we have, what can we share? And the reality is the more data we have, the more of interest we are to these companies to focus on our diseases. And so that's a major impetus behind why we're launching the natural history study here, the biorepository here, the patient-oriented outcomes here, surveys here, to help us to acquire this data that's needed to drive treatments forward. And so when you're filling out those surveys late at night that ask you the exact same question in a slightly different way and are so frustrated by it, I'll just ask you to remember you're adding one data point that will hopefully help us build up this data that we can use to advance treatments. Sunday morning, we'll share as well our updates and our vision going forward for how we can acquire this body of data to be used for developing treatments. The third question I get asked is, if we were to develop a treatment, is your community ready to test it? What's called clinical trial readiness. And the reality and the answer is not yet. And so that's a major reason why we're launching the natural history study here, to understand the long-term course of the disease, which is needed to test if a treatment can change that course. One thing that we're also interested in is developing biomarkers, ways we can test the function of the channel and tell if a treatment is targeting it and if a treatment's having a positive effect on it. This could be in blood, it can be in EEG, in other ways. And so if we have a biomarker, it makes it much easier then to test treatments, because you're not waiting years to see if there's a long-term clinical change. You can test immediately to see if there's a change in that biomarker. And that's been used successfully by other ultra-rare disease groups to develop treatments and get regulatory approval much faster. And so I'm going to ask the scientist here to help us brainstorm. What are the biomarkers that we can acquire in studying these diseases, in ways that we can help to develop these and test these going forward? We're also going to be launching the biorepository Saturday morning for those who are available and interested, to try and start to acquire the samples needed to be able to test and validate biomarkers. And then last, we need to understand from the families, what would be a meaningful change to you? What's a meaningful outcome of a positive outcome of a treatment? And that's used by regulatory bodies to determine whether a treatment helps or not. And so we'll be launching a patient-oriented outcome survey as well. And if interested, asking for your help to share your feedback on what would a meaningful treatment would be to you. Another lesson I've taken from these conversations is pharmaceutical companies are for profits. And so we have to make a business case, why it's worth investing in our community and our disease. And so one of the areas to do so is by understanding the broader impact of NALCN targeting therapies. So I'm really excited for the morning lineup of presentations this morning that'll share much on the broad, much about the influence and impact of NALCN more widely as well, to help us to think about this going forward. We also want to raise the visibility and awareness within the community. If people aren't thinking about, if people aren't talking about NALCN and related diseases, they're not thinking about it. And if they're not thinking about it, they're not working towards treatments. So how do we help to raise the conversations and get people talking about NALCN and related diseases? I also want to share some inconvenient truths that I've learned along this pathway over the last 2 years. The first is that ultra-rare diseases are challenging to treat. It's not an easy path forward. Patient populations are very small and they're spread around the world, which makes it really hard to efficiently run the research studies needed to do so. And one reason we're trying to do as much as we can at the conference here. Second, families are understandably already overwhelmed with medical care, that asking them to put more on their plate, whether research or advocacy, is often too much to ask. And third, pharmaceutical companies are just less interested in general in ultra-rare diseases because they're so rare. And then there's the realities as well of just in general how difficult it is to develop a new treatment and drug. In general, bringing a drug to the market takes 10 to 15 years and costs over $50 million. And 75% of rare disease drugs don't make it to approval. And so we have to be willing to take the punches and just recognize that, but move forward nonetheless. So how do we defy the odds? How do we bring treatments to families sooner and more affordably? And it's really going to take all of us here working together. The scientists here today collaborating across disciplines and channels, sharing data, ideas, resources, tools to do so, having creativity and bold innovation. So we'll ask the scientists here, please meet each other, share ideas on collaboration, ideas on applying for grants and other fundings to do so. From the families, also moving forward together, participating in research as much as willing and able to help us to fuel the discovery and build that data that's needed. Engaging in advocacy and fundraising as much as able, and helping us to raise awareness of these diseases. And then by building our community as well. And so I'd encourage the families and scientists to talk this weekend as well, to share really what's your story and journey that led you here to this weekend, and what can we do to help you? Whether that's a family member, to a scientist, asking how can we help? And so finally, just want to share that's many of the reasons behind why I'm so thankful to see so many of you here, and though so many logging on virtually as well, because it really means the world to us to make this happen. I also want to ask that when you see all of the amazing research volunteers here this weekend helping us to making it possible to start these studies, please thank them. They're going out of their way to do so and to help us out, which I'm incredibly appreciative of. And also, please make sure to thank our sponsors and to visit the exhibits as well. We're really appreciative of them helping to support our conference and our ability to be here today. And so just to walk you through the schedule for the presentations today. So we have 2 full-packed days and then 1 half-day. So today, from the research family side, the natural history study will be ongoing in the morning. Then throughout the day, whenever you have the time to do so, we would encourage you, if interested, to complete the Matrix platform questions, as well as the Citizen Health, to access the Citizen Health. And then we'll have genetic counseling available for families interested in the afternoon. This morning, we'll start with the basic science sessions. And then we'll move towards lunch. We'll start with a translational science session focused on pharmacology. We'll then have a workshop helping us to discuss and identify the basic science gaps. We'll have a break in exhibits then. We'll have an afternoon session on translational science focused on disease models and tools. And then I'm very excited to share with all of you one of my favorite barbecue places in Texas. He said if you're coming to Texas, you have to get some Texas barbecue. And Rudy's is one of them, so we'll be there this evening. And also very excited to share with the families Morgan's Wonderland, which is, to my knowledge, the world's only ultra-rare, ultra-accessible amusement park meant for people of all ages and abilities here in San Antonio, and it's one of Cora's special places as well. On Saturday, for the family and research sides, we'll have the biorepository ongoing in the morning. So what we'll ask, that'll be from 6:45 to about 10:45 in the morning. We'll have phlebotomists here and a lab team here to do so. So for those who decide that to engage in this, what we'll ask is to just come down then. That'll be in the lobby area. And just as a reminder, if able, what we'd ask is that you fast overnight, meaning to have the blood draw before eating in the morning. Drinking is very much encouraged. Please drink all your liquids. But if able to tolerate having the blood draw before eating breakfast, that would be ideal. We'll have the natural history study available throughout the day, as well as, again, the matrix questionnaires and the Citizen Health platform and genetic counseling available throughout the day. So if families are interested in those, please chat with Emily about that. Tomorrow morning, we'll start with talking about the drug discovery efforts and collaboration and updates there. We'll then talk about some of the clinical research in understanding the symptoms of these diseases and advancements there. We'll have lunch. We're very excited to have Dr. Fox speak afterwards to us. We'll then have a workshop focused on the translational and clinical research priorities. We'll have a break afterwards. Then we'll have an expert roundtable where families can ask experts in the clinical care and research related to NALCN-related diseases any questions that they might have. And then we're excited to have poster sessions and exhibit session. And then we'll have dinner tomorrow evening at the SeaWorld Aquarium, not too far from here. Sunday morning will be the lighter day. So on Sunday morning, we'll ask for families who are interested and haven't yet to complete the matrix questionnaires, as well as to sign up for Citizen Health if you decide that's of interest to you. And then our program on Sunday will be focused on the community and community building. So we have a number of sessions there to help us chart the path forward. And very excited to have Dr. Lugwood speak to us about patient-oriented outcomes and how we can help to build these and understand these within our community. So I'm going to hand it off there to Dr. Montiel, and thank you all again for coming.
Good morning, dear families, colleagues, and friends. I am very glad to be here today. So I have so many things to say, but I have to keep on time. So I will start by introducing myself. So I am a permanent researcher at the scientific center, the French Scientific Center for Research, located in France, of course. Sorry. Okay, here. Not good. Okay, so I started to work on NALCN, a four-domain ion channels that belong to the same family as voltage-gated sodium and calcium channels. So in my group, we are 5 people. So Dr. Nataya, who is visiting professor from Thammasat University in Thailand, and Dr. Nataya will give a presentation on pharmacological properties of NALCN during this meeting. Also, there are 2 PhD students, so Alexandra and Marion, and 1 research assistant. So we have 3 objectives, basically. The first one is to understand the role of NALCN in physiology. The second one is to understand how dysfunction of NALCN induce a disease state in humans. And the third objective is to propose therapies for patients. So as a first speaker, I would like to make a short introduction about NALCN, of course. So basically, NALCN belongs to a multi-complex, a large protein complex, actually, with several proteins, FAM155A and 79 to KT, that are necessary for the functional expression of the channel, its cellular localization, and the stability of the complex. And there will be several talks about the evolution of this complex, the composition of this complex, and how dysfunction of some protein of this complex induce a pathological state in human. Basically, NALCN is mainly expressed in brain, as you can see here, but also in arteries, in heart, and endocrine tissues. About the contribution of NALCN to cellular stability, the first key paper that demonstrated the role of NALCN was published with Dejian, who is here today. So basically, in a very nice study published in 2007, so Dejian made a knockout mouse, and as you can see in the knockout mouse, we lose electrical activity to compare to the control condition, wild-type mouse, and the resting membrane potential of hippocampal neurons are more depolarized. And actually, the name NALCN was from Dejian, actually. Conversely, so Hiromasa Funato published a mouse model in which there is a gain-of-function mutation, and compared to control condition, as you can see here, you have an hyper stability, by contrast to here, and the resting membrane potential is more depolarized. So these two key studies demonstrated that NALCN is a key player in neuronal stability. What we did in the lab is to study what happened in endocrine cells, pituitary endocrine cells. So in a first paper published in 2021, we used a pituitary GH3 cell line, and we manipulated the expression level of NALCN, and we found an effect on electrical activity and on the resting membrane potential, similarly to neurons. And this year, in a collaboration with collaborators in UK, we were able to reproduce these results in pituitary, primary pituitary cells, as you can see here. If we remove NALCN, we lost electrical activity, and the resting membrane potential is more negative. What does it mean? It means that NALCN is a key player in cell stability, not only in neurons, but also in endocrine cells. In terms of physiology, so there are several studies using animal models, so mouse, Drosophila, C. elegans, snail, and naked mole rat. And just to summarize, these studies showed an important role of NALCN in different physiological processes, like fatigue control of locomotion, sleep, cyclic behavior. Regarding sleep, so Hiromasa will make a talk at this meeting. Also, sensitivity, to external applied molecule like anesthetics, so there will be a talk from Cheng Zhu. Also, role in locomotion, respiratory rhythm, pain nociception, parturition, so there will be a talk from Sarah about that, and gastrointestinal motility. So basically, the message here, NALCN is involved in many fundamental physiological processes. And more recently, sorry, Youngmin, because we know that NALCN is also expressed in arteries and in heart, but there was no data until very recently where Professor Youngmin, so he is here today, will give a talk because he found that NALCN is also involved in arterial contractibility. So unfortunately, there are mutations in NALCN and that impact its function. And considering the important role of NALCN in its physiology, of course, the symptoms are very severe. So the patients exhibit a large panel of symptoms with different severity. So there are two kinds of mutations. The first one are biallelic inherited recessive pathogenic variants. So these mutations are involved in the IHPRF syndrome and are loss of function. So we can anticipate a diminished electrical activity. The second kind of mutation corresponds to de novo pathogenic variants. So right now the consensus is de novo variants induce a gain of function on NALCN and induce a Clifford syndrome. Is it always true? I am not sure, and I will show you a study we made in collaboration with clinicians worldwide. So what we did is to implement a cohort of 35 patients with 28 variants. So the clinical part was led by Dr. Nawal. So she took care of the clinical part of the study. And in our lab, what we did is to perform functional recordings of the NALCN current, wild type or mutated. The objective was to understand how mutations impact the channel function. So just, I am not clinicians, but the key finding we found are for the 35 patients, there are only 4 very common phenotypes, basically global developmental delay and facial dysmorphism, and a large panel of symptoms that are different depending on patients. So basically the message here is there is a high variability in terms of the panel of symptoms expressed by patients and their severity. And what we did then is to determine if this diversity in symptoms can be reflected by diversity in alteration of the NALCN current. So what we did is to reproduce mutations found in patients in NALCN. Then we transfected NALCN with the whole HK cells, with the whole NALCN channelosome, mutated or wild type, and we use a patch clamp technique to record the NALCN current. We use the HK cells because these cells are easy to transfect, easy to record, easy to manipulate. You can see here a typical NALCN current here. This is a control condition, so no current. So these traces result from what we call a voltage step protocol. So from this protocol, what can we deduce? First, we can determine whether there is alteration in amplitude of the current. From that, we can deduce the current density and make what we call the intensity voltage relationship curve. We can also determine the sensitivity to extracellular calcium because this is a key feature of an NALCN. We can determine what we call the reversal potential, that is, that reflect the permeability of the channel. And we can determine modification of the decay kinetics. So even if you are not electrophysiologist, you see you have control conditions, the NALCN current, and you can see the shapes are different for mutants compared to the wild-type channels. And in one case, we do not observe any current. So for our analysis, so it was published, and we also published in the past that de novo variants of NALCN are gain of function. We express 15 variants, but only 5 are really gain of function. And the most gain of function one is a D600H. And as Jeremy told, we generated in collaboration with the Baylor College of Medicine, we generated pluripotent stem cells that are available if you are interested to use these cells. Unexpectedly, we found a group that is loss of function. It was absolutely not expected. So 3 variants in this case, plus the ones that do not give rise any current. It's a little bit strange for us because we know that patients with heterozygous for biallelic recessive variants are healthy. So why? These patients here are sick. One mechanism may be a dominant negative effect because we show the existence of that kind of mechanisms in a paper about the CAV 2.1 calcium channels a few years ago. And basically, these loss of function mutants are expressed. We can detect the protein by immunofluorescence using an antibody against NALCN, and the channel is tagged. This is a construct from the Stefan Place lab. Thank you, Stefan. So you can see the protein is expressed in all the cases. So the question was, is it possible that the mutated variant impact on the wild-type NALCN? So to do that, Last 2 weeks, Natalia co-expressed the wild-type channel with the mutated channel. And as you can see here, when the wild-type is co-expressed with the wild-type, we have a nice current that is calcium sensitive, so it's perfect. However, when we co-express NALCN with this mutant, loss of function mutant, we lose the current. So it means that probably this variant is dominant negative and inhibits the current from the wild-type channel. So it means that if we confirm that inhibitors of the NALCN current will not be relevant to treat these patients. And to my knowledge, this variant is found in 2 patients. So another case, we did not find any effect on the wild-type channel. However, for one variant, I322T, we found it is gain of function, but only in low extracellular calcium. So it means the calcium is, the inhibition of NALCN by calcium is very important in physiology, and actually this variant is found in 6 independent patients, and we generated a knock-in mouse model that exhibits very strong phenotype actually, but I will not tell more about it. And we also generated the pluripotent stem cells that are available. We also find another group, actually 2 mutants, 2 variants in what we call the selectivity filter. The selectivity filter is composed of 4 residues, E, E, K, E, and in this case, it is modified in D, K, E, and G, and E, so it means the ionic selectivity of NALCN is modified. This is what you can see here, the reversal potential change significantly. Interestingly, we observe a linear IV relationship for this variant here, instead of an inverted bell shape. So this is another group of variants, and it is also loss of function actually here. And also for the decay kinetic, we can define 3 groups. So first, in the past, we published that this kinetic is slower, but only 7 variants exhibit that. 5 variants exhibit faster kinetics, what never described before, and there was no difference for 2 variants. What does it mean? So we highlight a large heterogeneity in both the panel of symptoms exhibited by patients and their severity. And finally, patients with de novo variant do not systematically exhibit contracture of limb face, a typical mark of Clifford. So we have patients with HDD without Clifford, actually. And finally, de novo variants are not always gain of function. What does it mean? Now, we have to confirm what happened in native cells, actually in neurons, and what happened on electrical activity, but I think that each variant now should be functionally characterized in order to determine which therapy is the best. And also, do we have time? Yes, 1 minute. Just to say that NALCN is suspected to be involved in many other diseases like neurological disease, but also non-neurological disease and cancer. So Cheng will make a talk about neurological disease, and I think Eric will talk about cancer because he published a very nice paper recently on this topic. I will go through. So everything is possible only through collaboration worldwide, actually. I think maybe I forgot some, but for me it is important to go faster, to go further, to propose therapies to patients, to work all together, to collaborate. This is my feeling, and when I say all together, I'm not talking only about researchers and physicians, clinicians, but also with families. And I will stop here, and I thank you very much for your attention. 5 seconds for a question. Excuse me?
Are we allowed to ask questions?
But we can discuss later if you want. It will be easier. Very short.
Yes, one or two questions, if you don't mind. It's a great work. I just wonder, what do you think if we do the transfection of that ion channel in different type of cell? Because Would you imagine if putting that kind of a transfect in different type of cell, do you expect the behavior will be similar, like what you see right now, different variants over there?
You mean if we transfect in other cell types?
Yeah, like say in neuron cells or like in other, like say muscle type cells or cardiomyocyte, et cetera, because you mentioned that mRNA expressed in other type of tissues too.
Yes, but the difficulty is to transfect native cells. You know, the channelosome is pretty big, so it's very complicated. So we have to use viral vectors actually. So we were able to manipulate gene expression in pituitary cells, but I think we can do it also in other cell types for sure.
Thank you so much.
You're welcome. So now we are going to switch to a very important person in the field of NALCN, so Dejian.
Can I ask one more question?
Of course.
So some of the variants, I couldn't see the figure very well. There seemed to be a shift in reversal potential, but also there's a reduction in current. So wouldn't that be a mixture of gain and loss of function in the interpretation?
I would say it's the first characterization in HEK cells, okay? So we selected variants that exhibit very strange phenotype, unexpected, and we are not generating some animal models. So we will see what happens in native cells. If you want to. So Dejian, I am very glad to introduce him. He's a professor at the University of Pennsylvania, and he published very important key papers in the field of NALCN. So, for example, he was the first to show that NALCN is a sodium leak channel. It was a paper published in Cell in 2007. Then he was the first one to show that NALCN is both positively and negatively regulated by GPCR. So these are papers in Nature in 2009 and in Yuan in 2010. And more recently, so Dejian published a nice paper about pathogenic variants in UNC80 and UNC79. So two subunits of the NALCN channelosome. So Dejian, we are very glad to have the opportunity to hear you.
Thank you. I'm not going to talk from here, but it's like prompt because I'm supposed to have, I think, about 12 minutes. I should know where I am. All right. So which is forward? This one? Yeah.
Oh, okay, sorry. Pointer.
Oh, I don't
Okay,
need this.
it's good.
No, but this is the last slide. I have to go back to. How do I go back slides?
For me, it's here.
Oh, the receiver is there.
Okay.
All right, thank you. So thank you for the organizers, volunteers, and the patients, family. This has been really inspiring meeting I have been. So I'll talk to you about some of work we have done, especially the modulation of this channel. In the end, I'm gonna try to point out maybe that might be one way to, to develop some treatment, right? Since as Arnold mentioned, I have been working. I'm sorry. Okay, so I've been working on this channel for quite some years. So you can tell from my hair color. So I started this when I was a postdoc in the Children's Hospital in Boston. So for totally different project, I'm not even going into detail. But I know some people in the audience were also doing the same thing. Look for channel proteins that have similarity with the voltage-gated calcium channel. Okay, someone is here, calcium channel foundation here. So I discovered, well, I cloned several channels have similarity with voltage-gated calcium channel. All right, so the one that now called the NALCN is in the labs we call YB5. Just in case you don't like this name, this YB5 is even worse. Okay, so it doesn't mean anything because I cloned it from brain X, Y number, Y, 5 KB open reading frame. Anyway, it took many years to figure out. One other thing we have been looking for, obviously, is a complex, right? At that time, we knew calcium channels and sodium channels have oxygenator subunits, largely from the late Professor Catro's work in this country. Right, so we have a lot of, mainly inspired by work from the C. elegans and Drosophila, including May's lab here. Right, so the idea at that time, there were several genes, UNC79, UNC80, were shown to to interact with NALCN gene. Okay, so we say, well, these are possible, they're in the same complex. So we make, we make antibodies, we look at the brain, and then they see if they're in the same complex. I'm gonna just show you a couple of experiments. The first experiment we look at, just say number one, we co-transfect, UNC79 was published first. We co-transfect UNC79 with NALCN here, tagged. We asked, do they co-assemble with each other? The answer was miserable. No, we actually spent a lot of time. So it was selective results. That took, we stopped for about a year until the UNC80 publication was there. And then we tried UNC80. We say, how about UNC80? Again, it's a clone from mouse brain now. UNC80 actually interact with NALCN. So that's nice. UNC80 also interact with the UNC79. Then we say, well, if we put 4 together, is it possible? Everyone will interact with each other, which was the case, right? So that was kind of interesting. Then we proposed a model that UNC80 associated with NALCN and UNC79 serve as a bridge of the complex. Of course, that's before the cryo-EM gets so powerful, right? It's kind of interesting to see today, obviously, the more or less a case, right? So UNC80, you can see there's some contact with NALCN and also contact with UNC79 that form this complex here, right? So again, I'm going to skip most of work now, but I'm going to now focus on what is the function of UNC80? That's essentially what I'm going to talk about, right? So obviously, first you ask you what's really the NALCN function. So this actually, we got some answer from the expression data. This is a rodent blot I did. I teach undergrads at University of Pennsylvania. I asked my classroom, how many people have done Western blots? There'll be a few people with this. And then how many people have done Northern blots? Nobody knows. Yeah, wow. This is really old technique. Nobody uses anymore, right? So it just tells you how old I am. All right. But what's interesting is really this pattern. Essentially, it's all the neurons. Any neuron you look at, you can find some expression. So this indicates maybe some of the very basic conductance, very basic function. Of course, one of the very basic thing is really the sodium lead conductance, right? This is recorded, you can sort of imply from 1949 paper, Hodgkin and Katz. So obviously at action potential, you have voltage-gated sodium channels, voltage-gated potassium channel, that has been known. But we know there is also very small sodium D-conductance that was not really characterized well, and that was not known at that time. So we, again, long story short, we tested, is that possible? That's a D-conductance. The answer is yes. So I would say the most convincing data from us is really if you knock out NALCN, the TTX insensitive sodium D-conductance is gone. You have here from Arnold, this probably is also true in some of the cells she tried. There are several labs who have done this now, right? So you see it, essentially a linear IV curve, leak-like conductance. That's gone when you make a lockout. All right, so now, in the past some years, we have learned a lot from patients. Okay, so we know some of the, at least some of the variations now in patients. Many of them published, some of them are not. They're communicated with us, to us by doctors or patients' parents. So I'm gonna divide them into 4 groups based on what they do, okay? So there is a group of patients who carry truncation that very N-terminus. So it's basically the protein is gone. Those are now. You don't really see any function, right? So that's group 1. And then you have a group I will show you that reduce the currents, and you have a group that disrupt the inter-subunit interaction. And then you have another group that actually teaches a lot to us as a basic scientist, which basically shows that they disrupt the association, localization of this protein, right? So the first group now, so there are several families reported now. I should mention that all the variations are inherited. So there's no de novo mutations be found at this point. They're all recessive. So this is slightly different from the one Arnold talked about, and we can discuss why is this the case. Okay, so now we made the knockout mouse line to mimic this. It's a knockout from very beginning, I think R45 or somewhere. And you can record this current. We record from neurons. So this current is extremely small. In the neuron, whole cell is roughly about 10 picoampere. Again, this is true for people who record these currents. I think this is probably the reason why for 30, 40 years, this was not really recorded well, simply because size, right? Too small. Just to give you a sense, the voltage gated sodium channel will give 1 nanoampere, so it's 100-fold higher, right, current. So we use a trick, we record the current size by changing the bath sodium concentration from 140 millimolar to 14. We're not going to 0 because problems. All right, and then we record, Arnold just mentioned this current is also modulated by extracellular calcium. We drop calcium from 2 millimolar to 1.1. It's an increase of the sodium D currents. Now, in the uncatenated knockout, you do the same. There's a little currents, tiny remaining, and this is statistics here, right? So you can just say, well, another experiment you can do, can you rescue these experiments by transfecting uncatenated back to the knockout? You actually increase this current, so this one. Okay, this is statistics say there's some currents here in wild type, but if you knock out, currents essentially is very, very little. You can transfect them, okay, then there's an increase of current. This to me shows that the uncatenated is actually a limiting factor here, because when you increase uncatenated, you can use increase current. But another thing interesting here is that you can transfect NALCN, which forms a channel pore into this neuron. Those neurons should not have uncatenated, right? You can ask the question, can NALCN form channel without uncatenated? Right? The answer is yes, because you can see this current here. Right? This is clearly bigger than the knockout. However, this current is not regulated by calcium anymore. Okay, so we conclude that NALCN can form channels by itself, but they cannot be regulated. Some of you probably have read the recent paper from Ardo where it shows that, remember, NALCN is a very ancient channel, right? In fungi, some fungi do not have ANKT, they have NALCN. So it's possible NALCN can form a channel without ANKT. This also explains that if we see the patients, generally ANKT patients have slightly milder symptom than NALCN. Some of you probably already know, right? So, okay, so group 2. So some currents are, some variations reduce the currents. This is one patient with, I didn't say, but a doctor contacted me who were seeing the patient in Arizona. So he has intellectual disability and no speak. He has carried those 2 variations, okay? 340P, 341S, those are in the same area, and E1295Q. This was actually an interesting case that might be useful for you because this variation is also found in the genome AD database, which basically, that database is supposed to only list normal people without symptom. Okay, you can see several thousand people with this variation. So that makes diagnosis very hard. Can you say that your patient carrying this variation can have the phenotype because of this? Very hard. And then we say, well, it's possible because of this experiment. We did, okay, so this is somewhere around here now. We asked, what does each variant generate currents? So this is a wild type, okay, ANKT, well, I'm sorry. This is all done in the ANKT knockout background, mutant neurons. You transfect the wild type, you get this current. You transfect this E1295, you get this current. You transfect this Q340P, P341S, you get this small current. Now if you mix them, which mimic the compound heterozygotes, right? And you get a current something like this. Okay, this is much, much smaller than this each alone. Okay, this tells you if you carry 2 variations, each reduce the currents a little, and then it may end up with reducing currents by roughly, we did a calculation, roughly by 75%. We conclude it's possible if you reduce the current by 75%, you will see symptoms. However, we know 50% reduction you won't see symptoms. We have seen this NALCN, many NALCN patients, many ANKT patients. UNC79 is slightly different. I won't go into detail on this. Yes, question, right? Group 3, am I running short of the time now? 1 minute, okay. I can talk about this. So we have another patient that was published by Opel. So we realize this patient's variation here is in the domain that we found that interacts with ANKT, or UNC79. We asked, does this variation, why is that this patient has this phenotype, this homozygotes? I'm not going to show you this should not reduce the currents. However, this person cannot speak. He can walk, but he cannot run. Be able to walk is pretty significant for ANKT variations, right? So milder, and then we transfecting the protein to each other, and then I'm gonna really quickly escape this because I'm running another short of time. Basically saying that this variation reduced association between UNC79 and ANKT. There are probably several mutations, several variations in the list I have that probably does do this kind of work. Now, group 4. Okay, this is from 2 sisters that we came across from Norway. So they have intellectual disability, hypotonia, but they can actually sit without support. Again, this is not true in in patients with the Unedited now. They cannot, they have to have support to sit up. Okay, so it's slightly milder. The parents were amazing. They filmed those two sisters for more than 10 years. So if you start watching, you can see actually some of the developments. So this one, again, I'm not going to show you the data. It actually generates channel currents. However, if you look at the localization of this protein, so this is a wild type protein. This is a protein transfecting neurons. It's everywhere. It's in soma, in cell body, but it's also in dendrites. I don't know if there's an axon here. It's everywhere, right? But the point is that in this mutant, if you truncation here, now the protein is only in soma. So protein is not in dendrites anymore. This is interesting, right? Because many people believe that soma and dendrites are electrically connected. So why do you have to have a protein, this protein in the soma? But here it tells you, one, this piece of the protein is important, this segment, to maintain this protein everywhere. Two, you have to also report protein in different compartments. Okay, so I just have shown you that some of variants actually, by just characterize them, we understand maybe why they cause disease. But to us, it's also important we learn a lot from those variations, right? What the channel does with each component, right? So, but for you, most of you here are more interested now. So maybe this is also important because, again, I'm not going to show data here, but Unedited is involved essentially for many regulations of this channel by G protein coupled receptors, by peptides, for example. So I assume it's possible to target Unedited or interaction between Unedited and NALCN to manipulate the function of NALCN, right? This provides another revenue to addressing this issue, right? So I'm gonna thank people who did this work, but most of people probably you, but this is the only time I'm gonna actually thank my postdoc advisor, David Clapham, because I started this work in his lab as a postdoc. I did not get any credit for that for years, okay? But because I did not work on it, I work on this. Anyway, so now David is actually retired. His last work date is the end of this month, so happy birthday. So I should actually, in some sense, the patient families are probably the most inspiring thing you can find for basic researchers. You notice how devoted they are, and so what you work on actually is useful. Obviously, my work has been continuously funded by NIH for almost 30 years now. I hope we still have a government after this that can still support those work. Thank you so much.
Okay, thank you very much, Dejian, for this nice talk. Are there one or two questions?
Dejian, great talk. I'm curious of the class 1, where you lose UNC80, you no longer have the calcium-dependent regulation.
That's right.
Can you elaborate how that occurred?
Yeah, so that's a long story. So we think, okay, so for those who probably don't follow the story, the calcium is known to regulate this excitability of neurons or sodium D. This was shown for, again, 1940s, right? So but it was not really clear how this does. Most of you, again, if you're old enough, you have learned a phenomenon called the charge screening effect, which basically calcium is positive charge and binds the membrane. However, we found this actually calcium suppresses current by a G protein coupled receptor, calcium sensing receptor. And G protein coupled receptors is actually useful because dopamine receptors and GABA receptors, those G protein coupled receptors can also go suppress the NALCN via G proteins. So we suspect that all the G protein coupling requires uncoating. Now, how does that work? I don't really know.
Thank you, Dejan. Sorry, but we have to move to the next speaker, but we can discuss later. So I am very glad to introduce Professor Sarah England. So Sarah is professor at the University of Washington University School of Medicine. And Sarah made a nice work about the role of NALCN in the myometrial cell stability. So you published plenty of papers. It will be too long to make the list, so I let you show us.
Perfect. Thank you very much. Thank you so much for inviting me to be here. I am going to talk about NALCN in kind of a non-traditional tissue. I'm going to actually talk about uterine activity. So, which is the one that advances?
This one to move, this one to go back, and this one to pointer.
Yes. Okay, great. So before we start, I want to put everybody kind of understanding that uterine activity is actually very important. It's important throughout the reproductive lifespan. So it's important for a woman to menstruate, in order to become pregnant, in order for the pregnancy to progress, in order for labor to occur, in order for kind of remodeling of the uterus back to kind of the pre-pregnancy state. So there are some really key features of uterine activity. One is that it is rhythmic, so that if you take uterus out of the body, it will naturally contract, and it's very phasic in its activity. Second, it's myogenic, so it's actually controlled by the muscle itself. It's spontaneous, but you can also have agonist or some type of different ligands that can actually induce this, and this is actually true. As you can see from the top here, this is actually from a human. This is from a mouse. This is taken out of a mouse, and you can see it's very rhythmic. But also, excitability is always changing. So I got into this because I actually trained in the cardiovascular system, and I was really interested in I studied the pacemaking activity of the heart, and I just thought I could just transfer this to the uterus, but it's actually a lot more challenging because there's no central pacemaker. So if you look, this is work done by my colleague Yang Wang, is that if you look at the red is where the excitability starts. What you can see here is the excitability kind of at the start of the menstrual cycle starts at the top of the uterus and goes down. But when a woman's trying to get pregnant, the excitability of the uterus starts at the bottom and goes up. So this is really constantly changing. However, nobody really knows what starts kind of this signal. And I don't know if Dejan remembers, but I talked to him when I was at the University of Iowa, and we went to dinner together. And I was really interested in the sodium channel for one reason. So one is that there's kind of three main groups of ion channels in the uterus that modulate its activity. And one, there's actually a depolarization stage, which is this in the uterus, which causes a contraction, and this is caused by calcium channels. Then there's a lot of burst of the activity that maintains the contraction of the uterus, and then there's a repolarization that is caused by potassium channels. However, there is this pacemaking region that is noted here, and we thought that this could actually be the sodium current that could do this. So the reason that why this is interesting in the uterus is, is that you need to reach a threshold potential in order for the uterus to contract. So, and the threshold is constantly changing during pregnancy. So the uterus is very kind of quiescent to keep the uterus quiet during pregnancy, but as pregnancy progresses, it becomes less negative. So as I said before, depolarization is caused by the calcium, the repolarization is caused by potassium, and the pacemaker potential we thought could be would be due to sodium. Now, this was actually much more challenging in the uterus. One is nobody knew what this was. There had always been talked about sodium-based action potentials, but they're really very fast, like in neurons. But we have calcium-based action potentials in the uterus because the uterus is kind of a very slow contracting tissue. And however, it was known that in tissues that produce kind of these regenerative calcium-based action potentials, that sodium channels will provide the initial depolarization. And then also, it was known by many groups that there is a sodium channel current present in the myometrial cells, but nobody knew what it was. And Mel Soloff actually identified a sodium channel in a microarray that was in fact NALCN, but nobody identified it at the time. And patch clamp studies a long time ago talked about a gadolinium-sensitive current. So kind of with talking to Dejan and finally going to Arnaud's group in France a few years ago, we identified this as NALCN. So we did some initial experiments, Erin Reinl, when she was in my lab, looking at NALCN. We made a shRNA against NALCN, and we were able to knock it down in the myometrial or the muscle layer of the uterus. We could see that it in fact did decrease the current density. And also when we made a smooth muscle knockout we got a myosin heavy chain Cree that's used in the reproductive field. And we have what I call labor and delivery unit for mice, where we watch them every second of their pregnancy and their delivery. And what you could see is that when we knocked it out of the uterus, that you can see that they had abnormal partition, they had dystocia, they aren't able to deliver, their muscles not able to contract, and this was kind of a dose-dependent effect between the HETs, especially between the HETs and the knockouts. However, we were a little bit surprised in that when we did sharp electrode recordings to look at the activity, we saw that it was required for kind of how long the burst lasted, but it really didn't affect the pacemaker activity. And further studies by Chenwei Amazu in my lab, we expected that NALCN would be upregulated by estrogen, which is a procontractile hormone, but instead it downregulated it. So for the last few years, we've been working with Celia Santi's group, because we think that the complex in the uterus is actually a balance between two currents. Using a scrambled shRNA, we could see a potassium current that was dependent on sodium. It was a high conductance, about 78 picosiemens. It was activated by sodium. It had low voltage sensitivity, low sensitivity to TEA, which took out the BK channel, which is highly prevalent. And it was active at negative potentials, which is not as common in the uterus. And doing a lot of studies that I won't talk about today, we found that it was slow 2.1, which is part of the sodium-activated potassium channel current. Also, by doing different PLA, we found that these two were actually in a complex in the uterus. Now, most of my studies are actually in human myometrial cells. We get them from patients who provide a C-section sample. But the one problem is that you cannot get uterine samples throughout pregnancy, so we had to change to mice. And we were trying to determine whether the sodium signaling complex actually regulates membrane potential throughout pregnancy to actually control uterine contractility. So in order this, what we would figure out is if the sodium channels were open and NALCN was open, that it would activate these potassium channels. And this would put a brake on the calcium channels by causing a hyperpolarization. And once we got towards the end of pregnancy, that this would in fact actually change, and that we would not have association that we would lose this NALCN current. It would close these potassium channels, and then calcium could in fact open. So we thought that there were going to be changes across the course of pregnancy. And in order to do this, we actually have done a lot of studies on this, but I just want to talk mostly today about the sodium, this complex using DIS3. This is a common methodology that is used in the Santi lab. And what this does is that it allows us to look at the membrane potential across the course of pregnancy. DIS3 will kind of integrate into the membrane, and so when there's a when there's a hyperpolarization, you'll see a shift to the right. When there's a depolarization, you will get a shift to the left. And we use valinomycin as the maximal kind of hyperpolarization that we can get, so this will let the potassium flow into the cell. And so the way that we set this up is that we get the baseline levels, then we add either lithium to not activate the slow 2.1, or the sodium, which will go through the NALCN, and then activate slow 2.1, which will lead to hyperpolarization, and then we add the valinomycin at the very end. So when we look at the non-pregnant tissue, what you can see is that we do in fact get a hyperpolarization of at the early part of pregnancy, suggesting that we have more activation of slow 2.1. By the end of pregnancy, day 18.5, which is the end of pregnancy in mice, the mice deliver at 19.5, what you can see is that we in fact do get a depolarization. So what we see is that, and that this was only caused, the hyperpolarization is only seen when we add the sodium, but not the lithium. So really what happens over pregnancy is that we are getting more activation of the slow 2.1 kind of early in pregnancy, so more hyperpolarization. And this actually progresses, so this actually changes over the course, so that we're starting to see that this NAL slow 2.1 does in fact regulate the membrane potential over the entirety of the pregnancy. And you can also see this by control. If we knock out the slow 2.1, or we would knock out the NALCN, we kind of lose this regulation of the membrane potential. So, what we think is now happening is that external sodium regulates intracellular calcium homeostasis in the myometrial cells by when sodium can go through NALCN, it will activate slow 2.1, which will in fact shut off the voltage-gated calcium channels. And in fact, when you activate slow 2.1, it will, when this is inhibited or kind of separated for slow 2.1, it will allow the calcium to enter the cell. Now, we wanted to actually see if this will regulate intracellular calcium homeostasis. So when we added sodium, we can see that we don't have any calcium, we don't have any calcium peaks, but that when we remove the sodium, this is in fact depolarized, and you can see the calcium oscillations. And so, in summary, we know that inhibition of the NALCN-dependent sodium leak currents reduces the activation of the slow 2.1. These channels are in spatial proximity. They induce the hyperpolarization, and the modulation is bifunctional complex. And finally, we also know that we think that this channel is also regulating the proliferation of the uterus and how it grows during pregnancy. And these are our current studies that we're doing now, is to understand why this is so high early in pregnancy. So, and I just want to thank our group who has worked on this, and I'll take any questions. Thank you.
Thank you, Sarah. Thank you very much. We have time just for one question, quick question. No?
Was there change in sodium concentration in those cells, in knockout cells?
We did not measure that, but I don't think that there probably was a big change in the sodium concentration. Okay.
Thank you, Sarah.
Thank
Just
you.
a remark. Actually, we suspect that we have the same kind of regulation in some specific neurons, because when we knock out NALCN in some dopaminergic neurons, we saw a hyperpolarizing effect of NALCN. So
Okay.
what you describe it may be also true in neurons. Thank you.
Thank you.
Adriano, you are very welcome. So I am very glad to introduce Adriano. We met when he was a young PhD student in the David Spassford lab at the University of Waterloo in Canada. So at this time, Adriano was interested by NALCN, and he used a snail named Limneata stagnalis as a model, and you came to visit me in France, so I was very glad to have you. Then you made a postdoc at the University of Atlanta, I think, and you came back to Toronto to set up your own lab, and you are interested by the evolution of ion channels and receptors. So please. Please follow the time. Oh, yes. So to go up, to go back. So this is forward, and that's back. That's it.
Okay, thank you very much for inviting me here today and for holding this event. It's really amazing to be with individuals that are interested in this channel, both from a clinical perspective and from a basic science perspective. So I'm sure most of us here have seen these beautiful structures that have come out over recent years of the NALCN channelosome with these 4 subunits, FAM155, the NALCN pore-forming subunit here, UNC80 and UNC79. So it turns out previous phylogenetic analyses of these proteins had identified homologs of the extracellular subunit FAM155 and the NALCN pore-forming subunit in fungi, where they were independently discovered as calcium channels that contribute to calcium homeostasis in these various fungal species. In fungi, these subunits were called MID1 and CCH1. And these various studies essentially demonstrated the following. First of all, we know that NALCN and all 4 subunits shown here are present in most animal lineages. Animals are here in this box here with that little red dot. Ctenophores, the basal animals, lack all NALCN subunits. And the previous work demonstrated here in brown, it's a bit hard to see, but this node here represents different phyla within fungi. So these include yeasts, mushrooms, but also a bunch of pathogenic fungi that I'll discuss in a moment. And they found that NALCN and MID1, or the FAM subunit, were found in a select array of species. So to summarize, most animals possess all 4 subunits of NALCN. Fungi were found to possess their versions of these called CCH1 and MID1, but UNC79 and UNC80 were not identified outside of animals up to that point. So why is this fungal calcium channel relevant to humans? It turns out several studies have shown that the CCH1 or NALCN counterpart contributes to virulence to numerous pathogenic fungi. Starting with Cryptococcus neoformans, this is a pathogen that causes meningitis in humans and affects immunocompromised individuals. It's responsible for about 147 mortalities per year, and this number used to be much, much higher because of the advancements in AIDS treatment. So a lot of the people that were dying, upwards of 1 million a year, were immunocompromised. Candida albicans causes infections of the skin and the bloodstream. It causes average mortality rates of about 995,000 deaths per year, primarily from organ failure, sepsis, and shock. Aspergillus fumigatus and related species infect the airways, the lungs, and also can get into the blood and affect organs, and they cause about 34,000 per year, but it's actually more like 1.5 million when you account for comorbidities with other diseases. So fungal pathogens and the involvement of this NALCN homolog in virulence doesn't just apply to human health. It's been shown that even in pathogenic fungi of plants, CCH1 contributes to virulence. So Botrytis cinerea causes a condition in plants called gray mold decay. Essentially, it will pre and post-harvest cause major damages to crops, and it's estimated to cost about 10 to 100 billion US dollars per year in losses because of this. So clearly there's some interesting aspects about this channel, its contribution to virulence that make it worth studying and understanding better. This is also relevant because it's becoming more and more apparent that pathogenic fungi are starting to escape our treatments through antifungal resistance. So it means we're in a point where we have to start thinking of additional ways to mitigate fungal pathogens, and CCH1, which contributes to virulence, becomes interesting in that context. So just to backtrack now, I mentioned that these subunits were found in fungi, but there's been major advances in genome sequencing, transcriptome sequencing, and the availability of sequences for a vast array of species has increased dramatically over the years. So we wanted to re-explore the presence of NALCN subunits in fungi, but also broadly in eukaryotes. So we humans are part of a huge group of organisms called eukaryotes that includes plants, single-celled algae, vast numbers of species. And again, there's tons of sequences there. So what we did was we took manually curated sets of sequences for each of the 4 subunits. So NALCN, CCH1, FAM1 mid, UNC79, and UNC80, that we were very confident were real homologs. And we used these to train a hidden Markov models for each of them. So these are models that statistical models that can recognize patterns in gene sequences to tell you, yes, this looks like a NALCN sequence, or no, this does not, and similar for each subunit. We then compiled a series of proteomes, so all proteins from a species, for a total of 185 eukaryotic species. So spanning the eukaryotic tree, 185 species full proteomes. And we also took advantage of this fungal database called FungiDB, where we extracted 256 fungal proteomes. And effectively what we did is use these hidden Markov profiles to look for homologs of each of these proteins in those large numbers of databases. Once we identified homologs, we ran several analyses. First is an all versus all clustering analysis. Essentially, you compare each sequence you identified to the others, and ones that are similar to each other form little clusters, and you can see proximity to each other that way. We also ran conventional phylogenetic analyses to see the relatedness, and some structural modeling to see how these predicted identified structures look at the structural level potentially. So starting with NALCN, this is our cluster diagram of the NALCN search that we did in eukaryotes, those 185 eukaryotic proteomes. You'll notice in each color here represents a different kind of lineage within eukaryotes. They're all over the map here. But what I wanted to point out was that NALCN here in red, these are metazoan NALCN channels, and the fungal CCH channels are here. We humans and other animals possess 4 major types of 4-domain channels, which includes NALCN, but also we have voltage-gated sodium or NAV channels, Cav3 or T-type calcium channels, and Cav1 and 2 channels. And those form a tighter cluster more in the center, whereas NALCN and CCH1 are off on their own. More important is to do phylogenetics, though, because this tells you more about the relatedness of all these various proteins. And what we found was that there are 4 separate clades that include animal or human 4-domain channels. Over here is what we call clade A. There's the metazoan NALCN, and there's the fungal CCH1, but we see homologs in other eukaryotes that kind of follow the species phylogeny. So we think these are bona fide NALCN homologs, not only in fungi, but in additional eukaryotic taxa. Over here, clade B, this contains the voltage-gated Cav1 and Cav2 type channels, and these are animal-specific calcium channels, and there are some from eukaryotes as well, but they're nowhere near as kind of diverse as we saw the NALCN channel. Over here, the CAF3 or T-type channels, which also show animal presence and some restricted eukaryotic presence as well. And finally, voltage-gated sodium channels in clade D, which is a little bit broader, but not as broad as NALCN in terms of eukaryotic representation. When we look specifically in fungi, and we looked for the CCH1 channel in those 256 proteomes, we found that most fungi possess CCH1 homolog. So this is a phylogenetic tree rooted in the human animal calcium and sodium channels, and here are the human and basal invertebrate NALCN, and you see the sister clade relationship with fungal CCH1 channels, and these tend to follow the species phylogeny or the diversification within fungal phyla. The main point, though, is that most fungi possess a CCH1. Our analysis for FAM identified in the eukaryotic proteomes, here are all the animal FAMs, here are the fungal FAMs, and we identified a cluster in a lineage of very distant eukaryotes called Cryptista. These are a type of algae. And our phylogenetic tree, our phylogenetic tree here, shows the metazoan FAMs here, and then fungal and cryptist FAMs over there. When we looked specifically in fungi, we found that everywhere we found NALCN or CCH1, we found FAM or MID. So there seems to be direct correlation within fungal lineages where if CCH1 is there, FAM is there as well. We wanted to look at these proteins structurally with AlphaFold, this amazing program that's available now. And so what we did was we predicted the NALCN and FAM interactions for various homologs. So let's start over here. I'm sorry, I keep pressing that instead of the laser. So in black is the solved human structure of NALCN here and the FAM subunit here, and we've superimposed the predicted structure of very basal homologs from an invertebrate species called Trichoplax adherens. FAM is in red, the channel's in blue. Over here is the predicted structures of the homologs from Cryptococcus neoformans. That's that pathogenic fungus that causes meningitis. In brown is the FAM, and the channel CCH1 is here. And then these two are from two species of very distant eukaryotes called Cryptis. What we wanted to show here basically is that the FAM proteins are very, very divergent outside of these three alpha helices that were shown in structures, alpha 1, 2, and 3, that form important contacts with the extracellular surface of the NALCN channel. And our predictions showed across the board that those alpha 1 to alpha 3 helices were conserved in all of these identified FAM homologs. But outside of that region, FAM homologs are extremely divergent. There's very little homology across them. This is just to demonstrate this in a protein alignment. It's a partial one because the flanking regions we don't show because they're just extremely divergent. But what you do see is that the alpha 1, alpha 2, alpha 3 helices are indeed conserved across all these homologs. This is where things got a little interesting because I mentioned UNC80, UNC79 hadn't been discovered in fungi or outside of animals, and we found that we could indeed identify here the animal ones. We found some fungal homologs, but also broadly in other lineages of eukaryotes, including Unedita, Malawi monadita, Crumb, and Discoba. Over here is just the protein phylogeny of these homologs rooted in the metazoan UNC80. I mentioned in fungi, CCH1 and MID1 are present in most phyla, but that wasn't the case when we looked for UNC80. UNC80 was restricted to a subset of Agaricomycotina and Mucoromycota into these four phyla here. Our search for UNC79 was similar. I'll say that this was facilitated while we were doing this work by a paper I'm going to talk about shortly that identified an UNC79 candidate homolog in Cryptococcus. I'll mention that in a second, but guided by that, we conducted this analysis and found that UNC79 is present in animals, but we identified fungal and other eukaryotic homologs as well. But like UNC80, UNC79 is restricted to those same groups of fungi that also have UNC80. So in other words, to summarize in fungi, most fungi have NALCN homolog and the FAM homolog, but only a subset have both UNC79 and UNC80, and that's these groups here. So again, with AlphaFold, we wanted to predict the structures. Here's, sorry, I'm almost 2 more slides. So here's the channel predicted, the solved structure for humans. And these are predicted dimeric structures for UNC80, UNC79 homologs from a basal invertebrate, 2 fungi, and these 2 cryptis. So I mentioned that while we were doing this work and it was in review, a paper came out finding that in Cryptococcus neoformans, these proteins form a complex. They immunoprecipitated them together, confirming it's indeed an active complex in fungi. So to summarize, this was the previous understanding of where these subunits were, and our work now extends it deep into the eukaryotic phylogeny, beyond animals and fungi, over to this other clade called the Diaphoroticus that diverged from our clade of Amorphia about 1.5 billion years ago. So to summarize what all this is telling us, first of all, NALCN, FAM, UNC79, and UNC80 arose in very basally in eukaryotes roughly 1.5 billion years ago. Fungi, Agricomycetes, and Mucormycetes possess a full complement, and that includes some of those pathogenic fungi. Apusimonids also contain all 4 subunits. And also, I'd like to say our analysis likely missed homologs because UNC79 and UNC80 are highly divergent, so is FAM, so there's probably even more out there that we didn't discover. And just as a general point, this suggests that it might be useful to start integrating our thinking about NALCN in animals and CCH research in fungi. Fungi and yeast have been very instrumental for core cell biology, like Saccharomyces and Cerevisiae. So it may be useful to integrate the literature in our thinking about this complex. And with that, I'd like to thank our lab who contributed and our collaborators, including Arnaud Monté and funding. Thank you very much.
Thank you, Adriano. Just time for one question.
Amazing. Yep.
Homeostasis, that was really interesting.
Thank you.
Did they know the source of calcium homeostasis, like a ER or?
You mean
or
in yeast?
Yeah,
Yes, in yeast, it's a membrane calcium channel that helps fungi cope with stressful conditions and recuperate cytoplasmic calcium when there's low levels of calcium in the environment. But it's supposed to be at the membrane, but that doesn't mean it doesn't operate intercellular as well.
Thank you, Adriano. The next speaker is Cheng Zhu, Professor Cheng Zhu. Please. You are welcome to join us. So Professor Cheng Zhu is a professor at the Neuroscience Research Center of Anesthesiology at Sichuan University in China. So Cheng made nice papers to show the involvement of NALCN in neuropathic and inflammatory pain. You also show some effect of volatile anesthetics on NALCN currents. And more recently, you published a nice paper on the role of NALCN in depression-like behavior in a mouse model. So please, okay, you will have to, there is a timer here. Okay, here is to go up, here is to go back. Here, this is a pointer.
Forward.
Forward is here. No, sorry, forward is here.
It's back.
Back and thanks.
Hi, everybody. Thank you for the invitation, and it's very happy to can join the second international NALCN conference again. And we're from China, West China Hospital, and my lab is always about anesthetics and analgesics. So during the research time, we find certain link channel also involved in anesthesia and analgesia. So I talk about first of all, this is our basic science question in our lab, how the general anesthetics work. It's a very basic science during the neuroscience. And for this question, how general anesthetics works, we know the modern general anesthetics used in clinical for about 180 years, but we still do not fully know how they work. So at that time, and now most of the theory indicates that multiple protein targets can involved in the effects of general anesthetics. And why and how we find general anesthetics can modulate sodium link channel, because during one-time slice recording, we find the commonly used general anesthetics, voluntary anesthetics, not inhibit some neural spiking, but instead they enhance the neural spiking at some very low concentrations. So we're very interested to want to know why the voluntary anesthetics can enhance some neural conductance or neural spiking, because it's very important during anesthesia, because during general anesthesia, the anesthetics do not really inhibit all the neutral spiking. It's very important because as we lose of consciousness or some emotional memory, the basic physiology function should be maintained, like respiration, like pace of our circulation functions. So we found, surprisingly, voluntary anesthetics like isoflurane can enhance the conductance of sodium link channel. It's very surprising. So until now, at cellular level, we know that sodium link channel can help maintain basic neutral activity under voluntary anesthesia that make the neutral spiking is not very, very inhibited during anesthesia. So at the behavior level, normal function of sodium link channel can involved in many behaviors during anesthesia, such like induction recovery hyperactivity, the memory modulation during anesthesia, and just I mentioned the respiration will not be very inhibited with normal function of sodium link channel, and also some anesthesia and recovery from anesthesia. If sodium link channel no work well, the neutral spiking cannot recover very normally during recovery of anesthesia, so make the process of recovery of anesthesia very, very delayed. And except anesthesia, we also know some pain behavior change if we modulate expression of sodium link channel. For the last year, we know one of the main symptoms of sodium link channel mutant patients is a change of anesthetic sensitivity. So in the last year, there's a paper from anesthesiology found in the normal patients with different sensitivity to the most commonly used intravenous general anesthetics propofol. The dose requirement quite depends on the SMP of sodium link channel. That means even in normal patients, the function of sodium link channel can determine the dosage requirement of propofol. And the first time we continue talk about how sodium link channel involved in some neurological disease. How we found this change is not at the beginning we use a neutral inflammation model, that injection of AVTS in the mouse, and we found some very typical behavior change like depression or social deficits. And for this mice, we found the expression of sodium link channel in DG, that is dentate gyrus, is part of typical trace synaptic structure of our hippocampus. It's very critical for memory consolidation, emotional things. And the expression of sodium link channel in DG, both in dorsal and the ventral DG, significantly decreased. So this kind of change, just like our knockout or loss of function of sodium link channel in some part of our brain. So we use AAV to local injection to induce reduction of sodium link channel in ventral DG or dorsal DG respectively, and want to know what happened. As expected, when we knock down the expression of sodium link channel in ventral DG, it's significant. You can see the reduced expression, and at a behavior level, the mice significantly found to be depressive. All the behavior like tail suspension for swimming models, the typical animal model for depressive-like behaviors, all changed. And the overexpression of sodium link channel in ventral DG can fully rescue the AVTS-induced depression behaviors. For the previous slides, the overexpression of sodium link channel rescued the behaviors in slices recording, and this slice indicates overexpression of sodium link channel in ventral DG rescue the behaviors. So, and then what about the dorsal DG? If we use the same the model of chronic stress, it's very common animal model for social deficits. The chronic stress can induce social impairments combined with the social deficit behaviors and the reduction of sodium link channel in dorsal DG also. Sorry. And it's very nice to see when the animal do the social behavior task, the dorsal DG spiking quite increased. But if the after chronic stress, the neutral spiking of dorsal DG impaired and combined with bad behaviors. Sorry. It seems to work. Sorry. Yes. Then we also found if we knock out NALCN expression in dorsal DG, it mimics the behaviors of social impairments. You can see this is the expression of NALCN in dorsal DG, and these are behaviors associated with social impairments after knockdown NALCN in dorsal DG. Then we also use the strategy to overexpress NALCN in dorsal DG and to see if this can rescue the behaviors and the neutral functions after chronic stress-induced social deficits. As expected, we use an AAV dependent overexpress of NALCN. The behaviors of social cognition deficits fully rescued by this overexpression. I don't know. It's very delayed. And then at last, we want to see, to know what is a neutral projection or neutral circuits involved in this social deficits or recovery. We use optogenetics activation from the VDG to MS projection. This is a new projection we found from dorsal DG to this. So if we specifically activate this projection, all the behaviors rescued after chronic stresses. So I can remember of this part. If we mimic the NALCN patients, some behaviors, we knock out NALCN channels in somewhere. So we found it's very high sensitivity to general anesthesia, partly due to the enhanced respiration depression. And also, we can find if the sodium channel are normal during some brain, lower anesthesia can be found during voluntary anesthesia. That means if you want to enough analgesia, you need make more depths of general anesthesia. That's very dangerous. And this is for last time. Some sodium channel mutant can also induce abnormality of sensory function. And for this time, I just talk about specific neutral circuits involved in the emotion and the depression and the social deficits change after sodium channel knockout. Although we use a model like neuroinflammation or chronic stress, they are not very directly related to the patients, but we can know this. Like if we can really overexpress sodium channel in somewhere of our brain, or directly stimulate our brain or some projection, which is technically available, of course, if this kind of therapy can really rescue our behaviors of these patients. So that is our main purpose and ongoing researches about this. And then, last, thank you for our foundations, and also thank you guys for if you can find some chance to come visit our city, and it's quite welcome. Thank you.
Only one short question, if any.
So, very interesting talk. You refer propofol as one of the propofol may act on the NALCN. Have you and any other labs tested the propofol affecting NALCN current? Because it has been shown that it really works on the tonic current of GABA. And I wonder if the phenomenon actually is partially contribute to NALCN itself.
Actually, at the beginning, we test different anesthetics on sodium lake conductance. Actually, voluntary anesthetics like isoflurane, sevoflurane can directly enhance this conductance. Actually, we don't know why, because may be relevant to AT79 or EVG protein, I don't know. But when we test propofol directly, actually propofol did not directly enhance the conductance. So for that paper, I think sodium lake channel may modulate neutral excitability indirectly, and then affect the anesthetic depths or requirement.
What type of cell did you
brain slices.
I know, it's inhibitor neuron, etc.
Both, but for most of time we test on excitatory neurons. Yeah.
Okay, I want to add something. You probably are aware of the early work by Mike Crowder that they showed that the reason this UNC79 and UNC80 was identified in C. elegans, they're hypersensitive, even more sensitive to the volatile anesthetist. So that means it's not functioning directly on the sodium leak channel. It has to be enhancing some other channels. In the loss of function, they become even more sensitive. There's still remaining sensitivity.
I don't know, because actually we want to use a loss of function animal model to test this.
That's already done in C. elegans, loss of function. That's what I'm saying. It is important to test again, yeah.
As you know, our animal model is not directly relevant to loss of function. It's quite different.
In the complete loss of function mutants in C. elegans, they maintain sensitivity to this anesthetist.
Yes.
In that paper, I think it's loss of one factor is not necessary all complex.
No, it's loss of function of UNC79, UNC80. Yeah, so they're the fainter mutants.
That's still feasible.
It's possible. Everything is possible, but I'm just saying there's literature there. Maybe look into it first. Yeah.
Yes, yes, yes.
Thank you, Cheng. This is now time to close this session, so I would like to thank all the speakers, including myself, of course. And please be back at 11.
I'll buy you some cookies. I already finished. We have to get
for the second session. So the first speaker is Professor Youngmin Bae. So Youngmin, I was very glad to meet you in South Korea last year. And this is my pleasure to have you. So you are professor at, how do you say, Konkuk University, at the School of Medicine, Department of Physiology. So for many years, I was wondering what is the role of NALCN in arteries. And this year, you published a nice paper on the topic, so please. Also, do you have a timer? Forward, back, pointer.
From I'm Youngmin Bae from South Korea, Konkuk University in Seoul, and now I'm a visiting scholar in the USF Heart Institute in Tampa. I'm very happy to be here, and it's a great honor to show my recent study in an inspiring conference. And first, before I start, please consider that my English is not so good, and yeah, thanks for your understanding. Yeah. Today, I'm going to talk about NALCN beyond the brain. Actually, even though the NALCN channel's role in controlling the neuronal regulation is well known, it is because the NALCN can provide some inward background current, which is very important in controlling the excitability of neuronal membrane, because there is a lot of potassium or chloride channel for hyperpolarization of the neuronal membrane. So when the NALCN activity is decreased, the other hyperpolarizing current can be dominated, and in contrast, when this depolarizing current dominate, the excitability of neuronal activity can be overactivated. Both of condition is not good for our health. So the NALCN channel is well known, rich in brain, and it can provide background depolarizing leak sodium conductance, and it is blocked by extracellular calcium. However, the role in cardiovascular system is largely unknown. Actually, this is my first paper when I was a graduate student. I think I actually observed the NALCN-like current in arterial smooth muscle cells. At those time, I did not know about the NALCN, and I reported that it's a non-selective cation current in arterial smooth muscle cells. Yeah. So at those time, I found that the resting membrane potential from the pulmonary arterial smooth muscle cell was compared to other kind of muscle cell or neuron. It's very depolarized. It's found to be around -40 millivolt. This transient hyperpolarization is due to the transient activation of some kind of potassium current. However, basically the resting potential was somewhat depolarized. So I thought that there must be some kind of background leak inward sodium conductance contribute to this depolarized resting potential. And so when I reduced the extracellular sodium by substituting with some large cation and methyl D-glucamine, the membrane potential was dramatically hyperpolarized, strongly indicating that the sodium background conductance is functional in this kind of cell. So with the voltage clamp technique, I can found that there is surely some background sodium-dependent current, and it was effectively blocked by extracellular calcium or magnesium. So when we remove the extracellular calcium while we record the resting membrane potential, due to it can activate this background conductance, the resting membrane potential was remarkably depolarized. So after that, while I studying the pathogenesis of hypertension, I found that the resting potential from the hypertensive animal model was clearly depolarized. And in addition to the this depolarized resting potential, when we apply some agonists like serotonin, it induced more remarkable depolarization. And this voltage deflection is for measuring the membrane conductance by applying given size of membrane current. From this relation, we can measure the membrane conductance by applying some current. And we could observe that when applied with vasoconstricting agonist, the membrane conductance was increased because the deflection was decreased the membrane, which means that increase of the membrane conductance. So this is not due to the inhibition of the outward current, but due to the generation of activation of some inward current, which is probably like a sodium-dependent current, sodium current. So let's introduce about the aldosterone, because aldosterone is frequently related with the pathogenesis of hypertension. So aldosterone usually is very important in fluid homeostasis in our body. When our body loses our fluid, it increases the sodium leak channel in distal kidney and increase the sodium reabsorption and consequently water reabsorption again. However, in many hypertensive subjects, the aldosterone activity is too exaggerated. So too much water and sodium can be reabsorbed during, so this is why usually believed to the pathogenesis of hypertension in this subject. However, there is also the aldosterone receptor in arterial smooth muscle cells. So if this is functional, this sodium leak channel can be expressed in the arterial smooth muscle cell also. However, when we applied the sodium leak channel inhibitor like benzamyl or amyloride, there was no effect on this hyperactive arterial property. So we hypothesized that NALCN could be regulated by the aldosterone-like hormone, mineralocorticoid. So the same idea in the neuron. If aldosterone can upregulate NALCN expression in arterial smooth muscle cell, they can contribute to the increased excitability of arterial smooth muscle cell, and contractility is increased, then the peripheral resistance can be increased, which can be contributing to the pathogenesis of hypertension. So in this study, we focused on the role of NALCN in the pathogenesis of hypertension in arterial smooth muscle cell. And the specific goal of this study is like this. Can mineralocorticoid like aldosterone can upregulate NALCN in arterial smooth muscle? And is this upregulated NALCN can contribute to the increased arterial contractility and thus pathogenesis of hypertension? And is this renal mechanism dependent or not? And finally, if the NALCN channel is upregulated in hypertension, if we can knock down that protein, does it effective in preventing or rescuing the hypertensive animal? So studies like this, DOCA is one kind of mineralocorticoid, and by using this, we generated a hypertensive animal model, and we assessed the effect of NALCN siRNA ex vivo and in vivo assessment, and Western blotting, we assessed the NALCN protein expression, and when after unirephlectomy, the animal was fed with the salt solution and implantation of the DOCA pellet was applied here. And after 3 weeks, the animal was sacrificed after measuring blood pressure, and arterial contractility measurement was taken at this point. And for measuring the effect of the NALCN siRNA, 4 days before we applied NALCN siRNA, to the tail vein. So the one remarkable characteristic of the hypertensive artery is like this. In contrast to the control arteries, the hypertensive arteries show some spontaneous activity like this, contractility. And this can be effectively inhibited by lowering extracellular sodium concentration or by the L-type calcium channel inhibitor nifedipine. This indicates that due to the increased sodium permeability, this artery is depolarized compared to the control normotensive arteries. So in addition to the resting condition, the agonist-induced, like the serotonin-induced constriction is much more exaggerated in the hypertensive animal model. And interestingly, when you apply the low sodium concentration extracellular solution, in addition to the normalization of the resting contractility, this exaggerated agonist-induced contractility can be beautifully normalized, which is indicated strongly sodium-dependent inward current is responsible for the not only in resting constriction but also in agonist-induced constriction. So this data actually shows it is independent of the renal mechanism. When we incubate the arteries for 2 days in the incubator, and when we apply aldosterone together, the phenomena was similarly observed. So it was not dependent on the renal mechanism. And we could verify that in this condition also, the aldosterone treatment for 2 days can increase the expression of NALCN ion channel, and siRNA was very effective. So finally, we examined whether the in vivo knockdown of the NALCN channel with siRNA can rescue the hypertensive animal from that disease. And so this is the time course of the animal model. After injection of the implantation of the DOCA pellet, after 2 weeks, the blood pressure systolic is about 190, and it can be maintained. And when we apply here, the siRNA through the tail vein, after 4 days, there was a dramatic decrease in the blood pressure. And the protein expression of the NALCN ion channel was also decreased. This is a positive control experiment in brain. In brain, there was NALCN expression, and the siRNA did not knock down, because maybe I think that the siRNA did not permeate through the blood-brain barrier. So the increased arterial contractility in the hypertensive animal also observed, and after knocking down the siRNA using the tail vein injection, the arterial hypercontractility was also normalized. So interestingly, the increased NALCN expression was also observed in kidney, and it was also effectively blocked by siRNA. So even though the major mechanism was renal independent mechanism, I suggest that the role of kidney also very important in terms of NALCN regulation. So now we can answer the questions. Aldosterone directly upregulates NALCN expression in arterial smooth muscle, and this increased NALCN expression can increase arterial contractility and contribute to the pathogenesis of hypertension. Although this is basically non-renal mechanism, but kidney NALCN is also very important, I think. And knocking down of NALCN channel effectively rescue the animal from hypertension pathogenesis. So the conclusion is like this, and thank you for your attention.
Thank you very much, Youngmin. Any question?
has anyone looked at the NALCN heterozygotes knockout? They presumably would have half of the NALCN to have a lower blood pressure.
I don't know actually. Yeah. Yeah. Patient. Yeah. Yeah. So I'm still wondering. Yeah. So if there is any change in blood pressure in the gain of function or loss of function patients in NALCN, I'm wondering now. Yeah.
Okay, thank you, Young-min.
Thank you.
Thank you very much. So now I'm very glad to introduce Eric Harman. So Eric recently set up his own lab at the University of Minnesota.
That's right, yeah, just about a year and a half ago.
Yeah, and before that, you were a postdoc in UK.
Yeah, about 9 years in Cambridge.
Yes.
It was a good stretch. We got used to the UK lifestyle for a while.
And you published a wonderful paper in Nature Genetics about
That's
the
right,
role of
James.
NALCN in cancer. So
Thank you, Arno.
wonderful one. So you have a timer.
Yeah, I held on to my Midwest accent, yeah. Is forward,
back, and point.
just forward, back. Okay, well, today I'm going to talk to you a bit about what we're doing in my lab. I'm going to start the background about what I've done for the past several years during my postdoc when I was at the University of Cambridge and Professor Richard Gilbertson's group, and then what we've embarked on next. So this is my lab, and what we're really interested in is cell movement in the body. That we believe our organs are more mosaic, and there's a lot more communication between our organs than we really appreciate. And that when this movement is disrupted aberrantly, it can give rise to diseases such as cancer and fibrosis, which we'll talk a little bit about later. This is my lab here. I will talk a little bit about what we're doing individually in a bit. So also, just to mention that my lab, we use, for model systems, we use human cell lines, organoids, which are many organs in a dish, as well as a variety of different transgenic mouse models. So really to start off with, what was the major finding? I'll summarize 9 years worth of work in one slide. That metastasis, how cancer cells spread in the body, is not a phenomenon of cancer. It has nothing to do with cancer. It's really a cell traffic mobility problem, and depending on what the cargo is that you move. So what we used to discover that was a variety of different mouse models where we could lineage trace our cells that we've altered with mutations that are cancer relevant to cause tumors in mice. We focused on the gastrointestinal tract. And when animals had mutations in cancer genes in stem progenitors of the GI tract, they got tumors. Those tumors would metastasize at a certain frequency. When we layered on NALCN deletion, even heterozygote deletion of the sodium channel, or systemic treatment with gadolinium chloride, we could enhance metastasis 10 to 100-fold. What was very interesting, because my training is in developmental biologist, that's what I am, a developmental biologist. And so when I looked at this independent of oncogenes and tumor suppressors, we knew that lifelong deletion of this channel didn't cause cancer in mice. It didn't have any properties of what we think is a tumor suppressor allele. But what it did was cause mobilization of resident epithelial cells from one organ into the bloodstream to seed in a secondary organ, like cancer metastasis happens. But the result was that they didn't form tumors. So those cells incorporated into the tissues and adapted the architecture of that tissue. That this indicates that NALCN may not only just allow for the mobility of these cells, but alters what we think is the ground state or plasticity of the cells and their ability to adapt into the local environmental cues. So my lab is really at the really focused on understanding diagnostic therapeutics and interventions for cancer metastasis for targeting this channel. Can we turn down cell movement? But we also want to look at regenerative medicine. Is like, can we repair our organs better by enhancing mobility, by altering the fine tuning of this channel selectively? So I'll go through a bit of data about what gave rise to that story here, is that we identified initially that the NALCN is significantly mutated in a variety of human cancers, including gastrointestinal cancers. We saw that these mutations didn't really have a lot of selectivity. It seemed to pepper the whole gene throughout. We modeled it in silico using the cryo-EM structures that had been developed by a number of you in the room here. And we layered these mutations on and saw more condensed 3D enrichments within that structure of what we thought were loss of function based on a number of simulations. Now, we then went to the next trial. It's like, as a developmental biologist, I wanted to know, well, what is this channel doing? So instead of modeling mutations, we specifically went into deleting this channel in specific cell types in the body. To do that, we used the conditional NALCN alleles, either wild type, heterozygote, or knockout, shown here. Is the green, the laser? Yeah. And then we recombined or caused deletion specific cell types. Here we targeted the intestinal tract. In the middle, we targeted prominent 1, which marks stem progenitor cells body-wide. But if we give them the right mutations, we tend to bias towards gastrointestinal tumors. And then we also modeled pancreatic cancer here with PDX1, recombinase. These are human-relevant mutations. And then importantly, we use this green fluorescent reporter in order to follow the cells that we cause recombination in. When we did this, all the animals, they generated tumors, as we'd predict, because we gave them cancer-causing mutations. Layering on NALCN deletion had no impact on the multiplicity, the severity of the primary tumor itself. We didn't see any vast changes in general architecture of this tissue. What we did see from transcriptomic profiling was enrichment in themes of epithelial to mesenchymal transitions, so a change in cell states that allow them to be more migratory. And when we looked at the distant tissues of those same mice, we started seeing green lesions here that we shouldn't be seeing. So in the context of pancreatic cancer or intestinal cancer, we shouldn't have any green recombination or green cells in those organs because we've targeted other organs. And when we look at them histologically, they had tumors. Those tumors looked like the primary tumor the animals had. Because we could see it, we could count it. We could see that when we deleted this channel, either heterozygote, in most cases even heterozygous is worse off than a knockout, we saw way more metastatic disease happening and spreading to selective different organs. So loss of this channel genetically was enhancing more metastatic disease. And one way to do that is to pump more tumor cells into the blood to get around the body. And when we looked in the blood of these animals that had NALCN deletion, we saw that, one, we could identify circulating tumor cells. Those are cells that leave a primary tumor, go into the blood, and can be the source for secondary disease. I can find them in blood films here. We could count them by flow cytometry. And that just generally, if we delete the channel and or treat animals with gadolinium, low doses chronically, that we can enhance the presence of green cells in the blood. These green cells are the cells that we targeted initially. We could profile them, we captured them for transcriptomic profiles. They look like human circulating tumor cells. So what we're putting into the blood could be circulating tumor cells. Ultimately, you do a test where you take an animal model and you deliver these cells from our tumor-bearing animals from the blood that we isolate. We inject them via tail vein, and they form seeded metastases in distant organs. So by and by, these cells that are being, at least some of these cells being pumped into the blood have circulating tumor cell properties. Now, at the same time, I was wanting to know, what is this channel doing in normal development in these same tissues? So I ran the same experiments, same recombinases, but this time I didn't include KRAS P53 cancer-causing mutations. We aged the animals for 2 years and never saw tumor formation above a background rate. There was an inherent spontaneous rate. Didn't see that. What we did see, what was very exciting, were two things. One, in our model, even in the wild-type animals, we could detect rare green cells in the blood, meaning that cells are trafficking at very low levels. And this is basically at the threshold of detection. We're basically at the point where it gets uncertain if they exist. So it's an extremely rare event. When we delete the channel, or even treat animals with gadolinium, we see a log-fold to 2 log-fold increased presence of cells in the blood of these animals that are green. And when we look at it, we can see by immunofluorescence that they have markers similar to their tumor counterpart. So ultimately what we did, there's 2 experiments, I'll just tell you about the ultimate one, is we harvested those green cells in circulation of the non-tumor animals and implanted them into recipient animals. And we could see that we could detect the cells over 74 days later, and that they contribute to various architecture. We saw that the kidney seemed to be the most affected, where we saw these cells that came from the gut go to the kidney and contribute to structures like renal tubules, as well as some glomeruli. Although exceedingly rare, it wasn't that it was robust, it was very rare, but it could happen. Furthermore, we saw one animal, for example, some large architectural changes happening in the kidney. Now, you're probably wondering, well, what happened to the animals if we aged them for 2 years? Well, after about 1.5 years, many animals developed systemic fibrotic diseases, predominantly the kidney and the skin were affected. And this actually was reminiscent of a human syndrome called gadolinium-induced systemic fibrosis. Thankfully, it's all been eradicated because gadolinium is one of the compounds that forms a backbone for MRI contrast agent. And the original contrast agents were in a linear form, and free gadolinium radicals could spread out. No one really knew what caused that disease other than gadolinium. And that when they changed the compounds, this terrible systemic disease has all been eradicated. But we think we found the genetic context behind this because the pathology that we're seeing when we worked with experts in the field said it looks just like what they saw in their human patients. So we think that NALCN is one explanation for this systemic human disease. Now, at this point, I'd wrap this up and I moved back to Minnesota. So I'm originally from Minnesota. Started here at the Hormel Institute, Hormel at the University of Minnesota. We have a site 90 miles north of the Twin Cities near the Mayo Clinic. And we're funded by Hormel Foods. And that's why there's a spam can there, because across the street is where they make spam. And those monies come in to help support our institute. And this is my lab down here. And I'll tell you a little bit about when I was moving to start my lab, I wanted to know, is there more broad applicability of the function of this channel in other epithelial cell types? And so I came across really looking into mammary epithelium and breast cancers. And we saw that essentially NALCN can be deleted in a variety of different subtypes of breast cancer. It's not lost. We never see it completely gone. It seems to be either diffusely spread apart or just lowered in expression. When we bin this, we see that patients that have copy number loss or reduced expression of this channel have a worse off disease with more incidence of lymph node metastasis. And thanks to Sarah, it gave me some confidence because NALCN is estrogen and progesterone responsive, which is a key hormonal signaling mechanism in breast cancer and breast cancer progression. So putting it together, we decided to go down that route. Well, first off, we need to know, is it functionally expressed? So we did some immunofluorescence with NALCN, and we saw that at least in immortalized human mammary epithelial cells, we could see distribution of NALCN in green here, and in MCF7 ER-positive breast cancer, we also see presence, but you can see that different distribution patterns are occurring. When we target for suppression, we can target suppression of this channel, and then working closely with Sophie Biosciences, actually, about a year ago, we spent months together optimizing how we can actually isolate this channel on that cube compact that they have out there that they're displaying. And we're able to do it to adapt the platform to these cell types that they hadn't looked at before. And that took quite a while to get the resistance. But once we got that, we adapted the protocols that many of you have developed to isolate this channel and applied them here. And we can see that when we suppress this channel, we lose the current with the short hairpin, suggesting that we have been able to target it, but more importantly, that it is actually expressed in these cell types. All right, so we're trying to figure out now mechanistically what's going on. Like, what would this channel do to allow for cells to mobilize in the body, to move around? And there are lots of routes we could go, and I'm going to just tell you about one of them. We were looking at transcriptomic data, and we started constantly seeing signatures around this structure called primary cilia. A cell often only ever has one of these. It's a sensory cilia. It's not the motile ones. It's the ones that sense what's happening in the surrounding environment. And I'm fortunate enough at my new institute that there's this gentleman here, Sergio Bradalone, who's an expert in primary cilia. And really, cilia are hubs for taking signals and having transduction of these signaling cascades through in order to allow for cell change dynamics, such as loss of primary cilia can lead to migration changes, proliferation, invasive phenotypes in cancer. This has predominantly been known in hepatobiliary diseases. There's a little bit in breast cancer, but the data is a bit murky about what it's doing and when. So we looked at our cell types. We could see primary cilia is there, but we could see, more importantly, here's NALCN in green. You see it's in lots of different compartments, but one compartment it overlaps with is where a primary cilia form. This is a gametulium, which marks the basal body for where the primary cilia starts and extends from. We know that we can suppress the channel, and when we do so, we could measure things like if NALCN is important in primary ciliogenesis, loss of this channel should have some impact in either the frequency that we see this in the cell culture and or the length. And when we looked at our data, we could see that when we suppress the channel, we saw a reduction in the number of cells, or the percent of fields where we have ciliated cells. And those that were ciliated were actually shorter. You can see here, here's what a standard primary cilia would look like. Here's an example of a cell that had a short hairpin suppression. You can see it's even kind of clubbed and misshapen. Those are indications of a diseased type cilia that could lead to a pathogenesis. So beyond epithelial cells now, we started noticing that this channel is expressed in other cell types in the body, in the periphery. And working with Ningling Kang, we came across that NALCN seems to be expressed in something called cancer-associated fibroblasts. So colorectal cancer, an epithelial cell type, can spread throughout the body and seed in organs like the liver and can grow out. That's the number one cause for colorectal cancer-related death, is metastasis to the liver. We saw that by immunofluorescence, NALCN in green colocalizes with alpha smooth muscle actin, a marker of cancer-associated fibroblasts. What we did then was looked at, well, is it expressed in a cell culture-based system? Can we get some confidence that's important in this conversion of a hepatic stellate cell to a cancer-associated fibroblast? And you can do this by treating cells with TGF-beta. You can see here's just examples of NALCN expression in culture, but this is the ultimate experiment, is that we could take these cells, treat them with TGF-beta. They should have these markers like this. Here's smooth muscle alpha actin. Here's the control, beautiful. We had a full collapse of the expression of smooth muscle actin, so loss of this channel inhibited that change. Now, we also saw in other cell types beyond colorectal cancer, so I'm just going to highlight a couple of the other ones. We saw its impairment in angiogenesis, where when we're looking at primary tumor models for metastasis in our model system, we saw that the channel is also expressed in endothelial cells, which would be one route for the tumor cells that are surrounding this to get into the bloodstream. And I started wondering, well, maybe if this channel is important in the maintenance of this and can actually have two cells working together to let cancer spread. And then this very talented summer student from Gustavus Adolphus came to my lab this summer, and what we found is that in an angiogenesis assay in a dish, right, you put in these endothelial cells, you treat them, and they form these nice, beautiful networks that when we suppress NALCN, we compromise their ability to form angiogenic structures. We've now started both the colorectal studies as well as this study targeting mice. We've already bred the floxed alleles that we used for the other studies, 2 different Krebs combinases, so we can start studying the role of this in physiology of these tissues. And lastly, I was a developmental biologist, but I spent a lot in neurobiology. Arno convinced me to share this data that we hadn't published. So I looked at cerebellar development. So cerebellum develops predominantly postnatally. And so example of what cerebellum would look like on an individual that's just born. You see these cells in the outer layer. They have to migrate inwards in order to form this structure. And we saw that when we postnatally delete NALCN using tamoxifen-inducible nesting CRE2, we saw these rests form. These means that there's stalled migration and differentiation of the cerebellum. 75% or 25% of the mice developed ataxia and seizures when they were aged to about 5 months. And we could see actually massive changes in even heterozygote animals as well. So it had to be as a dose response mechanism. We saw that massive altered structures also within the cerebellum of these animals. So just one last plug. At the Hormel Institute, we have developed the Minnesota Bioimaging Consortium because we want to understand how proteins taking from the tissue level or cell or tissue level all the way down to the atomic resolution here, which we can do now with all these new equipment in our institute. We've tagged NALCN with GFP, and we're going to look at the subcellular resolution so we can see who its interacting partners are in cell type specific contexts. We'd love to collaborate. We're opening up this is just recently launched initiative, so if anyone's very much interested in cryo-EM tomography, let me know. So lots of steps next. I'll just end there. I'm happy to talk with everyone about what we're interested in and just leave it on with my lab and collaborators. Thank you.
Thank you for this long presentation.
Yes.
Is there any question for Eric? I should stay here, actually.
So, Eric, great talk. I have 2 questions. The first one is that The resting membrane potential is critical for the cell cycle itself. I'm wondering, is this actually can be dated back, movement or metastasis phenotypes are actually due to more or less stem cell produced. As a consequence, there is this over. So this also relate to the cerebellum problem that you described. There's clear sort of lamination of different layers lost. I'm curious if this is actually a stem cell problem.
Yeah, I think it is. I didn't want to make this our talk for argument's sake, but what we are seeing is that when you target specific stem cell populations, the daughter cells, their proliferation rate, they don't seem to be impacted. But we're seeing some expansion of stem cell pools in the gastrointestinal tissues, and thinking that might be what happens in other epithelial cells as well. But the end result in vivo, as we get a mass metaplasia, that's happening, at least in the stomach, so we've seen not only more stem cells, but these stem cells now are inappropriately deriving daughter cells that shouldn't be there. We don't have a lot of strong evidence. I've seen proliferation aspects like Sarah has in organoids in a dish and culture of the expansion of them, or neurospheres, we see exhaustion when outside sleep. But in vivo, it seems to be a different complex. And the proliferation, at least in the model system we're looking at, the guts, has not been as bad.
Okay, thank you, Eric. So next speaker is Professor Hiromasa Funato. So Hiromasa is co-PI of a very important team at the prestigious International Institute for Integrative Sleep Medicine in Japan, University of Tsukuba.
Yes.
And you are a specialist of sleep, and you published a very nice paper a few years ago about NALCN in sleep.
Thank you very much.
You have the time there. Follow the time.
Okay. Oh, thank you. So, yeah, I'm Hiromasa from Japan, Toho University and Tsukuba, University of Tsukuba. So thank you for organizing this fantastic meeting. So in a previous meeting 2 years ago, I hoped to attend, but university, so I can not get the permission from the university still in the influence of COVID. So I really appreciate to be here and talk. So I'm going to talk about NALCN, the role of NALCN in REM sleep regulation. So I'm not an electrophysiologist or I don't have a strong background on ion channel, but what led me to NALCN was sleep. So let me begin with the basics of the sleep. So we human repeated wake and non-REM and REM sleep cycle, as you know. So during wakefulness, the brain is very active. So the EEG, electroencephalogram, shows very low voltage and fast activity. That's very characteristic EEG during wakefulness. So during wake, we are conscious and do move anything like eat and talk and walk. And during sleep, so sleep is classified into two, non-REM sleep and REM sleep. REM sleep accounts for the majority of the sleep time, 70% or so. So during non-REM sleep, EEG pattern is totally different from that of wakefulness. Large voltage and slow frequency. That's for the non-REM sleep. The REM sleep is totally different in terms of brain. So that's why REM sleep is also called paradoxical sleep. The brain activity is very active, and EEG pattern is similar to that of wakefulness. So totally different from the non-REM. So non-REM sleep and REM sleep are totally different. So this cycle, this two-stage sleep occurs before birth. So even human fetus have two sleep states. It's called quiet sleep or non-REM, active sleep or REM sleep. So those two sleep stages are classified according to body movement, eye movement, and heart rate. So usually it's impossible to record EEG, EMG, or EEG from the human fetus, but special apparatus used during delivery show that even before delivery, the fetus have a two-stage in EEG. So you can see the higher amplitude in green. This is correspond to the non-REM sleep, quiet sleep. Then in the red indicate the active sleep or REM sleep. So this is non-REM and REM, non-REM and REM. So even before birth, two-stage shows clear cycle. In other words, machinery for non-REM and REM sleep cycle already developed prenatally. So usually heart rate without EEG, heart rate variability shows the presence of two stages of sleep. So during REM sleep, heart rate change goes up and down, so very variable. But during the non-REM sleep, heart rate is very stable like this. So depending on the heart rate pattern, you can see the REM sleep and non-REM sleep, even in the fetus. But one case, so in this case, there is no REM sleep-like active phase, or heart rate is very stable, so never goes up and down like this. So this case is shared by the Dr. Ikeda. So Dr. Ikeda is a professor of the Department of Beijing Mie University, Japan. So he's here today, and anybody can talk him to this case in detail. The point is, so after birth, this fetus shows several severe symptoms, then perform whole genome sequencing to identify mutation in the NALCN gene. So the infant was diagnosed with the Clifford syndromes. So, but as a basic literature, we use mouse for the sleep study. So good thing about using mouse for sleep is very easily stage wake, non-REM, and REM, depending on the EEG. So the presence of hippocampus theta during REM sleep so you can see very homogeneous wave during REM sleep like this. So frequency is 6 to 9 peaks. So Now this mouse is in REM sleep and woke up, then it has fallen asleep again, returning to non-REM sleep. So depending on the EEG and EMG, very easily to stage wake and non-REM and REM in mice. So using this system, we took a whole genetic approach to understand the molecular genetics of the sleep regulation, to find a novel gene regulating sleep. That's a major purpose. So he is Masashi Yanagisawa. So he was a professor in the University of Texas Southwestern, so UT Southwestern in Dallas. So at that time, his lab, 25 years ago, discovered orexin. So orexin is a major neurotransmitter for the sleep and wake regulator. And now orexin receptor is a major target of sleeping pill. So he got good, large amount of money to start a genetic study in mice for sleep to identify novel gene in Japan. So I was involved from the beginning. The scheme of the forest genetics is here. The first thing is to inject chemical mutagen, ENU, to the male mice. Then we look for any sleep abnormality using EZ-EMG, the next generation, F1 generation. If we found some abnormality, we proceed to the next generation to the sleep abnormality is heritable or not. So the idea is very simple, to make novel heritable sleep abnormality, sleep disease in mice, then find the causative gene or mutation. So this scheme really works, but one exception is a pedigree we call the dreamless. The founder of this pedigree shows the very short REM sleep episode duration like this. So this phenotype was inherited to the next generation. So the half of the return age shows very short. Then we performed chromosomal mapping and for exome sequencing to identify the mutation in the NALCN gene. The mutation we found was result in the amino acid substitution at the asparagine in the domain 1, segment 6 to lysine. So this mutation incorporated in the fresh BRAC6 mice to reproduce this phenotype. So the mice shows very short REM sleep episode duration and total or daily non-REM sleep time, while maintaining normal wake and non-REM durations. So these mice also showed decreased theta power and increased delta power during REM sleep. And when this mutation expressed in the HEC293 cell with UNC80 and SERC, they show higher conductance. So we continue this screening. Then we found another pedigree very similar to the phenotype. So that we named this pedigree Dreamless 2. Then we performed several similar processes to identify another mutation in NALCN. So in this case, the mutation was found on the domain 3, again, segment 6. So similar position of the NALCN result in a very similar phenotype in REM sleep. So because brain is very complicated structure and composed of different type of neurons, and NALCN is broadly expressed. So we like to know which brain region is required for the normal REM sleep, depending on REM sleep. So when NALCN is deleted in the cortex using EMX1 query, so you can see a blue NALCN deleted region. So those mice shows very frequent switch between non-REM and REM sleep like this. And REM sleep is very dirty because of the presence of the delta power and decreased theta power. So this is similar to the original dreamless, but phenotype is more stronger and pronounced. Sorry. So we use different variety of gray line to delete neurons, region-specific way. The most pronounced result is the EMX1 query mutant shows very short REM sleep episode duration and daily REM sleep time. So this is done by Tomoyuki, so here, and he's the next speaker to me, but he going to talk different topics. So in summary, so loss of active sleep during pregnancy led to identify an NALCN gene in human. And similarly, forward genetics in mice repeatedly demonstrated NALCN plays a critical role in REM sleep regulation. NALCN involved in sleep in the cortex is important for or required for the normal REM sleep regulation. And hopefully genetic study in mice may provide but insight into human NALCN and related diseases. So lastly, I'd like to thank all people who involved in this study, especially for Masashi Yanagisawa. He involved me to this for genetic project and Tomoyuki Fujiyama. He has been working on this NALCN project from very beginning, early stage. So thank you very much.
Thank you very much for this great work. Is there any question?
I don't need this. So, okay, anyway, so those mice are really interesting. Do they, besides the sleep phenotype, I know you have some memory phenotype, do they live the same, do they have a normal lifespan?
We didn't lifespan study. So we after we use for EEG or those sleep study, we suck mice, so we didn't know. But as far as we observed, they look basically healthy. Without EEG, EMG recording, we can tell which is normal, which is mutant.
No seizure or anything? No seizure or anything?
No
seizure.
Seizure.
Yeah, do you have epilepsy?
No. As far as we recorded EEG, EMG, so no conversion, no epilepsy, but a little bit of abnormality in EEG, but very subtle. No clinical epilepsy.
Okay, thank you. Thank you very much.
Thank you.
So next speaker is from the Hiromasa lab. So how do you pronounce? Tomoyuki Fujiyama?
Yes.
Okay, thank you. So you are assistant professor in the Hiromasa's group, and you are looking for binding partners and regulators of NALCN related to sleep. So we are very glad to hear your talk. Please, there is a timer here. See if you can.
I understand. Thank you. Thank you for your kind introduction. So I'm so excited to be here because I'm facing the very great research as a study on the NALCN things. So thank you. So I'm Tomoyuki Fujiyama from Yanagisawa Furniture Laboratory from Japan. So I'd like to share my ongoing recent research on the proteomics. So how can I change the slide? Okay. So in 2 years ago, so we were not there. So, but finally we could be attending here. So thank you for organizing a very fascinating conference. So, this my today's talk builds on the presentation by Hikari. So she attended the meeting from 2 years ago. So sleep is evolutionary conserved in all animal species which have the nervous system. But when asked what is happening when we become sleepy, so it cannot be answered clearly. So it was stated almost 20 years ago, but the situation is still like this. And so we have been hiring the methods of the genetics and molecular cellular biological approaches to solve the mystery of sleep. So we have been conducting the What genetics screening, EN mutagenesis, to find novel sleep-related genes. Then, as Dr. Funato told, so we reported the NALCN channel gene is involved in the REM sleep regulation. The dreams mutant mice shows REM sleep abnormality. The cause is NALCN. So let me briefly talk about this. But we can, so I can skip this. So as Dr. Funato told, so there is three phenotypic features, typical phenotypic features of the NALCN dreams mice. So REM fragmentation and reduction in total episode duration REM sleep and waveform change during REM sleep. So and the mutation was found in transmembrane domain 1, segment 6. So, and as we expected, DREAMS mutation is gain of function mutant. So we observed increased membrane potential and higher neuron excitability of DPLM neurons, which contains a lemma of neurons. But it is, there's issues. So we don't know that much about the molecular mechanism of REM sleep regulation. So, can I go back here? Okay. So REM sleep is, what is REM sleep? So REM sleep is many things like cortical activation, rapid eye movement, and muscle atonia. Then REM sleep is characterized by the predominance of theta oscillation. It's a basics. So today's topics, I'm going to talk about the recent research. So I have three topics. Then I'm getting started from number one, name and methods. So what is the molecular mechanism of REM regulation by NALCN? So this is what we don't know much about. So for example, the unknown components of NALCN channel complex, or we don't know the subcellular localization of NALCN in the neurons. So to identify, to identify clarify the molecular regulatory mechanism of NALCN, we decided to conduct the proteomic approach by using the iBio-ID techniques, in vivo bio-ID techniques. So we chose two components from NALCN complex. NALCN and ANKT. So the bio-ID techniques is known as one of the proteomic approach to find the proximity proteins by using the ligase activity of the BIL-A. So to label the biogene within the 10 nanometer nearby. So we could detect neighborhood proteins by using this protocol. And BIL-A tagged knock-in mice we generated in each gene, and NALCN and AVTS. And as you can see here, that we observed the tag-fused protein expression by Western blotting like this. This is a brain tissue. Then this is in vivo bio-ID workflow. So this was very kindly supported by Dr. Sodaring and Daichi from Duke University. I visited there. Then this scheme was originated from his laboratory. Then so biotin injection using knock-in mice, and second, proximity of biotin labeling by biotin injection in living neurons, and streptozin pull down by sampling the neural tissue from the brain. Then lastly, performed the LC-MS to detect the proximity proteins. So let me move on to results. This is confirmation of the expression of biotin-related proteins in knock-in mice. So then we perform the LC-MS. So this is just a visualization of each bio-ID trials of NALCN and ANK-AT. So as you can see, a lot of the biotin-related protein was detected like this. Then the circle size indicates the force change of the detection level of the proteins compared with the control. And samples were collected from the cortex and the hippocampus tissues of the knock-in mice. So this is just from Volcan plots. So surprisingly, the proximity proteins of each both knock-in mice are very highly overlapped like this. Then we also performed a hypergeometric p-value test to test the reliability of the overlapping. Then the p-value is quite small, indicating that this overlap is highly and significant, statistically significant. And we also noticed that ANK79 are involved in both trials, suggesting that this iBio-ID trials works well. So this is a scale-free graph of iBio-ID network, and here is the overlap, and this is not NALCN only detection and ANK-AT only detection. And the overlaps are here. As you can see here, the many proteins are represented in here, like ZBP1 or RPLP2 or GPL1C7C or BCAS2 chains. So it's very nice for us. And this is just the overlap examples of top detection of this. I can skip this because it's too much complex. And this is clusterogram figures. And we noticed that the most favorite representation are the membrane-associated proteins like this. But also, interestingly, cytoskeletal and mitochondrial-associated proteins are highly represented like this. But on the other hand, the trafficking-associated proteins are only represented in ANK-AT, but not so much in NALCN bio-IDs. And this is, we also tried enrichment analysis of both iBio-ID. Then, as I told, mitochondrial and cytoskeletal associated components are highly enriched in iBio-ID trials. So finally, this is my discussion part. So in vivo bio-ID could be achieved in both NALCN and ANK-AT knock-in mice. And detected proximity proteins in both trials are were highly significantly overlapped. So indicating, I think this detection goes well. So but these those detected proteins has not been proven to be a part of the functional partner of the NALCN complex. So, but I hope there must be the potential protein partners of the crucial for NALCN channel complex function. So, our interest is sleep. So, in the future, so I'm going to try the functional analysis of the candidate of the protein partner of NALCN complex. So we are just planning to do AV-mediated knockdown of those genes and perform a sleep analysis by using EEG recording. So, but I hope the more functional analysis to be achieved. So the collaboration is very welcome. Yeah. Then final part. So I would like to say thank you to Dr. Yanagisawa and Dr. Funato for kind support and very great support of the BioID to Dr. Scott Soldering. So I'm happy to engage your questions from audience. Thank you so much.
I have one. So you use BioID for NALCN and UNK80. Why you did not do UNC79?
Yeah, so I thought that we should try it, but we just started from NALCN and UNK80 and NRF1, so from 155A, but we failed to make the knock-in mice of from 155A, so then we just tried.
Great job anyway. Thank you so much.
Yes, thank you.
So the next speaker is Isabel Del Pino from Spain. Unfortunately, she was unable to come here, and she sent a recorded presentation. So she is a researcher at the Institute of Neuroscience in Alicante in Spain, and she's working on a cortical development, actually, using loss and gain of function mouse models.
Good morning, everyone. I'm Isabel Del Pino from the Institute of Neuroscience in Alicante in Spain. I'm going to take you through our recent work on the role of NALCN in cortical neuronal excitability and circuit integration. But before starting, I would like to thank the organizers and the local hosts for inviting us to share this work today at the second NALCN conference. I very much appreciate that you guys made possible my remote presentation. It's a privilege to present our work on how NALCN regulates the development of the cerebral cortex. Ion channels, such as NALCN, do much more than enabling action potentials. Way before neurons elicit mature spiking, ion channels such as NALCN are responsible of setting the electrical competence by influencing the resting memory potential during development. Therefore, they are crucial in shaping activity-dependent developmental milestones called critical periods, some of which are illustrated in the schematic, during embryonic, postnatal, and juvenile stages. Our ultimate goal here is to understand how NALCN dysfunctions, which lead to few millivolt changes in the resting memory potential, can drive an important spectrum of developmental symptoms observed in both IHPRF1 and CLIF AHDD patients. We approach this problem by thinking on a cell type specific manner and using the cerebral cortex as a model here. We focus on two different types of excitable cells in the cerebral cortex, which are glutamatergic excitatory neurons, which make up to 80% of neurons in the cortex, as well as GABAergic inhibitory neurons, which make up to 20% and are constituted by a wide diversity of electrophysiological, molecularly, and anatomically different cells, which are illustrated here in this schematic. We hypothesized that NALCN perturbations would differently affect the development of these different kinds of neuronal populations. And to test this idea, we use genetic mouse model by which we manipulate NALCN selectively, specifically in neuronal types and specifically selectively during development. I will present today our results on the contribution of NALCN to development of cortical neurons using NALCN deficient mice. And in a subsequent part of this talk, I will briefly mention our findings on the mouse model in which we have introduced gain-of-function mutation of NALCN in different types of neurons during development. Let me start by mentioning who is behind this work. This project is part of a big initiative called the Restore-LIC Initiative, which is funded by the ERANET European Framework. We are combining here experts in NALCN physiology and pharmacology. These are Arnaud Montet in Montpellier, France, who is our coordinator, Stefan Pless from the University of Copenhagen, with clinical neurologists such as Antonio Gil in Madrid, and experts on gene therapy. The idea here is to try to convert all the basic scientific findings from NALCN function and regulation into therapeutic approaches. The work on the basic neuroscience of NALCN has been spearheaded by a very, very talented student, Candela Baretino, who is today there in the NALCN conference. And also our work has been helped a lot by all the NALCN experts who attended the first NALCN workshop in Alicante back in 2023. Therefore, what you will see today has been definitely a very collaborative effort. I'll start by telling you a bit about what back in Alicante in 2023, we knew from the developmental expression of NALCN in neurons. From developmental data sets of single-cell RNA sequencing from the mouse brain, we could expect that NALCN is expressed in both excitatory and inhibitory neurons since early and embryonic mouse development. To some extent, we could observe this in a few data sets from single-cell RNA sequencing from the human brain. But what was definitely very valuable was to confirm this information, these results, with the very valuable tool, which is the NALCN GFP reporter mouse, who was generally provided by the Jean Ren from the University of Pennsylvania. Using this reporter mice, we could observe that NALCN is expressed across glutamatergic and GABAergic cell types since early in postnatal development. These findings motivated us to pursue specific deletions of NALCN in different types of neurons since early in brain development. We first measured intrinsic excitability across cell types in different mouse models. First, in these experiments, I'm showing you data from mouse lines in which NALCN has been deleted in GABAergic neurons since early in development. A specific type of GABAergic neuron called parvalbumin expressing, which is also named fast-spiking interneuron, showed the reduced and marked reduction in global excitability upon NALCN loss when compared to controls, together with hyperpolarized resting memory potential, suggesting that NALCN is important for the excitability of parvalbumin GABAergic neurons. In order to understand whether this was a phenotype that was specific for parvalbumin neurons, we also moved to somatostatin interneurons, another type of GABAergic cell that has this morphology, is also located in the hippocampus. When we recorded it from these cells, we also observed a marked reduction in input-output function, so reduced number function potentials upon the polarizing current steps, along with hyperpolarized resting membrane potential, suggesting that any NALCN loss is impacting the global excitability of both GABAergic and somatostatin interneurons. We wanted to compare how the impact of any NALCN loss on global excitability of GABAergic input of GABAergic inhibitory interneurons could be different from the one on glutamatergic excitatory cells. And in order to answer this question, we generated a mouse line in which all lineages of glutamatergic pyramidal cells are deficient in NALCNs since early in brain development. We examined the excitability of NALCN deficient glutamatergic cells and compared it to controls, and we didn't observe any significant effect, any significant difference in global excitability or resting memory potential. So altogether, this data indicated that developmental disruption of NALCN preferentially affected inhibitory GABAergic neurons and spared the excitability of principal excitatory cells. In addition, as we know that the developmental changes in intrinsic excitability can impair neuronal wiring, we assess for morphological features and inhibitory synaptic currents developed by GABAergic neurons. We observed that reduced excitability of interneurons was accompanied by abnormal dendritic morphology in subtype of GABAergic cells, such as parvalbumin fast-spiking interneurons. In these interneurons, the dendritic arbors were slightly more complex, with more dendritic branches. In addition, we recorded inhibitory postsynaptic currents from excitatory cells. These are inhibitory currents that are received by principal neurons from inhibitory neurons. We observed that the developmental disruption of NALCN was impacting and reducing spontaneous inhibitory postsynaptic currents, suggesting that the changes observed in excitability were impairing the dendritic development and also inhibitory synaptic connectivity. We then wonder whether the consequences of an abnormal cortical network with altered inhibitory tone could be observed functionally at the behavioral level. And to answer this question, we perform a battery of behavioral tests. Here you have a summary of the findings on the behavioral phenotyping. Mice with developmental disruption of NALCN in GABAergic population show impaired short-term spatial memory in the Y maze, which is a test used to assess for a spontaneous alternation of mice, indicative of short-term spatial memory. In addition, we observed that in an increase in anxiety-like behavior and then strong anxiogenic stimuli, as well as deficits in habituation to a novel context, as displayed by the distance that traveled by these mutant mice when compared to heterozygous or control animals. These phenotypes indicate that early life loss of NALCN in interneurons produces a lasting cognitive and affective consequences. To summarize the findings on this loss of function story, we have shown that developmental disruption of NALCN reduces interneuron excitability of different types of GABAergic neurons, but not principal cells, leading to morphological deficits of the dendrite arbors and impaired circuit integration of GABAergic neurons, resulting in memory deficits and problems in habituation to novel contexts, as well as affective-like behaviors. This strongly supports a model in which NALCN is essential for normal resting memory potential inhibitory neurons. In addition, we also used a mouse model with a gain-of-function mutation of NALCN. Here, our goal was, on one hand, to generate a mouse model relevant for the CLIF AHDD syndrome. And on the other hand, we wanted to determine whether NALCN activity during development, increased NALCN activity during development, produced the opposite physiological phenotype that we observed for the NALCN loss of function part. In this slide, I try to summarize the findings of the second study that you will find tomorrow in the presentation, poster presentation of Candela, which takes place tomorrow in afternoon. A brief spoiler of the findings are here. First of all, I would like to thank the Funato and the Yanagisawa labs for providing a crucial tool, which is the genetic transgenic mouse line necessary to develop this project. Thanks to this mouse line, we could introduce a different genetic dosage of NALCN gain of function mutation variants into GABAergic neurons. Our findings include surprising results on developmental milestones of GABAergic neurons, also phenotypes on intrinsic excitability that were not expected from the previous working hypothesis on what's NALCN doing on receiving potential of excitable cells, and also include the behavioral testing of these two mouse lines. In addition, in taking advantage that we are part of this RestoreLink project, we used these two mouse lines to test for blockers of NALCN and their impact on behavior. So all these results, you will find them in the poster presentation of Candela tomorrow. I highly encourage you to drop by and tell us what you're seeing and share your thoughts with us. So in the interest of time, I'm a little bit late, I'm sorry for that, but I wanted to share all what we are doing here. We are very excited with all the input that we have received in the past. I'm very sure that this is going to happen in the near future, and I hope to meet you again sometime soon in person. I want to thank my lab members, present and past lab members, especially Candela, who has done tremendous work, Alvaro for the bioinformatic part, and Gilsera and Sofia for technical assistance. I would like to thank all collaborators on the RestoreLink consortium. You know who you are, and I'm very, very, very thankful to you, as well as the rest of collaborators that have provided us with very valuable and crucial tools such as mouse lines, which are to date the most important models to understand neurodevelopmental disorders. And with this, I would like to close and give some time for questions. Thank you very much for your attention.
So it was recorded. So is it possible to communicate with Isabel or not?
Not live.
Okay.
So if they have questions, they can send them to me or enter them into the.
Okay. Okay, so I just sent her a message actually. So the session is over. So it is time for lunch now, thanks to every speaker, and not me this time. And see you at 1 p.m. And Stefan, please. You will be... 1:15, okay, better.
All right, welcome back to the next session of today. I'm going to be chairing this session, and I'm also going to have the awkward task of introducing myself. So I'm Stefan Pless. My group works on NALCN at the University of Copenhagen, and I'm going to share with you a few bits of data that we have generated a few years ago, and now some very new data that might be helpful to some of you in the context of what we've been talking about this morning also. So I'm going to talk to you about the regulation of NALCN by both endogenous proteins as well as de novo designed protein binders. So this is the channel structure as we've seen it multiple times this morning, and basically in my group we have been wondering, are there any other proteins that sort of interact with these ANCs here? And there was a beautiful talk just before the break basically about this question, which other proteins interact here? And recently we've also gone into the question of, like, can we actually make new binders rationally that bind to these complexes, as well as what Dejan brought up earlier, can we actually use it to disrupt the interactions between the channel and the ANCs? So I'm going to start by talking about some work that has been initiated by a previous postdoc in the group, Sam Usher. And it's basically, he came to the lab and he said, I want to use bioinformatics to identify new protein-protein interactions within this NALCN channelsome. And initially, I have to say, I was very skeptical. So I grew up not being surrounded by computers or TVs, and so I have a sort of natural skepticism against these kind of fancy devices. But he convinced me, and I gave him 3 months. I said, within 3 months, I want to see some results, otherwise we move you on to the project that I had in mind for you. But he did succeed. So basically he said, look, if there are proteins that have co-evolved with NALCN, if there are proteins that co-express with NALCN, and if they show similar disease associations, there might be actually indications that these proteins might actually interact and affect the function of NALCN itself. So he did some bioinformatics studies, basically he developed a pipeline that allowed us then to come up with a UMAP projection. I'm going to walk through this very briefly, basically. So these are a bunch of genes, and it's all about the proximity to NALCN and the UNCs here and FAM155A. And essentially what we did was we picked the top candidates from this list, and we screened them functionally for any potential effects on the function of NALCN. And just in heterologous systems. And I'm going to go through this very quickly. Basically, we found that in the presence of certain SNAR proteins, so proteins that are normally associated with a SNAR complex in synaptic fusion events, for example, they seem to have a very strong effect they would reduce the wild-type current to almost nothing. Sort of motivated by this, we did some, I call it structural predictions here. We basically did some mass spec cross-linking. We couldn't get a full structure, like an actual cryo-EM structure of the complex, but we did some mutations, some AlphaFold predictions to narrow down the likely interaction site. And we finally did some mouse work with the group of Christian Rosenmund. And all of this led us to propose basically that there is an interaction between NALCN and SNAR proteins. And this was all based on this computational approach where we use coevolution, co-expression, and disease association to come up with this sort of new interaction. We found that these neuronal SNAR proteins inhibit heterologously expressed NALCN quite potently. We saw that syntaxin knockout increased sodium leak currents in hippocampal neurons quite dramatically. And actually what was really interesting was that inhibition of NALCN enhances the survival of syntaxin knockout neurons. And this has been a mystery in the field, why this would happen. Usually syntaxin knockout neurons they die very quickly in culture, and it seems to be related, at least in part, to NALCN. We have additional candidates, and I had kind of optimistically put it in my abstract here that I might be talking about them. We're not quite there yet, so we are screening additional candidates. So this was the part of the talk that I just briefly want to get out of the way because it's all published work. You can look it up online. So I wanted to move on to something that's new and hasn't been sort of broadly distributed yet. So basically, there is this idea that yielded the Nobel Prize to David Baker last fall, where basically he motivated Sam again to go back to the computers and come up with a new approach to make new binders for NALCN. So he was later joined by Emma on this particular project, and this is the work that I'm going to show you here. So essentially, we use an approach that was pioneered by David Baker. It's called RF diffusion. It's a computational approach where you take a protein, you basically need a structure of known, like a known high resolution structure of your target, and then you tell the computer, so this is our target here in blue, and then you tell the computer, come up with peptide or protein designs that will actually bind complementary to that target interface, okay? And this approach has been used very successfully in the Baker group to basically design mini protein binders with very high affinity against various targets, for example, viral proteins, and also recently an amazing paper actually to neutralize snake toxins. Actually, the group of Manu Benjoni, they have been using a similar approach recently to also target calcium and sodium channels. So the workflow that we've been using is relatively simple. So in the computer, we make backbone designs for these binders, and these binders are typically between 40 and 80 amino acids long. We then fit them with amino acids. We ask AlphaFold if AlphaFold thinks these are good binders. AlphaFold is not always right, but many times it is. Then we have different routes. So depending on the target protein, we can either heterologously express these potential binders, or we can actually purify them. And then we can test them either in single-cell electrophysiology or high-throughput formats, either in high-throughput screening electrophysiology or flow cytometry or things like this. So in this particular case, we wanted to target intracellular protein-protein interactions that this NALCN channelsome is involved in. So this is the channel complex now turned by 90 degrees, essentially. And so there's this interface between the young proteins and NALCN, where basically you see this so-called D2, D3 linker making extensive contacts with the young proteins. And we thought maybe we can compete this off by designing a new binder against this interface. And the reason was that we had actually shown previously that if you just express the D2, D3 linker in its entirety, you can outcompete the interaction with youngs. And this looks, experimentally, this looks relatively simple. So this is just the experimental paradigm here. So this is the normalized current. It's actually the outward current at plus 80 millivolts. So if you have NALCN just here that's normalized to 1, you have a substantial current. But then if you express this linker construct in trans, you actually get a massive reduction of the current. However, I should point out that this is the entire linker sequence. So this is no longer a peptide, it's a protein, basically, 250 amino acids. But if you co-express it with the complex, it's very good at killing its function. So we thought this might be a good point to actually compete for the interaction, but at the same time, we knew that we probably needed very large constructs to do this. So we also wanted to look at the D1, D2 linker interface, which is much smaller. So it's this tiny loop here from NALCN that reaches out to UNC79. And at the tip of this loop down here, there's a tryptophan that's very important, that's going to play an important role in just a second. So this particular interface we were not so sure about. We thought maybe we can design a binder against it, but we wanted to know, is it even worth it? Because if we displace the linker, is there any effect on the function? So what we did was, this is again the same sort of experiment here, when we now express the D1, D2 linker, which is much, much shorter than the D2, D3 linker, in trans with NALCN, there's almost no effect. So the question that we asked was, can we displace the linker? And the answer was kind of like, yeah, not so sure. We were not so sure about this. So we wanted to see, can we maybe anchor this construct a bit closer to the membrane? And we designed a few constructs where essentially we have the linker sequence in red here, always on a membrane-anchored construct and separated by a little linker. And then we did this with different membrane anchors, essentially. So the idea was that we wanted to bring this small loop closer to the membrane. And when we do this, we can actually see that you get a good inhibition, both when we put it on something like NAV1, this is NAV beta 4, or an ASIC-derived construct, we get reasonable inhibition. And in both cases, when we mutate that all-important tryptophan, the inhibition is actually quite a lot relieved. Actually, much more so in this construct than in this construct. In both cases, the deletion has a strong effect, and we know actually when you, can I go back? Yes. When we make the linker longer or when we scramble the sequence, the inhibition goes away altogether. So we were relatively certain that, yes, we can target this interface. And so we went back to our computational approach and we said, okay, we want to target this area and this area. So we went ahead, went about it like this. We made about 5,000 backbone predictions. We then used MPNN to come up with 30,000 sequences that were optimized for stability and solubility. And then we used AlphaFold again. We applied a cutoff, and we used the so-called PAE, or predicted alignment error, to select the binders that were supposedly good. And we then went into a functional screen. Initially, we screened about 40 of them, and we used a very crude approach, DNA injection, so nuclear injection in oocytes. This yielded a relatively messy picture. So this is our positive control here, the D2, D3 linker, and then there was a broad spectrum of different inhibitors or potential inhibitors that we tried. None of them did very much except for one that looked slightly promising. So then we followed up with a more sort of subtle screen where we actually did proper mRNA injections. And then we saw that this sort of binder that we now call D3, and which I will show in red here, actually has a very good inhibition in these mRNA-based sort of inhibition assays. So we went ahead with this one and wanted to validate the predicted interaction site. And now, to apologize that the binder shown in red should be shown in red here, as in blue now, but it doesn't matter. The main point is that there are a few amino acids here that were supposed to be very important for the interaction, especially these two side chains here, and especially this phenylalanine, because that is exactly occupying the spot where normally the tryptophan sits of the native linker, essentially. So we went ahead and did the same experiments. So there's the outward current, NALCN alone, the D3 construct, it potently inhibits it. If we now make mutations at this phenylalanine, or this one, or both together, we got rid of the inhibition altogether. If we make this, if we make this phenylalanine tryptophan, it's still a very good inhibitor, as we would have predicted. And if we mutate this tyrosine here, we get rid of the inhibition. If we get rid of this one here, it's not quite as strong, Sorry, the other way around, actually. But the interesting thing was actually when we mutated them both together, the inhibition was actually still retained. So we think if we mutate both of these side chains, these two interact tighter with their target sequence. Our control in this case was this leucine up here, which doesn't have much of an effect when we mutate it at all. So at least broadly, this seemed to confirm the predicted binding pose and interaction site predicted by the computer. So we actually wanted to know if it can also inhibit gain-of-function variants. So this is just a wild type. As I've shown you before, with a D2, D3 linker, we get good inhibition. With a binder, we get good inhibition. And the same picture is true for actually a relatively strong gain-of-function, the R1181Q variant. So we get good inhibition for those as well. So in conclusion, we've sort of designed and validated a mini protein that's capable of intracellular binding to the channelosome. We can use mutations to verify this, and it is quite good at even inhibiting strong gain-of-function variants. We are trying to deploy this binder now in neuronal cultures, but we haven't quite gotten there. We have also tried the obvious thing, which is obviously to make binders against the extracellular side. We have so far completely failed, but we're still trying. But if anybody's interested in the sequence of this intracellular binder, we're happy to distribute it and for you to try whatever you want to try with it. And with that, I would like to thank the groups, and I'm happy to take any questions that you might have.
I was wondering if some more small molecules might be more useful than this.
I guess it depends on what you want to do. I completely agree. This is not something that you would want to sell as a potential drug or something. This is not a drug. This is just a tool compound to basically, along the lines of what you said earlier, it might be useful to disrupt these protein-protein interactions on the intracellular side using something like this. And this is just a relatively easy workflow that we have implemented in the lab, and we just thought we'd try it here. So by no means do I want to sell this as a drug. If somebody develops a small molecule inhibitor of this channel, it's going to be much more useful than this. But I think it can still be used for example, knocking down the activity in various types of tissues or things like that.
Maybe another quick question is that when you looking at the interactions, have you also looking at, like, say, proteome, whatever database, see that interactions, is that very unique for these two proteins, or could it also occur for other protein-protein interactions?
I'm not sure if I'm understanding correctly what you're asking, but so are you asking whether our binder might bind to other interfaces also? Yes, that's absolutely possible. And this is a question that we get a lot, and right now there is no computational pipeline that would sort of look at all structures that are out there and sort of do a negative screen of this kind of binder to see if it would still bind somewhere else. So it's something that we cannot rule out at this point. So what we're doing is, sorry, it's gone now. So what we're doing is we're using the binder that is exactly the binder that we've worked with, except the two all-important phenylalines are mutated. And those we're using as a negative control, basically, in our experiments that we're now developing in neurons, basically. So we have a binder that binds, and we have a binder that shouldn't bind. That's our control. But you're right, they could still bind to something else. We don't know. We don't know for sure. Good. In the interest of time, I'm going to introduce the next speaker, Han-Chao Chua from the University of Copenhagen. No, he used to be at the University of Copenhagen. He's now a group leader at the University of Sydney, and is going to talk about making sense of missense and nonsense, a functional assay to assess variant pathogenicity.
Hi everyone, my name is Chao, and as Stefan said, I'm currently at the University of Sydney in Australia. So my connection with NALCN actually started in 2016 when, right after I finished my PhD, fresh, naive and gullible, and Stefan convinced me to work on NALCN, and the rest is history. So today I'm going to talk about a functional assay to assess variant pathogenicity for NALCN. So thanks to all the speakers this morning, so I don't really need to do much more introduction about NALCN, so I'm going to go straight into the goal of the study. So we really wanted to create an assay that is sensitive and accurate to help coherent genotype-phenotype correlation and hopefully guide precision medicine in NALCN channelopathies. So that's a lot of words, but what does that really mean? So essentially, if I can get this to work, so essentially what we want is to have a way that can confidently measure the changes on a molecular level of different disease variants. And the idea is that if we screen sufficiently large number of pathogenic variants, and in parallel with detailed clinical description of patient phenotype, we should be able to classify the molecular and clinical phenotype in a meaningful way. And of course, the beauty of having an assay like that is that it also hopefully would allow us to screen different clinically available drugs on different variants of interest to guide precision medicine. So typically, work as such, so creating assay that measure pathogenicity of variants would involve the characterization of wild type and also novel variants. But thanks to the rapid evolution of sequencing and electrophysiology techniques, but also, I guess, the continuous refinement and standardization of variant categorization criteria, it is now recommended that an assay as such should also include recurrent pathogenic variants that would help us to characterize the molecular changes in both directions. And we should also include common population variants that are believed to be benign, so non-disease causing. The assay should also be measuring relevant parameters. So these are parameters that would reflect the channel function in vivo, but also it should have the sensitivity, like I said, to measure direction in both ways, so increase and also decrease in function. And hopefully once we've done enough numbers, then it should be, the molecular phenotype should correlate with the clinical phenotype. So with that in mind, we went straight into de novo cleavite-associated variants. So shown here is a simplified topology of NALCN, which I'm sure all of you are very familiar with. So highlighted in pink circles are all the previously studied de novo cleavite variants. So at the time when we first started working with this, it was an overwhelming GOF picture. So a lot of these variants have been shown to increase NALCN function. I've also highlighted in red circles some of the more novel, at the time novel, pathogenic variants. I've also highlighted in green circles 4 of the most common, frequently occurring population variants in this cartoon. So we express some of these variants. So I'm showing some representative current traces of different Z-apoptosis expressing wild type. This variant called W1287L, which was detected in IHPRF1 patients in 3 siblings. So this, at the time, was the only variant that caused a decrease in function. We also chose V1020F, which was detected in a cleavite patient. And the reason why we chose this as our internal control is because it was the oldest cleavite variant that we have characterized in the lab. And of course, we've also picked benign variant that was detected at very high allele counts in the NOMAD database. And there are about 600 people in the population that carry this variant on both alleles, suggesting that this is unlikely to be causing disease. And as you can see, there are clear differences in the current level. So I've indicated on the wild type trace here, the IMEX, which is clearly different for the different variants. But because the maximal current amplitudes is only showing a value at a specific time point, we thought we should also characterize the total charge that occur over time. So we're also measuring the area under the curve to better characterize the the total charge transfer across 1 second of hyperpolarizing pulse. So this is basically a summary of the data of IMAX and also the AUC normalized against the individual wild-type batch value. So as you can see, our gain-of-function V1020F variant is consistently showing increased current levels and AUC compared to wild-type, whereas the IHPRF1 loss-of-function variant is consistently decreasing the current or showing less current level and also less charge transfer, total charge transfer compared to wild-type. And the 4 population variants that we've chosen are, I hope you can appreciate from this graph, fall within the wild-type range. And so that gives us the confidence that the assay is actually sensitive enough to pick up both loss and gain in function, but also benign variant. So we then went on to characterize 7 novel cleavage-associated variants shown here. So in red are the representative current traces, and I hope you can appreciate straight away that the maximal current amplitudes of most of these variants and also the AUC are all larger than that of wild type, which is summarized here in this bar graph. And the orange circles are essentially us trying to mimic the heterozygous nature of these variants. So we're mixing wild type and variant RNA and inject into oocytes. But this is just to confirm the dominant nature of these variants, as there is no real difference between heterozygous and homozygous condition. Now, what's really interesting about these variants was the K1115N mutation. So this is found in the selectivity filter of NALCN. So when we look at the IMAX values and the AUC values, the variant is actually not that different from wild type, which is interesting because in our hands, we've worked with this variant before and we know that it behaves very differently compared to wild type. So then we thought maybe we're not measuring the right parameters. So we then went on to look at 2 extra parameters. So the first one being the ratio of current at steady state to instantaneous. So essentially, this is an indicator of how broken the channel is. So if you look at the wild type trace, you can see that with the hyperpolarizing pulse, there is a large initial increase in current, which very quickly go back to a resting state, if you will. Whereas with some of the gain-of-function variants, the channel remains open across the entire pulse. And as I think Arnold and many other speakers have mentioned, NALCN is highly sensitive to inhibition by extracellular calcium and magnesium ions. And so we've also measured the current ratio in the presence and absence of divalent cations, and then we compare it to wild type. So how am I doing in terms of time? Okay, 3 minutes. So I'm just going to summarize that very quickly. So essentially, we found that a lot of the gain-of-function mutations are affecting the ISS to I instantaneous ratio. But what was really interesting was that no matter how affected the maximal current levels or the AUC are for the different variants, their sensitivity to divalent cations remains unaffected, except for the lysine at position 1115 that was mutated to an asparagine. So that was the only mutation that we found that statistically significant affected the sensitivity to divalent cations. So the way we're interpreting this data is that depending on the location of the mutation, the way that they're affecting the channel might be different. And so that would be totally dependent on the parameters that we're measuring. We also look at IHPRF1 associated variants. So these are found on both alleles of patients, and a lot of them are actually truncating variants, deletions, and also frame shift mutation. So you would expect with this sort of mutation, it will kill function, which is essentially what we found. But what was really interesting for us at the time was that there is this missense variant, G883V. This is the second IHPRF1 associated missense variant. And so we were really interested if it would show loss of function, just like the W1287L mutation. When we express that in xenoposoocytes and characterize the maximal current value and also the AUC to that of wild type, we found that there is no significant change, suggesting that, at least in our assay, this is not consistent with the molecular phenotype changes associated with a pathogenic variant. However, it's very interesting that it was initially reported in a young patient that showed developmental delay, but also seizures. So it'd be interesting if we can get clinical information about this patient, if she's carrying other variants, or perhaps another possibility is that our assay is not picking up other, I guess, potential changes in vivo. There are also a couple of compound heterozygous patients. So these were detected in about 3 children, and they all had mild to moderate developmental delay, hypertonia, and sleep apnea. So compound heterozygous, so on one allele, they carry truncating mutation. On the other, they have a missense mutation. So we tested individual variant, but also the combination of it. And what I've essentially shown here is, as expected, the truncating mutations kill function, whereas the missense variants, surprisingly, did not seem to change significantly the function as quantified by the maximal current values and also the AUC. So we don't really know what to make of this, but what is interesting, I guess, is that 3 out of the 4 mutations mentioned here are found in the population database, suggesting that on their own, they are less likely to be the monogenetic cause of severe neurodevelopmental disorder. I should also mention that all 3 patients actually carried mutations on other genes that have been associated with neurodevelopmental disorder. So whether or not these NALCN variants are actually pathogenic is still unclear. So in conclusion, we wanted to create an assay that is sensitive and accurate. Did we do that? I think to a certain degree, we have shown that we're able to pick up both increase and also decrease in NALCN function. We've also shown that benign variants or population variants are not causing any significant change molecularly. And I want to, I guess, stress that it is important to have controls. And also to have the right controls is extremely important. So looking back, I think we could have chosen better recurrent pathogenic variants. So for example, the arginine at position 1181 that was mutated to glutamine has been reported in different patients. So perhaps that would be a much better recurrent pathogenic variant control. I also think it's very important to have significant throughput in the assay so that we are able to test different variants on the same day. And I think it's very important to think about the parameters that we're measuring, if it's actually relevant to the function of the channel. And hopefully in the future, with more patient information available, we'll be able to correlate that with the changes in molecular function. And with that, I'd like to say thank you to Stefan, my postdoc supervisor, and also people from the PLES lab who have contributed to this project. And if you're interested in the story, we've published this on bioRxiv, so yeah, have a look. Thank you.
Thank you very much. Questions for Chao?
Oh, okay, sorry. Sorry, guys. Okay, so you seem to that many of your what they call gain of function, you do see gain of function currents. Now, Arlo this morning presented 5 out of 32. This rate is much lower, right? Do your mutants, the mutants you studied, overlap with the Arlos?
I have to say, it is on my to-read list, I know, but I just haven't had the time. But it will be interesting to compare. But I guess, regardless of which system you use, I think it is important to standardize the assay, whatever you're expressing the channel in, to the different pathogenic controls. So I'm sorry, I couldn't answer that question because I haven't had a detailed look into the data. But yeah, it will be interesting to compare and see if there is any similarities or differences. Yeah.
Good. In the interest of time, we'll move on to the next speaker. Thank you very much again, Chao. The next speaker is Mei Zhen. She's a senior researcher at the Tannenfeld Institute in Toronto, and a professor at the University of Toronto.
Oh, wow. Okay, thank you. Can you hear me? Yes. How did this? All right, so I will start slightly differently. I thank the organizers for sure to inviting me to be here, but Jeremy at the beginning actually mentioned everybody was here through a journey. So I think almost all of you only know me for being here. If you know me, it's my work, my group's work on identifying the channelsome subunits. But I want to share the other side. I just come from China because I was there to visit my father, who's 93 years old now. And the reason is he has been diagnosed with Parkinson's almost 30 years ago. And me and my mother has been the primary caregiver for the last 30 years until a year ago. I have to send him to China because he wants to go home. And so, you know, as a researcher, I am very, very much fundamental biologist. But as a caregiver, I also understand that the need from a patient side or family side is completely different. And that explains to you, Lou, sometimes I have this visceral response, the repulsion against scientists who use curing disease or related disease as a mean to get funding or get competing ahead to get a paper. So if a lot of people said May is very naive and always openly sharing things and ideas and never ask for credit, I'm not naive. I just have to tell you, this comes from my personal experience. I want you to see research done in a fundamental way, in a high quality way, but on the other hand, it needs to go faster to help the patients and the parent. All right, so that take my 2 minutes. So let's go on. And then the next thing is I'm going to present you the work that really has never been published, and it still hasn't been published so far, but then these are the trainees who really fundamentally in the first phase, build a foundation for us to identify the cause of CLIFAT and then the biology of channelsome. And also Tian and Lu, who's here, will present in the later sessions what we're continuing to working on that help to understand the biology as well as maybe some useful things for the patients. So the first real causal mutation for CLIFAT is caused by gain-of-function mutation of this sodium leak channelsome was in fact identified in 2015 in Montreal, and then in collaboration with my group. And it was done and confirmed in C. elegans, not using HEK cells. The reason is in C. elegans, and that's the R1181Q mutations, and this has turned out to be one of the most recurrent mutations identified in different ethnic groups. I call it a specific things is causal because we know this is single mutation is sufficient and is unnecessary to generate this syndrome. And then we also know that we talk both loss and gain of function mutation of the channel activity cause two opposite genetic effects and has two disorders. First one is when you talk about the CLIFAT, everybody heard about here, this is caused by increase of the channel function. But loss of channel function was caused by loss of channel is the other order that was first reported in Spain in 2013. Is IHPF. They do have overlapping syndromes, but they are really caused by genetic mutations, has really the opposite effect on the role of the function of the channel. The reason that the cyanogen system is a unique one that can definitively differentiate the nature and the causality of the genetic variants is they cause two opposite behavior phenotypes. On the left is the behavior phenotype of a normal C. elegans that generates sinusoidal movement that continues to move. In the middle is the loss of function. You can see this worm just keep on going back, keep on moving, but then you can see the head has this kind of a very, very unique kind of a stopping phenotype. It's not just a complete loss of locomotor behavior. It's one unique behavior. And the gain of function is the exaggeration and then complete switching. These are all the basic biology side we first noticed in C. elegans community and then in my group. We cloned the genes and realized that the middle is caused by loss of function, either partial or complete loss of the channel. Any of the channels in subunit in the right is really all the gain of function mutations that increase the channel activity. So by basically putting all those variants, if you can put the variants back into C. elegans, we have not failed at every single time to identify which one is gain and which one is lost. But since we find the first patient, I have been already thinking about knowing that this is an increased channel activity. How can we do something to help that patient's mother and then the family to elevate some of the symptoms while we wait for the gene therapy to come into place? So in 2015, I've been thinking about a few approaches, and then I was very lucky. I met Han Lan, who was my college friend, and then at that time in Cambridge, and was setting up her own pharmaceutical company, Ibergen. So we come up with a roadmap, communicating with them, is first of all, we know that this is a rare genetic disorder. There's very little incentive for pharmaceutical companies to develop new drugs for it. We know it's going to happen later, but its time is too long. So she proposed the idea to repurpose the compounds or FDA-approved drugs because most of the compounds that's been developed previously using cation channels are non-specific or has very broad spectrum. And then we also know the C. elegans gain and loss of function mutations has very unique phenotypes, so we can use this to start to screen those compounds. And then once that has been screened, we can use it to test for specific effects on the primary neuron cultures using rodents. And then the step one is to develop vertebrate animal models and then test them. And then with this way, we can accelerate the application to the patients. So I can tell you what we have done so far. We completed the first screen about 3 years ago. From 75 drugs that's selected by our colleagues over there, we found 5 compounds that generate very consistent behavior responses. We prefer to use C. elegans to start with because of the uniqueness of this phenotype. Does the movie play? So essentially, if you just do a drug to change the worm behavior, most common thing you will see is essentially that they will just stop moving. And that's not good because anything that stops anywhere in the neuronal function will stop them from moving. But use this, what we're looking for is the compound that will affect the gain of function mutant and wild-type mutants to produce this kind of straightening of head while they're stopping to move. Because that was the phenotype only generated by subunits deletion of this channel sodium unit complex. So that's why we decided to use C. elegans. So this is basically the R1181Q endogenous mutation in C. elegans, and then on the right is the compound that we selected. So from this, we come up with 5 compounds, and then we move on to do the primary rodent neuron culture to check the efficacy. Everything we used is use verapamil as a benchmark because verapamil was the original compound that we used to identify or isolate or characterize the sodium recurrence before we know what the genetic composition of that is. So from here, we basically started with this concentration that clinically used for verapamil, that's 0.5 millimolar. For the culture. So we found basically one compound that will increase the neuronal activity and then one compound will decrease the neuronal activity. And then there are specific things we check for consistency, the inward leak currents, the resting membrane potential, and then the action potential firing. As you can see, all those compounds has very compound effect, but nemethen is the one that's really consistently showing a decrease of both membrane excitability as well as activity firing. Sorry for time. So at the same time, while we're doing the screen, we already started looking into developing mouse models that would specifically help to test the compounds. Again, I want to give credit to Ibergen, who never failed to have faith in us, because we've been trying to generate this R1A1Q mutation endogenous model for 3 years and then failed in Toronto. And then there's a biological reason for that. But then this one finally succeeded. This is the approach we come up with. Essentially, we generate this mutation endogenously, and then we put a LOX P size in front to rescue this mutation. So basically, this is a conditional knock-in, but then we can drive the expression of this variant in specific tissues afterwards. All the other non-conditional ways we generate, we fail to generate it basically there's biological inability to inherit that mutation. And at the same time, we applied single-cell RNA-seq together with our Toronto colleagues, looking for specific brain neurons that's most suitable for test this assay. On the left is the data still we haven't published, this developmental single-cell RNA sequencing analysis of the rodents from P1 to P15. This is the specific cerebellum. These data basically says the most highly and consistent abundant expression persists during development is Purkinje cells. And this data has later been verified using published data in Allen Brain Institute and then the Brock Institute. It's very consistent. And of course, 2 years ago, I met with the doctors there and showing that the cerebellum atrophy is one of the hallmark features of a lot of patients. And I'm really glad this is the case. The fundamental research and then also the patient-driven research come together to meet us, meet in the right area. Sorry, it's almost done. So Tian is going to talk about it in this afternoon, that the assay that we developed and the electrophysiology phenotype that I'm absolutely confident about, what this cell model can tell us is essentially driving this gain-of-function mutation, increased excitability, but silenced the hyperactivity of the neuron. And meanwhile, knowing that this is a bit harder assay to test the drugs, we've been developing drug models that have more obvious phenotype that easier to look at the drug test. This is still the work in progress, but I think we have one that is reproducible. The one on the right is the mouse that carried the homozygous mutation of R1A1Q. It has developmental delay and then potentially motor deficit. I want to end by saying, for me, I have two sides. I'm a researcher who's really not that interested in saying to understand the research is only about curing patients. I'm not having any offense about this. It's just fundamentally, I am a scientist. I'm driven by trying to answer questions. But on the other hand, I have families that I care for, and I know why there's so much drive to push research to be a means to cure diseases. And then I have so many wonderful people around me that help me to do both. And this is an undergraduate student who devised something to help my father to gain motility in his last phases of his walking stage. And I just could not say more than what I say. Channeling help is modern science, it's not modern research, it's modern technology, but just don't tell me when you write grants to say that I'm curing disease when you actually don't mean to. Okay, that's it. Thank you.
Thank you very much. Questions for me? So we've talked about this before, so the inhibitors.
Yes.
Do you know what pathway they act through?
This identified inhibitor?
Yeah.
All the compounds that we selected to do the repurposing screen are so-called cation inhibitors, mostly with sodium, calcium, and also potassium. So at least some of the effect is coming in is with inhibiting of the sodium. This one is supposed to be sodium and calcium.
Yeah, so it's a general effect, not specific to any given target.
This is the idea, but then the reason I saw this one, looking at this consistency over here, to me, it has relative sensitivity, not just a general blocker. So I did not mention, when you actually look at the inward and outward leak, we actually did all the sweep of the analysis of inward and outward, and then with the potassium blockers and non-deployable. This one is relatively specific for non-potassium.
Okay. Thank you very much. That's, sorry, one more question. Good.
Just a quick question for the drug screen. Did you screen adults, the adult C. elegans, and what happens when you stop treating with the drug?
Yeah, so these, there's a lot of intricate details about this. These drugs has to be dissolved, right? So in some DMSO. So this is a solvent for those assays, because most of the compounds are not soluble in the solutions. And then the least toxic solvent, the time it requires for 1% DMSO maximum solubility is, we felt like after 5 hours, up to 5 hours, the animal will show aversive effect already in the solvent. So most of the compounds test, we limit it into 1 hour, and then for that reason, we do it in the adults. But we tested all of them. After we found it, we test them in the throughout developmental stages is consistent, and then the efficacy takes up very quickly within 30 minutes.
Good. Thank you very much again.
I can provide more details later on, yes.
I think in the interest of time, we'll move on to the next talk, but thank you again, May.
Yeah, but yeah, looking forward to, don't ask me the basic things, but then I think basic biology really matters. What he found there will be useful, and we'll be looking forward to talking to you. Yes.
Good.
Thank you, yeah.
Then it's my pleasure to introduce the next speaker, Linda Shea from the University of Texas, San Antonio, so a local. We're looking forward to your talk. Thank you very much.
All right, good afternoon. My name is Jenny Shea, and I'm going to be switching gears a little bit. And first of all, I want to figure out how to switch the slides. Is it just this button?
This one will move forward. Can you bring up the slides? This one is forward. Okay. That's the pointer. Sorry for you.
That's okay. Okay, so again, I also want to thank the organizers for this opportunity. Yes, my lab is here at UT San Antonio, just up the street, so it's really nice to have this very wonderful conference here in town. And so we heard a little bit today and throughout tomorrow probably, is that seizures is a common phenotype for patients with NALCN related disorders. And so what I want to do is I want to talk about a brain organoid model that is designed to model epilepsy in a dish, and how we could use this sort of case study or example to maybe think about translating it to other types of seizure-related disorders. So on this slide, this is just a table of some of the commonly prescribed anti-seizure medications. You can see the various different drug classes on the left. They're either altering sodium channel, blocking sodium channels, modulating calcium channels, GABA receptors, and other types of mechanisms that are related to generating excitability within the central nervous system. And then the examples of drugs in the middle and their indications on the right. But despite all of these very common anti-seizure medications, yet we still know that patients commonly, about 50% of patients respond, and then with each drug that they don't respond, the effectiveness diminishes. So this is still a very big problem. There's hundreds of thousands of patients that are deemed refractory to even the very common anti-seizure medications. Okay, so then, so many of these anti-seizure medications were screened and developed in rodent models, and despite the wonderful aspects of having an in vivo tractable model like an animal model, there are still limitations in the sense of how do they accurately model the human brain. And so we do have two-dimensional cell cultures that we've heard about, and those are also very amenable to drug screening. But there are species-specific differences and also the way these cells organize themselves during development. And for many of the disorders we're interested in this conference, development really matters, particularly the differences in the development of the human brain. And so we know there are human-specific genes that guide neuronal differentiation and migrations particularly, and also how the complex repertoire of cell types. So what I'm here to do is talk about induced pluripotent stem cells, and I hear there's other sessions that are also talking about iPS cells, but specifically how we can take iPS cells and form three-dimensional brain organoids. So these models have emerged in the last 10 or so years, and so you can pick your disorder, and we start with a monogenetic disorder, so I think that is very amenable to this conference. And we reprogram them from these patient, either skin fibroblasts or blood samples. And then what we can also do in order to, because there is quite a bit of variability for different pluripotent stem cell lines, even the genetic background of the patient, is we can use CRISPR gene editing to also control for that particular gene variant by making an isogenic control. And so we're very interested in looking at how excitatory neurons and inhibitory neurons develop and how they integrate to form circuits. So in this example that I'm going to share today, I'm going to be sharing you examples of cortical organoids that contain excitatory cortical neurons and also these ganglionic eminence-derived organoids, which come from the ventral part of the developing telencephalon, which inhibitory cells originate, and then they migrate into the cortex. So what we do is we make these cortical organoids shown on the upper right. At early stages, they have expressed markers of neural progenitor cells, and they also form these ventricular-like rosettes. You can see these tubular regions. And then, of course, when the organoids gets bigger and bigger, the neurons start to differentiate, and then they migrate from those rosette regions. And then also these ventricular zone or these ganglion eminence-derived organoids that express inhibitory neuronal markers shown here. And then the idea is to make these organoids, and then when they start to form those neurons, to put them together or generate these assembloids so that the interneurons from the ganglion eminence-derived organoids migrate into the cortical organoid. So then, okay, so again, the example I'm going to share is actually a gene that my lab has been studying for the last 5 or so years. It's actually one of the first single gene mutations identified more than 20 years ago called ARX, Arysilus-related homeobox gene. So it is a DNA binding protein, it's a transcription factor, and it regulates both positively and negatively other genes, particularly important for interneuron development and migration. So without really going into all of the detail for the sake of time, suffice it to say, we recruited patients with ARX mutations in the second polyalanine tract. So these patients have an expansion of their polyalanine, and it's thought that generally speaking, it's thought to be a loss of function mutation. So we have patient iPS cells that we've made from 3 patients with an expansion of their second polyalanine tract, and then we have control patients that we've recruited. And because ARX is on the X chromosome, all of our control patients are also male. And then more recently, we've generated an isogenic control line where we took a patient-derived iPS cells and we corrected the expansion of the polyalanine so that it resembles the control sequence. Okay, so then after a lot of studies, we've done a lot of phenotyping with immunohistochemistry and also single-cell RNA sequencing. But I think because of, again, for the sake of time, I really wanted to hone in how we might be able to start looking at functional consequences of this mutation. So specifically, how do we measure brain activity in a dish? So we finally decided to use multi-electrode array recordings, and this is the work of Sarah Mishra-Degi, a graduate student in my lab. She has a poster together with her undergraduate tomorrow in this meeting, so I invite you to look at her poster and ask her for a lot of the details of the work. So we collaborate with Ali Husseini and Michele Giuliano's lab in Italy, And our recordings are done in the incubator. The organoids are grown separately in the incubator, and then when we make the assembloids, then we place them gently on the multi-electrode array. And these multi-electrode arrays have 60 electrodes, and they're three-dimensional electrodes, so that they also help promote attachment of the organoids to those the 60 electrodes. So typically what Sarah does is she does a set of baseline recordings, and then she tests different pharmacology just to confirm that these organoids are active and they're behaving or responding properly, like for example to glutamatergic or to GABAergic transmission. And so what we found, at least with these ARX organoids, is that they show a lot of bursting activity and even a lot of synchronized bursting activity. And this is a sort of a representative example of a 5-minute recording in about of a 30-minute measurement. And so shown on the right, you can see that the ARX-derived assembloids have a lot more active electrodes, and if you normalize per active electrode, the mean firing rate is higher. And so in very preliminary data, we thought, well, maybe we can now begin to use this assay to look for drug repurposing. And so then we sort of wondered, well, what could be a target of this ARX hyperactivity? And this is a collaboration with Nick and Peter here in our preclinical pharmacology core, and also with Jonathan Bowman at the Southwest Research Institute. And so what we notice is that CXCR4, which is a G protein couple receptor, a direct downstream target of ARX, is overexpressed in the ARX patient-derived organoids. It's a little bit difficult to see, but this is the immunohistochemistry of CXCR4, which is at about 4 months in vitro is overexpressed. So there are known inhibitors of CXCR4. One of the drugs is called AMD 3100. It's a drug that's already FDA approved for mobilizing hematopoietic stem cells. So we thought, although AMD 3100 is not the best drug for the CNS, but we thought just for proof of concept, maybe we could test this. So we do have an assay where we're monitoring interneuron migration. So in these assembloids, remember, I just want to remind you, those inhibitory cells come from the ganglionic eminence and they migrate into the cortex. So you can model this by labeling with viruses both the excitatory and the inhibitory organoids before you fuse. And then after you fuse, you can look at the migration of those reporter positive cells from one part of the organoid into the other. So in this assay, what we found with AMD 3100, which is the CXCR4 inhibitor, rescue of this accelerated interneuron migration. So then we thought, well, this is interesting. However, interneuron migration interneuron migration is a process that occurs during development, and it would be very difficult to treat patients that are in utero with this particular drug. So we asked, well, could we actually treat the hyperexcitability? And it appears that in preliminary data to be the case. So even in assembloids that have this hyperexcitable phenotype, a very acute treatment of AMD 3100, I believe just for 3 hours, was able to diminish this elevated activity. So we are testing other types of molecules, and again, this is a topic of an ongoing collaboration, but I just wanted to show you this very preliminary data today that potentially you could think of a complex cellular model like the one I'm describing for drug repurposing. Okay, so kind of bringing it all back together, circling back to the first question, can we use this approach to improve treatment outcomes? And, I'm a very optimistic person, so I believe yes, and I believe that if we had an organoid model in part of the screening process after the initial EEG and the MRI, wouldn't it be great to have an organoid model on the side and then to be able to test at the same time as the physician is deciding which drugs to use, sort of this empiric approach to then also have the organoids see if we could also test and maybe even, help with a more precision approach. And then that could, again, maybe that would be a way to again, treat some of these patients that are medically refractory. Okay, so again, I just want to thank you again for this opportunity to present our work. I know that was really fast, but we believe that these more complex models could fill a gap between and just complement a lot of the models we have. It's probably not a great high throughput model because of the months and months it takes to, again, derive iPS cells and to make these organoids. But I think, one of the possibilities is to combine it with other more high throughput technologies, C. elegans, zebrafish, AI, and that could be very promising. So I just want to stop and thank everybody in the lab and for doing this work, and also thank our funding sources, and also for your attention. And if there's any time for questions, I'm happy to take any.
Thank you very much. Questions for Jenny?
I'm convinced this model system is really useful. But on the other hand, Davis asked, okay. So I wasn't convinced why the inhibitory neuron in this case is needed actually to promote synchronicity or hyperexcitability, because in a way, GABAergic neuron at this stage probably is excitatory anyway, right?
Right.
They are not producing inhibition.
Right. So that's a really great question. And because of the sake of time, I didn't even talk about that at all. So we actually have organoids at sort of mid-gestation, second trimester, compared to these later ones that we believe are more third trimester. And we do see differences both in the response to GABA and blocking GABA receptors. So I believe you're right. At the stages we're testing, it's probably depolarizing and excitatory. I will also say that we've done these mix and match organoids where we make a control ARX organoids fused to a patient-derived ganglion eminence and vice versa. And it actually looks like, as you suggest, that the mutation on the cortical side is also contributing to the hyperexcitability. So that's actually the topic of Sarah's project. I invite you to talk to her in more detail to see how we're thinking about addressing that.
Oh, thanks.
Thank you very much. That was very interesting. I'm curious, what's known about the nature of synapses in organoids, and in terms of both structure and connectivity between cell types? Does it mirror what you would expect in a native tissue?
Yeah, that's a really great question. And to my knowledge, very little is known. So in some of the more recent papers, people are using like the rabies virus and also optogenetics to look at connectivity. And I think that's really important. And we are also trying to adopt some of those techniques over to the organoid. So at this point, we know that we see this accelerated migration and it's correlated with this hyperexcitability, but is it because they're migrating and they're not forming synapses, or is it because they are, but there's some difference with the intrinsic properties? And I think having those tools could give us a lot more insight.
I know there are additional questions, but in the interest of time, I suggest we move on. So thank you, Jenny, again for your talk. Next speaker on the program is Natasha Jacobsen from the University of Copenhagen, where she's a PhD student working on iPSC cells in the context of a NALCN loss of function variant.
Hi, so my name is Natasha Jacobsen. I'm a PhD student in the PLAS group, and we're based in Copenhagen. And my project concerns establishing a iPSC-based human disease model for NALCN loss of function variants. So, yes. So I'm here to talk a little bit about IHPRF and loss of function variants. So IHPRF is a disease, rare neurodevelopmental disease caused by variants in UNC80 or NALCN. And these variants cut the protein short by introducing stop codons early on, or by introducing frame shifts or deletions. And this means that a lot of the protein will be truncated off. And in this case, this often leads to mRNA decay, nonsense-mediated mRNA decay, which results in no protein produced at all, or it can result in truncations of the protein, meaning that the protein might not be assembling correctly due to missing parts of the protein, or that the channel complex might not even be able to assemble. In both cases, this may lead to a lack of functional NALCN channels, leading to disease. So right now, there's no human disease model for IHPRF, and this is kind of where we want to explore iPSCs, and we want to look into the variant called Q642X, which is the first variant reported for IHPRF. And this variant here is located in between the D2 and D3 linker, and cuts the protein short by quite a bit, as you can see here. So to talk a little bit about the model that we are creating. So we are using CRISPR-edited iPSCs, using a well-characterized iPSC cell line we have integrated in the variant. And this means that we also have a variant cell line as well as a isogenic control, which is a healthy cell line. And we have validated these stem cells in terms of pluripotency and as well as in differentiation capacity, meaning that the stem cell lines are able to differentiate towards all three germ layers. So moving on, we have then differentiated these iPSCs to neurons using a NGN2 induced differentiation protocol, generating cortical glutamatergic neurons. These neurons have then been validated using immunocytochemistry. So once we have differentiated neurons, we can then go on to evaluate them, and we use electrophysiology to determine whether there is a phenotype in these neurons, and we evaluate both the passive and the active membrane properties. So now I want to show some of our first results from the model. So this is very preliminary, I must say. So we have successfully generated a control cell line. We have control neurons, as well as a cell, like a patient variant neurons as well. And we have positive stainings for beta-tubulin, which is a neuronal marker, as well as the GLUT, which determines that we have excitatory cells. After 5 weeks, we see that both cell lines are quite active and both fires in trains. And a bit unexpectedly, we see that the firing pattern seems to be quite subtle, not subtle, but I mean, they look quite similar. And at first glance, the phenotype seems to be quite subtle for the variant cell line. But if we look a bit further, so looking at the RMP, the resting membrane potential, we see similar RMPs around -50 for both cell lines. However, if we look at the action potential amplitude, there is some differences. So it seems like the variant cell line has larger amplitudes compared to the control. And if we look into the action potential frequency, we see that the control actually fires more action potentials compared to the variant cell line. So there seems to be some differences, even though the phenotype at first seemed subtle. I want to also tell that after 6 weeks, we see that the neuronal pattern might alter a little bit, but we need more data to actually say more about this. So just to sum up really quickly, we have successfully differentiated our iPSCs into glutamatergic neurons. This means that we have now a model where we can investigate NALCN loss of function variants, or the Q642X, which is active and fires action potentials using our protocol. What we see is that the phenotype appears quite subtle, and it appears to evolve a little bit over time. And this suggests a more complicated or complex mechanism of action. So we are excited to kind of explore the variant a bit further and figure out what exactly might be the course of disease. So just to make sure, we have established a iPSC-based disease model for investigating this variant. Yes, so lastly, I just want to tell my appreciations to the research group, so to the PLAS lab, especially Stefan and Nadine, as well as some collaborators.
Terrific. Thank you very much. Questions for Natasha?
else ask, I'll ask. And anyway, so I look at the current clamp recordings. It makes sense that amplitude goes up if the leak goes down. But on the other hand, the hyperpolarization seemed to produce very different effect. I wonder if HCN or other channels have been compensating for the loss of function of NALCN.
Yes, that's also something that we were excited to dig deeper into. So I think the next step is proteomics to try and explore if there might be any compensatory mechanisms. Yeah.
So the knockout neurons have been recorded by several labs. This is from mouse neurons. It's hyperpolarized by roughly about 5 to 20 millivolts. I noticed that the resting membrane potential of those differential neurons are very high. Is that the common amount? Is that comparable to those recorded from slices? Or is that maybe that's an issue?
I think it's difficult to compare iPSCs and slices. I must look a bit more into exactly how to compare them, but these are comparable to other iPSC models that I've seen in terms of controls. Yeah.
One last question.
I think this IPS, how long is your culture? I think, when we recorded the human iPS cells action potentials before, there's also like, they look immature. Like if you look at the actual shape of the action potential,
yeah,
and also the baselines and everything. So how long was your culture? Like if you monitor, how many weeks?
Oh,
month, yeah.
These are after 5 weeks. Yes, and we want to explore a bit like later as well, so go into 6 weeks.
Good. Thank you very much for your talk, Natasha. Good. That brings us to the last talk of the session by David Nagy. I don't know. So you'll have to tell me how to pronounce your last name. Nagy. Okay, there we go. David Nagy.
It's Nagy, but it's too complicated.
From Sophion Biosciences.
Welcome everybody, and thank you for the organizers to give us the opportunity to present this short presentation today. So instead of sharing some novel data of the NALCN channels, I would like to introduce a technology that can help scientists to record more data easier and quicker. And as Jeremy pointed out in his introductory remarks, that's key to have more data to inspire more collaborations in between academic and industrial research groups. So that's exactly what this technology is meant to be. So we heard it in several earlier talks today that ion channel studies have long used recombinant human proteins expressed in human cell lines, just like HEK293 cells, to screen ion channel modulators and to develop potential disease-modifying drug candidates. So these cell lines are very popular for several reasons. They are relatively easy and cheap to maintain and to work with. You can overexpress your channel of interest, although they have several limitations that I will discuss a little bit more in a later slide. So human-induced pluripotent stem cells also came up today a lot. Jeremy also mentioned that that's one of the main goals for the foundation, to have more of these human-induced pluripotent stem cell lines available from this disease. So they are expressing native complements of human ion channels and their accessory subunits, so they provide an enhanced translatability from early in vitro studies to patient biology. So these human iPSC cells encapsulate the signaling pathways, genotypes, and disease phenotypes better than the recombinant stable cell lines. So that enables them to define the critical ion channel activity in healthy and disease states better compared to stable cell lines. If you combine these cells with high throughput automated patch clamp technology, then that provides a foundation for significant physiological, medical, and drug discovery advances. So basically, the goal of this study in a nutshell was to test whether it is feasible to record these human iPSC-derived neurons or other cell types with automated patch clamp studies. As Eric mentioned, every cell line behaves differently, so it usually takes a little time to develop and optimize an assay. So we tested several different variables, so I'm going to share only a few that made a meaningful improvement in success rates with automated recordings. So many of you in this audience is familiar with the patch clamp technology, but for those of you who are not, the patch clamp technique is still considered the gold standard method to measure ion channel activity. This is considered a relatively old method. Neher and Sachman received the Nobel Prize back in 1991, so it's relatively old, but it's still the best method to measure ion channel functions. So this is how a typical traditional manual patch clamp setup looks like, and if you think if it is a little bit intimidating, then you are not alone. The first time I walked into an electrophysiology lab as an undergrad student, then I wasn't 100% sure that I want to be an electrophysiologist, but I am now. So usually a traditional setup involves dozens of different components. Just to mention a few, you need a pressurized air table to eliminate any vibrations, a large Faraday cage to filter out outside electrical noise, micromanipulators to precisely move your electrodes, analog digital converters, amplifiers, signal generators, and so on. It's hard to cover everything. But the basic of the technique is basically just use a glass pipette as your recording electrode. There is a reference electrode sitting in a butt where the steel adherent cell is sitting inside your recording chamber. So one of the most important criteria for a clean and high quality recording with patch clamp is to form a strong seal in between the tip of your glass electrode and the plasma membrane of your cell. If there is not a good seal, then there will be some leakage that will be a bad quality, noisy recording. So, as you can imagine, this technique is very labor intensive. You need a very highly trained electrophysiologist to perform these recordings. It's low throughput, so even a very good electrophysiologist can produce probably 10 to 15 data points in a good day. And as a consequence, it's very time consuming. So the solution is automated patch clamping. You can record way more cells with much shorter time and with ease. So how does it work? This is a schematic cross-section of one of the recording wells in our consumables that we offer. So basically we have microfluidic flow channels. Two of these tunnel-like flow channels are sitting on top of each other. The bottom flow channel will include the intercellular solution. The top channel will have the extracellular solution, and then we will add the cells on the top channel. The two channel is connected by a patch hole, so that replaces basically the glass pipette that we use in the traditional method. And there are built-in electrodes that can measure basically the ionic currents flowing through the cell that will sit on the patch hole. So it seems that this 5 minutes was really, really short. I have a poster outside, so if anybody is interested in more learning about this technology or how we optimize the assay for iPSC-derived neurons, then we are happy to take more time at the poster or at our booth outside. So, but I will just jump to the conclusion very quickly. So basically, we demonstrate that we are able to measure the fundamental neuronal properties obtained with automated patch clamp systems. We drastically increased throughput relative to manual patch clamp recordings. Yeah, that's basically the take-home message.
Terrific. Thank you very much. Then I suggest, in the interest of time, I suggest that you take your questions to David's poster. And I would like to take this opportunity to thank all the speakers of this session one more time. And with that, I'm going to hand the mic over to Emily and Jeremy. Whoever of the two wants to take.
Thank you. And thank you all for amazing presentations today. Next, we have something a little bit different. We're calling it a workshop where we want to sort of take what we've talked about already and align some priorities for the research community, keeping in mind our patient community and everything we've talked about so far. So what I'm going to ask is that if you are sitting at a table where you have fewer than 5 people, please move to a different table so you have people to talk to.
This part's meant to be fun, so come on up. We'll get folks chatting. You can have more than 5. It's not limited to 5. That's just a minimum. So come on up. Make friends.
All right. Looks like we have some pretty great groups. And now we're going to spend the next 15 minutes, and hopefully our timer can help keep me honest on that. And we're going to talk about what we know about NALCN biology. We're going to talk about what we don't know, where our gaps are, and then we're going to see if we can identify any priorities for where we need to go next. And I'll be coming around and handing out some Post-it notes. And with their pens on the table, you can write what you know, where the gaps are, and what you think the priority should be on the Post-it, and we'll cycle them up onto here, and then we'll mix our groups up, think about the same questions with some different people, and then try to come to a consensus about where we need to go next. Right? So start with some introductions, and maybe with what we know based on what we've talked about already today.
And as Emily said, so really focused on the strengths and then what are the major gaps, and then the priorities, and I'll specify really over the next 2 years. What do we as a community want to prioritize, and saying these are the key things we need to try and get done to be able to move the basic science forward towards being able to eventually advance towards treatment. And so we'll post them up here, then we'll have you regroup and we'll rediscuss and we'll go from there. And our goal is to eventually publish this as well, and just sharing this is where we are as a field and where we need to go going forward. So thank you all. And you're on the clock. Yeah, 15 minutes to discuss these 3 questions and write down. You can have as many Post-it notes as you want for each topic.
Same result like their resistance, but one was non-small cell lung cancer, but their resistance is GFR. A lot. What is going on? Oh, I mean, I don't know. Let's run some more.
However much you discuss.
Did you do my writing? Oh, that's great. I was really concerned.
Yeah, exactly. You're not around here with it's blowing out of the water. And it's so
Got non-traditional roles.
Up there, screening. Yeah, sure. Yep, these are all gaps. There's a lot of gaps. The further you get into the career, the more you're supposed to know, the less you're going to know. We took it serious. You can call me Eric. It's okay. Yeah, I just attended their meeting. They say the lowest line is the 9,000. That's what they love it. I'm not surprised by that. All right, let's try to wrap up our thought. Try to move on to the next section. I think it can modify the cell. You have the cell that where you're at, and like create a state where they can so we did that model, that's what was going on. Talk about that. Blown? No, I haven't. All right. Has a pen reader added to our board? Right, so it follows off of this all right. What can you tell? There's a lot of gaps. We got a lot of work to do, which is great because there's always more questions to ask.
So I've been this year alone in 7 national organization meetings. There's a lot now, given the relatively small number of identified patients. I think the ratio is actually from existing knowledge from basic science to years or decades of it's incredibly more than statistics. But I would say that this is more coordination, more focus there. But no, we need this work. This is misrepresentative about what is really present.
Well, thank you for that. And we'll when we re-swap, we'll focus on what's known a little bit more too, and the priorities as well. And I think your insights as well as moving towards therapies is really important. How do we channel what is known from the basic sciences to address the next gaps that are needed from the basic science standpoint to help us translate those into therapies and clinical advancements. So I'm just going to read off some of these here, and then we're gonna ask you to keep brainstorming, and then we'll move you around and reshuffle, and then have the same questions and discussion. And if you feel like the gaps are already addressed, great. We'll say maybe spend more time talking about the knowns and the priorities. Really, the priorities are going to help us to make a plan for the next 2 years and beyond. And so far, for the known, that we know that it impacts multiple systems, that it's highly conserved, that there's many and diverse partners, contextual. Maybe I'll ask whoever wrote that one up to clarify that a little bit more.
Yeah, we're saying that maybe based on different cell types in the body, that the complex could have different binding partners. Like in some cell types, I don't see UNC79, UNC80 present at all, but the channels will function. So that we'll have other binding partners. That's really great. And some of the work that we did earlier with BioID, that was fantastic. That there is more diversity that we appreciate.
So many proteins involved. And then understanding CLIFAD, IHPRF 1 and 2, pain, cancer, sleep, hypertension, so understanding the multi-systemic impacts from the basic science standpoint. When we move over to gaps, so we have understanding some of the gaps, understanding the specific pharmacology in the assay, PHTS? HTS, I do have training. Okay, got it. Under the variant characterization, so better characterizing the variants, and we had a discussion at our table about that as well, identifying representative variants across the spectrum, and really focusing on for those representative variants, whether it's strong gain of function, whether it's a clinical phenotype or otherwise, starting to have models for those variants across having for the same variants, seeing them across various disease models, whether it's textile models, iPSC models, or otherwise, for those representative variants, so that we can have.
Maybe just a comment on that. This is maybe overstepping my area of expertise. My understanding is that there are patients who have the same variant, but they are displaying very different symptoms. So I think just having a few different ones is a great starting point, but it will not get us there. Even the same variant gives very different phenotypic experience.
Absolutely. So to that point, a gap is understanding the phenotypic variability. And why is that when somebody has the same variant in different phenotypes? Early diagnostics in interventions, I'll go there with biomarkers for the diagnostic and really getting both genetic testing but then biomarkers as well. Gene therapies, absolutely. More focus on delayed current versus decay burn versus activation of current, if I'm understanding that one correctly. Mutations, known and unknown. I'm going to ask for whoever wrote that one to help me understand that one as well.
That was me. Apologies. Does a mutant channel have altered binding partners? So, is the same or have other binding partners when different variants are present that might not affect its gating ability, but other roles?
Sure. And thinking as well about the various downstream modulators and other factors as well. Specific targeting of mutations, phenotype disconnects, hormonal regulation of channels, their circadian impacts as well when doing experiments and otherwise. Subcellular localizations and context interactions. That there's limited research tools, limited quality research tools. And so I'll ask for this one to think about what are the tools that are needed most, what would be most helpful as to prioritize over the next few years. Talking about contextual compensation and function of other channels in relation. Gap pharmacology, so a rational approach to finding NALCN-specific blockers and modulators. When during development does the disease phenotype arise? Understanding the patient variation, so that heterogeneity that we were talking about, as well as the therapeutic window for when to intervene. Clinical trial inclusion-exclusion criteria, absolutely, in disease mutations of patients. Exploring various transcription factors, so going to those regulators as well, the upstream and downstream effects to see if there's targets there for treatment. Isoform and splicing of the channel, screening FDA-applied drugs and generating selective tools to probe selective pharmacology tools. Maybe whoever wrote that one to help. Okay. Sure. And then screening drugs to help filter out the main ones, and why gain-of-function mutations comprise a broad range of clinical manifestations, so that broad clinical phenotypes.
We're going to leave the priorities for now because we're going to get into them a bit more. So nominate half of your table to move to the next group counterclockwise.
And what we'll ask is for each known gap or priority to try and have them each on a separate sticky note, because we're going to vote on that. So have one per sticky note.
I'll go to the next table. If you want me to go, I can go.
All right, I'll start.
Some great power I should finish that. Thank you.
This table has no money left.
We have to take
At least one more. At least one more. Okay, so back.
If we lose a group here.
Maybe they just got it.
Yeah, it's okay. Come on here, this looks like a great table. Sit down, chat, post it. We have another 15 minutes to talk.
Priorities.
Yep, so all of these again, but maybe we have a lot on the gap. There's no gap or perhaps, so feel free to add them. But if you feel like I covered it already, I'd focus more on where our priorities go. That's a priority.
Desi, no. More problems. All these variants are popping up. I don't know if lots of children have which could be timing during development. So when were these acquired? With our blocker answer. We can come on the same. That's what I was hoping to pitch. I hope it came across while that's what we want to look at to see the related statements. Yeah, so I tagged with the GOP. Yeah, absolutely. That's why we can talk about this. Yeah, absolutely. Oh, so we have a basic part that is like native state, general native environment, native environment. Yeah. Cellular subset of the organism. Spatial biology. No, on my list. Use the army skills on various other fields. It seemed like yeah, right. So it's like one or two learning some data that they were proposing. I thought about me. Level barely detect one of the brain. This whole is just constantly translating off of there. But all right, I'll go ahead these up to our yes,
we like to have a lot of
No, I'm making sure I can read them this time. Yeah. No more blood tests. Got it. Okay.
Oh, yeah, okay, so. Now he's making. Yeah, no, it is.
We did that, I held a workshop this past summer on the past session, exactly like this. Literally the check. Find the common lines where
Well, no, I couldn't, I had to cancel. But I was invited. I missed.
Yeah. Great food.
Okay, that is good. It's cheaper than we thought.
Yeah, let's get the prices. Yeah, exactly.
What are you?
All right. Thank you all for chatting. We're going to go ahead and get started. So part of our goal here is really to try and understand now and to discuss and to think about as a group and really to organize our thoughts here. And we'd like to, as mentioned, be able to put this in writing going forward. And I'm going to start with what's known. What are our strengths? And what are really our selling points when talking to industry and trying to share? This is some of the that we already have available. So I'm going to bring up what's been talked about and then potentially open up for discussion as well. Just as a heads up, so after we go through all of these, we're going to give everyone stickers. And we're going to ask you to vote on which of these to you feel is the top known, to vote on the various stickers in each category. And so think about it as we're going through these. So we're going to start with what's known. So as mentioned before, that we know that NALCN impacts multiple systems, that it's highly conserved across species, that there's many and diverse partners involved in the channel functioning. We know about its role both in neurologic diseases as well as more systemically. That there's minutes for the functional NALCN complex. That we know that both gain and loss of NALCN causes disease. So really that we know in many ways that haploinsufficiency is tolerated as well. I'll just mention that heart rate monitoring as a diagnostic for disease in prenatal stages, so as one potential biomarker. That there was no signature REM sleep observed in babies with CLIFAD. We know that NALCN UNC80, UNC79 knockout mice die. That there's potential diagnostics in utero, including sleep validation, as well as the heart rate monitoring that we just talked about. And that we know that gain of function is well characterized and that the tests are available there. What else do people feel is known? What are some of the strengths that we know about NALCN and the related diseases? So there's a lot known about the structural biology of the channel that's valuable for therapy development. So I'll throw that up here as well. Okay, so we know it has link channels as well. So voltage gated and Stefan, I'll ask for your help to help write down some of these as we're talking about it. Sorry. We can swap off. Mm-hmm. So multiple potential pathways involved that missing targets. Yeah. So I'm going to try and rephrase that. So that there's representative clinical phenotypes and diseases that are known, and we understand more about the underlying neurobiology from those. So we have at least 4 syndromes and diseases that are known. Any other kind of key knowledge strengths that anyone wants to mention? Oh, so that's helpful. So there's a known and well-characterized target, therapeutic target for the diseases as well. Great. And so now we're going to move on to gaps. And so I'm going to start at the bottom this time. So some of the gaps are things that we don't yet know. These are things that the scientific gaps that we'd like to look forward to. So generating selective pharmacology tools to better probe the channels. Understanding the isoform and splicing of the channel, better understanding why each genetic, the clinical heterogeneity, even within those with the same variants or those with the same diseases, so understanding the genotype-phenotype correlation better. Understanding whether a mutation is pathogenic or not. Screening drugs, and having methods to do so, and modalities, identifying modalities that are optimal to help filter therapeutic candidates, exploring transcription factors, screening for approved drugs, understanding, as mentioned, that patient variation, as well as optimal therapeutic windows, identifying when in development the disease phenotypes arise. And I'll share much of that is known, as we talked about already. It seems in utero, there's already signs for the disease phenotypes. We see that in terms of musculoskeletal manifestations, some of the heart tracings, as well as for the sleep phenotypes. Pharmacology, so having a rational approach to find NALCN-specific blockers and modulators. The contextual compensation function of other channels and the roles those have in the interaction of other channels with NALCN. So that could be of interest for the RARES1 network and for the channelopathies as well. Research tools, and particularly quality research tools. Subcellular localization in context of interactions. Understanding the circadian impact when performing experiments. Hormonal regulation of the channel, clinical trial inclusion exclusion criteria, and disease mutations in patients. As mentioned, that phenotype-genotype disconnect, specific targeting of mutations, not just the native wild type, so having more tools that target mutations and variants themselves as opposed to just wild type. Mutations, I think I got stuck on this one before as well. Mutations that are known to unknown in trying to understand the mutations further. The specific pharmacology and high-throughput screening assays, variant characterization, early diagnostic tools for intervention, therapies, and more focus on current versus activation of the current. Any other gaps that we're missing? Oh, it's a great point, and I appreciate Stefan summarizing it much better than I could already. What are the effective variants on other proteins and genes as well, and really understanding those interactions and potential interactions and the implications of that. Absolutely. So biomarkers as a gap in proteomics, I guess, as well, and thinking about the multiomics as well for those, and talking about some of the other effects of variants on other genes and other the expression of other proteins. Oh, no, and so I think to that. Yeah, so further characterizing the broader implications of NALCN and the benefits of targeting NALCN as therapies. We heard earlier today about sleep. Sleep is one of the biggest. NALCN as a potential role in sleep would be huge. Cancer as well. But really trying to hone in on the underlying biology and role of NALCN in broader and more common diseases as well. Uterine contractions as well being another key point and therapeutic target. And so thinking about further refining that neurobiology, pain as well. And so for the anesthetic uses, and so that would be a key one. Hypertension would as well. So really for each of the more on the underlying biology. Okay, so now going into priorities. There's a lot of priorities listed here. We can't do, I would love to say we can do it all in 2 years. So I'm going to go through them. And then what we're going to ask is afterwards, as Emily's been handing out, you're going to have 8 stickers. We're going to ask you to come vote on the priorities, the gaps, the known, to your judgment. You have 8 votes. You can, I don't know, this keeps coming in and out, it sounds like. Sorry if that's happening, or if I'm just hearing it. But you have 8 votes across the stickers, and we'll use that to identify our paths going forward. So going through the priorities, so deep characterization of phenotypes, again, going to that genotype-phenotype analyses, but understanding the clinical phenotypes. More focus on the non-traditional roles outside of, outside of current. Am I reading correctly? Okay, awesome. So similarly, oh yeah, this sounds better, thank you. Talking about the role outside of the currents, and I think to the same point outside of maybe more systemic diseases as well. More robust genotype-phenotype studies, bucketing patients into groups for relevant disease treatments, so really trying to hone in on those phenotypic groups. Biomarkers, better characterization of the channel and the channelosome in the native context. Unlimited funding, wouldn't that be nice? More clinical perspective. Identifying representative variants to study across the phenotypic and genotypic variability, and having models within those representative variants across multiple systems, and so different models. Endpoint and outcome measures. Increased research collaborations related to NALCN, AI-based structure-based drug design, additional regulators of the proteins, newborn blood test and blood spots collected, and I know there's some work in thinking about that for genetic diseases as a whole. So really understanding specific drugs that can help to inhibit or block the channel at varying degrees, so partial inhibitors, as well as activators or enhancers. Improved clinical diagnostics to identify cases, as well as prognosis and progression, so prognostic biomarkers. Developing reliable drug screens for assessing drug repurposing. Better understanding the basic spatial biology and the assembly of the channel in native state and in various tissue, subcellular and other spaces. For gain-of-functions variants, first consolidating knowledge about the gain-of-function effects, and then understanding the N-of-1 approach to better understanding the variants for gain-of-function. Pharmacology, AI, structure-based, I can't read the middle one, but we'll take it as such. Patient priorities, genotype-phenotype correlations, better understanding the dynamic structure and representative patient mutations as well. So really identifying what are representative variants to really focus studies on. And then a lot of the things that were talked about throughout was repurposing, collaborating, community, and engagement. Any other priorities that we haven't talked about yet that anyone wants to add to the list? Yeah. What I'm going to say is if you see similar ones, just vote for one, and we'll consolidate after the fact. So if there's multiple ones for genotype-phenotype correlation, if you vote for one, you're voting for them all. And so. Yeah. So the meaningful outcomes for patients as well. And I think we'll have the chance to talk more on that tomorrow as well in trying to identify that. But that is a key priority and gap.
Can I suggest 3 priorities?
Absolutely.
Instead of 8. It's really basic science priority, translational science priority, and analytic science priority.
Absolutely. And that's our exact goal here, is this session's focused on the basic science priorities. Tomorrow, the session will focus on the translational and clinical science priorities. So with exactly that goal in mind, is trying to identify here today, what are the basic science priorities for us to focus on. So we have a lot of ideas just for the basic science one so far. So thank you all. You've been amazing. Please come vote, take a break. There's coffee in the back. All your registrations bags should have snacks. Unfortunately, we couldn't afford all the snacks at each session, so we just gave them to you at registration to eat throughout the conference. So those are your snack packs for the conference. And we'll resume again at around 4:10, unless Emily tells me otherwise. Okay, and we'll go from there. So please come up and vote. You can do so privately and anonymously, and we'll go from there. So.
Hi, everyone. Welcome back to the second part of the translational science session. I'm Chao. I'm taking over the moderator role from Stefan because he's too tired and needs a nap. So we're going to start this session with Manu Benjoni from Columbia University. Unfortunately, he's sick, so he's not here with us today, but we'll be playing a video of his talk on developing nanobody-based inhibitors of NALCN by targeting an intracellular domain.
Hi, everyone. I hope you're all doing well. I'm very sorry to not be able to join you in person today as I had originally planned. I picked something up a couple of days ago and I've been unfortunately a little sick. Still, I'm very glad to have this opportunity to share with you a brief update on our work on NALCN channels. So every thought that we have and every action that we take begins as electrical impulses or action potentials generated by neurons. Classically, we think of each of these impulses as starting with the opening of sodium channels or voltage-gated sodium channels, which lets in sodium ions and depolarize the cell. And then the potassium channels, which when they open, they let potassium out and the cell can repolarize. But this basal potential, or the resting membrane potential, is set by both potassium conductance primarily and a little bit of a sodium conductance through the leak sodium channels. And this resting potential has an enormous consequence in terms of cellular function. If the resting membrane potential is too low, it's harder to fire an impulse. And if it's higher, it's too easy, or if it's too high, then the whole system shuts down. And so it can have a profound consequence. The basal sodium leak current is mediated by the NALCN channel. And this channel is made up of, at the minimum, 4 subunits. There's the pore-forming NALCN subunit, as well as auxiliary subunits FAM155A, UNC79 and UNC80 subunits, all of which are essential for its function. And as we've heard about, genetic variation in NALCN results in diseases, including CLIFAD, which results from gain of function variance in NALCN, where the leak sodium current is increased, or IHPRF1, where you can get a profound loss of the leak sodium current. And for many ion channels, there are a wide range of pharmacological agents that are available that can often inhibit the channel or even activate them. Unlike the related voltage-gated sodium and calcium channels, the NALCN channel, there are very few pharmacological agents that are selective. And so there's an urgent need to develop new pharmacology targeting NALCN channels. And we also know that a complete lack of NALCN currents is detrimental. And so genetically encoded approaches to reduce NALCN currents, potentially in a cell type dependent manner, may also be beneficial. And this is what we set out to do. So if it's not easy to just block ion flux through the pore, how else can we control these NALCN channels? And one idea that we had is inspired by one of my colleagues at Columbia, Dr. Henry Colcraft. And the idea is this: you may not need to control the ion flux through the channels, but what you can do is control the abundance of the channels at the membrane. And actually, the cells do this naturally through the ubiquitin system. When membrane proteins are modified by ubiquitin or polyubiquitin chains through E3 ubiquitin ligases, these channels get internalized. And then you can naturally reduce the amount of currents. And so the idea is this: if we can locally recruit E3 ligase enzymes to the NALCN channel, then we could add the ubiquitin chains and we could retrieve the channels from the surface membrane and reduce NALCN currents. And of course, it may be possible to also remove ubiquitin and stabilize the channels at the membrane, and it could work the other way just as well. And if this works, it gives us the motivation to try to develop molecular glue-like approaches that so that we can do this with not just proteins, but also with small molecules. And so the first task is to find a way to get the E3 ligase to be localized near the channel. And to do that, we wanted to use a nanobody, and nanobody that could bind to the NALCN channel. And nanobodies are single-chain antibodies that come from camelids like llamas. They're small, and they can be expressed recombinantly, and they can be engineered to have E3 ubiquitin ligases or other kinds of enzymes, which will allow us to modify or potentially modify the function of NALCN currents, NALCN channels. Okay, so how do we develop nanobodies to target a domain of the NALCN channel? And to do this, we used a synthetic yeast display library. And so we first purified a part of the carboxy terminus of NALCN channels. We tagged it with strep tag. And then we use this yeast display library to pan for nanobodies that potentially bind to the NALCN carboxy terminus. And so we do multiple rounds of screening through magnetic cell sorting or FACS. And through each round, we can enrich for nanobodies that potentially bind to NALCN. And so each dot here potentially represents a yeast cell that is displaying a nanobody that could bind to the NALCN channels. And ultimately what we can do is to select a subset of these and do either single cell sort or identify individual NALCN binding nanobody clones. Okay, and so we did that, and we identified a number of sequences for nanobodies that could bind to the NALCN carboxy terminus. And to test whether these nanobodies that we identified do indeed bind to the NALCN channel, we used a FRET2 hybrid assay using a flow cytometer. And so the idea here is that we tag the carboxy terminus of the NALCN channel with a fluorophore. And we also tag the nanobodies with another fluorophore. And if the two bind or they come together, then the fluorophores are going to be in close proximity, and they're going to undergo a process called FRET, or fluorescence resonance energy transfer. And then we can quantify the FRET signal and try to get a sense of relatively how well the different nanobodies interact with the specific channel domain. And we did this for a number of nanobodies that we identified. And what we saw is we found a sort of variable relative affinities for the various nanobodies that we identified. And so we went forward with two of them, E8 and B12. And so if you look at the full channel, do these nanobodies still interact with them? And to test that out, we again used a FRET assay. So here we tagged the full length channel with the venous fluorophore, and we attached cerulean onto the nanobody, and then co-expressed them and looked for FRET. And you can see if you just had cerulean and no nanobody, you would see no signal. But when you have the nanobody, you start to see FRET. And both E8 and B12 show binding to the NALCN channel, the full-length channel. If we know that all the auxiliary subunits are important for the function of NALCN, and so we wondered if you express that, would this nanobody binding be altered? And what we saw is that the nanobody still binds to the channel, the full channelosome. And so it seems like a fairly good candidates to develop functional modulators. So how do we study the function of these channels? And to do this, we reasoned that it would be convenient to have all the NALCN subunits in one single plasmid. And so we generated this mega plasmid that includes all four NALCN subunits so that we can transfect it into HEK cells and potentially obtain stable expression of NALCN currents in these cells. And this would allow us to functionally characterize these channels robustly. And so we did that. So if you take and it turns out the plasmid, this megaplasmid itself has a convenient flip recombinase site. And so we can put it into a flip-in HEK293 cell line, and this will allow us to generate stable cell lines expressing NALCN currents. Okay, so if you look at empty flip-in cell lines, there's very little basal leak current. And if we so we first start by patching these cells with extracellular 1 millimolar extracellular calcium. And if we deplete the extracellular calcium by adding EGTA extracellularly, then you see that this leak current doesn't really change significantly. However, the NALCN knock-in line, we see that when we deplete calcium, we get a large increase in the leak current. And this is presumably mediated by the sodium leak channel. And so what happens with our nanobodies? To study this, we expressed NBB12 first by itself in the flip-in line. And what we saw is that this nanobody alone could actually reduce the NALCN currents. Now, going back to our idea of using the ubiquitin ligase, what we see is if we attach a NED4L E3, the catalytic domain of the NED4L E3 ubiquitin ligase enzyme to the NBP12, we see that it can mediate an additional reduction in the current. You virtually go back to kind of the background leak current in these cells. We see something similar with E8 nanobody as well, except that the E8 nanobody by itself inhibits the currents quite strongly. And we can again get a small increase in the inhibition, but it seems to by itself be an inhibitor. And so in summary, we have used a yeast surface display platform to identify a nanobody that binds to the carboxy terminus of NALCN. And our functional analysis shows that these nanobodies can, in fact, bind to the NALCN carboxy terminus, and it can inhibit the leak sodium current by themselves to varying degrees. But also we can then recruit E3 ubiquitin ligases by fusing them onto the nanobody, and this will allow us to further reduce the NALCN currents. And some of the future direction, and so you can see the kind of variable degree of NALCN inhibition attained by the different nanobody modulators here. And so the two interesting future directions we're thinking about is to probe the efficacy of these nanobody modulators in sort of cellular models, and to see if they could reduce the changes in basal leak sodium current. And also we're starting to think about how we can extend this work to engineer small molecule analogs. So in the last minute, I want to acknowledge some of the people involved in this work. Really, the star here is Alan Shu, who is actually in the audience. And Alan has really single-handedly taken this project
a drastic loss of current. On the opposite, we have a drastic increase in NALCN current, so we're interested in deciphering this a little bit. Yes, and I'm focusing, as I mentioned, on the gain-of-function Clifford variant. That is mostly the de novo dominant gain-of-function variant. And it is located in the D3, D4 linker. And it is very interesting to know maybe that it's a very prevalent variant. Yes, so basically last year we had 3 very brave patients coming to Denmark to visit us and to contribute to our study. And they are from Germany as well as from the UK. And they donated us some blood. So basically what we did is we choose to use human-induced pluripotent stem cells, or iPSCs, as disease model. And we took blood from the 3 patients alongside with a healthy control, which was their siblings or a parent. And we harvested and isolated peripheral blood mononuclear cells, or short PBMCs. And then we reprogrammed those 2 iPS cells to get them to a pluripotent state so that they can potentially be differentiated into all sorts of different cell types. We moved forward to differentiate those 2 glutamatergic cortical neurons, and then we characterized those both electrophysiologically as well as with immunostainings. Yes, so the first thing you always have to kind of show is if you reprogram some cells to pluripotent states is that they're actually pluripotent. And we looked at different markers for that and did some staining. So we can actually show that for these 2 nuclear stainings, they are positive in terms of pluripotency, as well as for 2 transmembrane markers that are usually looked at when you reprogram cells to pluripotent state. So this is true for both the R1181Q for all the patient families, as well as all the controls from the relatives. Then we also have to show that they can actually, or that they have the capacity to differentiate towards different germ layers. And we can as well see here that they are possible to differentiate to endoderm, mesoderm, or myofibroblast, and also to ectoderm. This is obviously the most interesting for us as well, since we want to look into neuronal cell types. Yes, and in terms of neuron markers, we differentiated towards the glutamatergic neurons, which we can show here that they're positive for VGLUT, glutamate transporters, so they are indeed excitatory neurons. And we also looked for beta-tubulin as a neuronal marker, and they seem to have all the different markers present, both of the R1181Q and the controlled cell lines as well. Yes, and then we looked into electrophysiology. Basically, this is how they are looking like after 3 weeks. Nothing much going on here. However, after 4 weeks, you can see that the controls start firing action potentials. However, the R1181Q is quite silent. Then if we look after 5 weeks, however, we can see that there's quite a bit of change in current, in excitability. Basically, they are firing quite a lot of action potentials. So in the beginning, they are quite hypo, and then after, quite hyperexcitable. So if we look a little bit more into the details here, we can also see that that's reflected in the number of action potentials that are fired per injected current after 4 weeks. Then complete change here after 5 weeks. And you can maybe appreciate that we also don't really face any depolarization block in a lot of these 5-week-old R1181Q cells. This is also true for the average number of action potentials, basically. When we look on average, we have to switch from 4 to 5 weeks. Then, so far, the data that we have, I should stretch that these are quite preliminary. We only looked into 2 of the 3 different patient families for now, so the first one is going to come up soon. But yeah, you can appreciate that basically they are very similar between the 2 patient families. Then we looked as well into the average action potential peak values. And as you can see here is that after 4 weeks, we have lower peak currents p-values, sorry, which makes sense since we also have less excitability. And after 5 weeks is pretty much the same. The same is true for both the different patient families that we looked at. Then we also looked into passive membrane properties, for instance, the resting membrane potential. And you can appreciate here that after 4 weeks, the cells are pretty much depolarized compared to the control. However, after 5 weeks, they kind of catch up and have similar resting membrane potentials. Yeah, again, the same is true. It's pretty similar between the two patient families that we've looked into so far. Then we also looked into input resistances. And what we can see is that in the beginning, it seems like R1181Q after 4 weeks has quite lower input resistance. So basically, we use that as a measure for excitability. So if they are having like a lower input resistance, that means that the current that we're injecting leads to smaller changes in voltage. So of course, it's easier to cross the threshold if you have a higher input resistance. So that makes sense since the cells are also less excitable overall. And then after 5 weeks is pretty much similar. Yeah, and then again, you can appreciate it's pretty quite similar between the two patient families. Yeah, then we also looked into holding currents, and what you can see is that R1181Q displays massive holding currents compared to controls, and that is true for both 4 and 5 weeks. So that basically means that if we hold them at minus 70 millivolts, for instance, we can see that there's quite a lot of sodium leak into the cell, probably. And that is also true for both the patient family 2 and patient family 3. So as a summary, if we look at the action potential numbers, we can appreciate that between 4 and 5 weeks, we have some sort of change. In the beginning, we have lower amount of action potentials compared to the control, whereas after we have quite a lot of excitability in the R1181Q. The action potential peaks are a bit lower after 4 weeks than similar after 5 weeks. The resting membrane potential, so in the beginning, after 4 weeks, we can see that they're quite depolarized. After 5 weeks, however, the resting membrane potential is pretty much similar. Input resistance is first a little bit lower, then afterwards similar. The holding currents are always quite increased, no matter at which week we looked at. Yeah, so as a conclusion and outlook, we can see that R1181Q can kind of result in two very different looking phenotype. We can see that there's a change between 4 to 5 weeks. So basically, it could be that they are just too depolarized to actually fire action potentials after 4 weeks, and that's why we can't really see anything going on here. I mean, that also makes sense since if you look here, we have this hyperpolarizing pulse in the beginning, and then we get a little action potential here in the end. So basically, if we force it to be a bit more hyperpolarized, the cell can actually fire an action potential after. However, so it seems like it's just too depolarized to have action potentials. However, after 5 weeks, it looks like if there is some compensation happening, it kind of is just too much, and the cell gets more excitable or more active compared to the control. So yeah, for understanding a little bit more differences between the weeks, we plan to do proteomics and look into if any sort of potassium channels or anything is upregulated, or if there's other proteins that might compensate. Then the other explanation could, of course, be that the R1181Q just makes neurons like mature much slower, and that's why they just start firing after 5 weeks instead of 4. So for that, we want to do some stainings, and we want to look a little bit more into detail into the morphology. And then we also plan to do some modulation experiments where we do some pharmacology, of course, and try to rescue the phenotype and get back to wild type. Yeah, and then thanks a lot for your attention. Thanks a lot to my working group and to all the patients that contributed to the study, and to the collaborators that made it possible for us to do all of this work, of course, and funding. Yeah, and are there any questions?
Thank you, Nadine, for a great talk. We have time for a couple of questions. So does anyone want to? Oh, sorry. Sorry, I didn't mean to interrupt. Any questions?
So for in the 5 weeks of the iPSC-induced neuron is a point that there's this critical sort of transformation that AIS become very important a part of the spike jet initiation. I wonder if you have looked at that, some labeling to see if that AIS somewhat is different between these two genotypes.
We haven't looked at that yet, but it would be very interesting to look at. Yeah, it's a good idea.
Any other questions?
I have a quick question about the change between weeks 4 and 5 or something. Is that a change of the current recorded also coordinating with the change of behavior?
I'm not sure if I get the
So
question.
you also recorded currents, right? The mutant gives you much bigger current. Was that recorded at the same time when you see difference in firing, or the time when you don't see difference?
So at the moment, we don't have enough data to support that in the voltage clamp studies that we did so far, but we will try to look into it and see. And yeah, but in principle, it's hard to correlate these exact time points to any major things that happen in development, since we use like a forced expression as well. So it's like, I mean, we look at this time point by chance in the beginning, right? So it could also be that we miss out on things.
We have time for one more question. Anyone? Oh, okay.
Did you measure the holding current level at minus what millivolt did you measure?
Sorry, I didn't get the question.
I mean that the holding, I see that the input resistance is decreased in 5 weeks after maturation, but I feel that some potassium current must be more expressed at 5 weeks.
Yeah.
And however, I'm wondering, so at 5 weeks there is some decrease in the NALCN current, or it's just because there's some potassium current compensated for the inward current. That's why I'm wondering why the inward holding current was decreased after 5 weeks maturation. So I'm wondering that's why if you measure the holding current at the voltage of potassium reversal potential, it should be we can tell the change in NALCN current, but that's why I'm wondering what voltage did you measure the holding current level?
Yeah, so basically it's very possible that there's like more potassium channels or something that are expressed after that time. We don't know that yet, so that's why we want to do the proteomics. And yeah, the change in basically the change in holding, is that true? The holding current that you were pointing out, yeah, that's the same between 4 and 5 weeks. So they have the same amount of big amount of holding currents. So it looks like there's probably more. So it could be more sodium inwardly, right? But we didn't look into that into detail yet. It's very preliminary data. But yeah, it would be interesting to look into that as well. And then for the input resistance, that one is basically decreased in the R1181Q after 4 weeks. So we think that that's just because they are less excitable basically at that time point, and then we have less changes voltage per injected current. But then it seems to be similar after 5 weeks, so it's probably different protein sets that are upregulated or something like that, but we don't know. We haven't confirmed that.
Thank you, Nadine. Let's thank Nadine again for a great talk. So the last speaker of the session is Tien Du from the University of Toronto, and he will be talking about the same variant and how it affects cerebellar Purkinje cell activity.
Okay, thanks. Firstly, I want to figure out how to use this.
You go forward with this, and then that's the pointer.
That's the pointer. Okay, thank you. Okay. Good afternoon, everyone. My name is Tien Du. I'm a third-year PhD student in University of Toronto, supervised by Dr. Mason and Dr. Liang. Today, I will be presenting our unpublished study on the effect of NALCN pathological mutation on the excitability and activity of cerebellar Purkinje cells. I think that is the previous version of the presentation. Sorry. Hello? Sorry, is Emily here? That is still the previous version. I think I sent a version this 3 p.m. Can you find this? That's okay. Yeah, V2. That's okay. Yeah, okay. Yeah, that is the one. Sorry about that, making a lot of last minute change. Okay, yeah. As we hear from the previous talk by Nadine, CLIFFA is a autosomal dominant neurodevelopmental disorder that is caused by the gain-of-function mutation in NALCN, with R1A1Q being the most common CLIFFA mutation. Previous study in our lab used a HEX 293 cell expression system. We find out that the R1A1Q can increase the inward lead current of NALCN, as shown in the magenta trace compared to the black trace of the wild type, and having a peak inward current at around -40 and 60 millivolts. Consistently, in C. elegans, by CRISPR knock-in of R1A1Q, we are able to induce the worm to exhibit an exaggeration of body curvature during locomotion, which is a phenotype we call coiler that is shared among the NALCN gain-of-function mutants. CLIFFA causes a range of symptoms, and many of them point towards cerebellar dysfunction. Many patients exhibit speech impairment, gait ataxia, and some of the patients exhibit the cerebellar atrophy. As we can see, the cerebellar volume of this patient shrink within the 4 years of development. However, the underlying mechanism of this cerebellar dysfunction is still unknown. To study this, I focus on type of cells in the cerebellum called Purkinje cell. This is a type of cell that is important in integrating the signals in the cerebellar cortex and serve as the only output of the cerebellar cortex to innervate the cerebellar nuclei. The impairment of this type of cells is known to be associated with ataxia and atrophy in patient and mice model. Electrophysiological study on these cells reveals that it is a tonically active neuron that is able to fire action potential even without any presynaptic input. As mentioned in the morning talks, NALCN play a critical role in regulating the neuronal excitability of the neuron. Consistent with that, by RNA sequencing, single-cell RNA sequencing analysis, we find out that the Purkinje cell exhibit the overall highest transcription level of NALCN 4 subunits in the cerebellar cortex comparing to its neighboring Golgi, inhibitor interneuron, and granule cells. In collaboration with IBGene, we generated a conditional NALCN knock-in mouse model which can selectively express the mutation R1A1Q at its endogenous locus. Specifically, we induced this point mutation at E31 and inserted a wild-type NALCN cDNA to rescue it in front of this mutation, which is fluxed and can be excised by Cre recombinase. So without Cre, it will still express the wild-type NALCN. To specifically express it in the Purkinje cell, we use a Cree driver called PCB2 Cree. And as shown here in the co-immunostaining, I verify that it is expressed in nearly all the Purkinje cells in 8-week-old mice cerebellar cortex. And that is consistent with previous studies that suggest the PCB2 to turn on Cree expression in Purkinje cell after postnatal 2 weeks. To examine the effect of R1A1Q mutation on Purkinje cell excitability and activity, I performed patch clamp electrophysiology recording on Purkinje cell within cerebellar lobule 5 and 6 in 8-week-old mice. Briefly, 4 different assays are applied. We measure the membrane excitability by examining the inward lead current and the resting membrane potential. And I also examine the neuronal activity, looking at both the spontaneous firing of action potential after blocking the presynaptic input, and also the evoked firing of action potential when we are injecting different level of current. I observed that the holding current for negative 60 millivolts, which reflects on the inward lead current, is significantly higher in R1A1Qs, Purkinje cells, compared to both control groups. Consistent with this effect of increased inward sodium leak, we also found a depolarization of the membrane potential of R1A1Q compared to the 2 control groups. And overall, that suggested R1A1Q gain of function mutation could lead to increased membrane excitability. To examine that effect in the R1A1Q, how R1A1Q affect Purkinje cell activity, I first performed cell attached recording to record the spontaneous activity. As shown here, both spontaneous active and silent neuron can be recorded. While most of the control cell exhibit a spontaneously active phenotypes, as shown in the black proportion for both control group and as the black trace shows, the R1A1Q Purkinje cell, most of them are silent. They do not fire any action potential at all. And that is interesting because usually the increase of inward sodium leak and the depolarization of membrane potential is linked to increased firing, but in our case, it is the reverse. It resulted in a suppression of activity. To investigate this, I performed current climb recording, injecting different steps of current from negative 100 picoamps to positive 600 picoamps. And as the result shown here, a typical trace, the control cells is able to fire sustainably during the 600 millisecond of current injection of 400 picoamps. However, for R1A1Q cell, while it is able to fire at first at a comparable rate to the control cells, the action potential amplitude reduced quickly, and finally it stopped firing. By injecting different level of current, I find that the maximum number of action potential fire within the 600 millisecond of current injection to be less than 10 spikes for the R1A1Q, which is much less than the average of around 60 for both control groups. One potential mechanism that could underlie this impairment is the depolarization block, or depolarization arrest, which can happen when the membrane gets extensively depolarized, that it prevents the voltage-gated sodium channel from properly recover from inactivation, and therefore those channels cannot fire together for the next action potential, so it will just arrest. And as shown here for the R1A1Q, we can see the baseline membrane potential being gradually depolarized when it fires at first, and finally be trapped at a steady state potential for around negative 33 millivolts. However, comparing to the control, which is able to fire sustainably and having a steady state potential in her spike at around negative 44 millivolts. And this depolarization arrest can be mimicked in control cell when we're injecting higher level of current. As here showed, the same cells that is able to fire at 400 picoamps of current injection fail to sustain firing when we increase the amount of current from 400 to 600. And we can see that the steady state potential get gradually depolarized and finally trapped at around negative 32 millivolts, which is similar to what we see in the R1A1Q, suggesting that the depolarization arrest we see in the R1A1Q Purkinje cell could be caused by the higher amount of current being conducted through the NALCN. To determine whether the RON1Q indeed increased the likelihood of this depolarization arrest, I compared the steady state potential during the 400 picoamps current injection, and I find that the steady state potential for RON1Q is significantly higher than the two control group, with most of them, over half of them, above negative 40 millivolts, where the depolarization block started to happen. So, in conclusion, we find out that the Purkinje cell carrying RON1Q mutation failed to sustain high-frequency firing, probably due to the increase of depolarization arrest. And this inability to sustain tonic high-frequency firing may contribute to the motor cerebellar dysfunction we observe in the Clifford patient. As Dr. Zeng mentioned earlier, our lab is currently screening for Clifford drug. And recently, I tested one of the candidate compounds, NAFLPAN, which is one of the candidate blocker of NALCN. We applied 35 micromolar of NAFLPAN on controlled Purkinje cell, and I find out that it is able to reduce the lead current and also hyperpolarize the memory potential, which act opposite of the RON1Q's effect. And in the future, we'll be focusing on testing this drug in the RON1Q Purkinje cell to examine whether it is also able to reduce the leak and hyperpolarize the memory potential, and whether it can rescue the activity. Finally, I would like to acknowledge everyone that contributed to this work. Zeng and Tang collect all the hex cells data. Tian Kong collect part of the Purkinje cell data. And I would like to thank our funding, IB Jin and Dr. Han Liu and Dr. Yibing Xiang from IB Jin, and also the Lundbeck Foundation for funding and supporting our research. And also I want to thank Dr. Stephen Plass for sharing the plasmid for hex cells recording, and Dr. Julie Lefebvre for sharing the PCB2 query line. And I want to thank Dr. Adriano Senatori for training of our previous former lab member for hex cell patch-comp techniques, and also Dr. Tatsuya Sugahara for general suggestion and comments on our mouse model. Thank you so much. Thank you.
Thank you, Tian. That was a really interesting talk. We have time for quite a few questions, so does anyone have questions for Tian?
I have a quick one. Your lead currents, a few hundred picoampere, seems remarkably high for a neuron. How did you record them?
Sorry, what?
Your lead currents, a few hundred picoampere, that's very large for a neuron. How was it recorded?
So you mean the lead current for these cells is too high. So these are from Purkinje cell, which usually have a holding current of around 100 and 150 picoamps when we're holding it at negative 60 millivolts. So it is higher, and for RON1Q it's even higher. So we injected a higher holding current to sustain it. Yeah.
So that can reflect many other.
Yeah. But we can see that the RON1Q increased very significantly.
Just two questions. The first one is, which cray line did you use? Which cray line?
Sorry?
Which cray line did you use?
Cree. PCP2 Cree. PCP2.
Okay.
Yeah.
And my second question, what was my second question? I forgot to. Okay. I will ask you later.
Maybe when it comes back to you,
then I have another one. So you did these recordings after 8 weeks, is that correct?
Yeah, at 8-week mice.
And so did you ever try at another point in time?
For me, I didn't try another time, but I do believe that a previous study in our lab did try to do it at 4 weeks, but I'm not too familiar about the result for that.
Okay, I remember my second question.
Yeah. Thank you.
No, maybe I missed the information, but did you observe cerebellar ataxia, cerebellar degenerescence and ataxia in your?
Yeah. So that is actually a great question. So we try to characterize the motor phenotype of this mice model, and as a result, the phenotype is really mild. We only see an increase of stride length, which is a step-say move. So one potential reason behind that is that we're actually expressing it in adult Parkinson's. The expression of it only turns on after 2 weeks. So what we think is that it may be affects the early and postnatal development of this type of cell in the patient, so leading to this motor ataxia and very dramatic phenotypes. And now we're using a new creline to express, called carbonyl 1 cre, to express it earlier during the development to see whether it is able to affect and cause the disease phenotype. And actually, the mice model is already shown by Dr. Mason earlier. So we have a mice that is very small compared to the legitimate, and we're currently screening for their behavior phenotype. Thank you.
Hi. I'm curious if you've considered trying to use dynamic clamp recording with perhaps HEK cells to, because I guess there's always a question of compensation.
Yep.
Just to see how the CLIFAD mutant versus wild type behaves under dynamic clamp, where you simulate potassium conductance in the background and see how that way you can measure a direct contribution of NALCN mutations to excitability.
To be honest, I didn't know too much about dynamic clamp, but I will look at it. Thank you. Thank you for your comments.
It's time for one more question. Then I'll ask the last question. I was just wondering if, so my experience working with gain-of-function mutations in heterologous systems, the cells tend to be less happy. So I was just wondering if you have the same problem working with brain slices.
So you mean the overhouse of the Purkinje cell?
Yeah, the Purkinje cell that you patch.
Yeah, we do observe that the Purkinje cell are harder to survive during the recording when we're patching them. That is happening, and I think it will be a good idea to test the viability of those cells in the mice cerebellum, which it is known that the impaired activity may contribute to those developmental apoptosis also.
Thank you, Tien.
Thank you.
Thank you so much for a wonderful presentation, Tien. So I'm very excited to introduce Dr. Dennis Lal. Dr. Lal is a faculty member and director of neurogenetics at the Center of Neurogenetics at UT Health Houston. He has a broad expertise, including in bioinformatics, channelopathies, large-scale genomic analyses, and is a major champion for the ultra-rare neurogenetic disease community and channelopathy communities. So very, very excited to have him join us from Houston. And we'll hand it off to you, Dr. Lal.
Thank you so much. So I can definitely guarantee that my talk will be confused, and that's what everyone needs at the end of the day. So let's say start mirroring. Is it mirroring? I can see it here, but not yet on the screen on the side. Yeah, perfect. So I directly jump in and I will show a lot of different things, I think, very different compared to other things shown today, and just to get more attention now that wakes everyone's up. You know, before we have barbecue, we have like some different stuff going on. Okay, so I'm very weird. I'm a biologist who became a data scientist, computational scientist, and now I lead clinics. Super weird, right? So, but I would say, based on this, I have some good overview what's going on. So we work with electrophysiological data, which others generate in consortium settings. But on the other hand, we are really discussing every week people with channelopathies and deciding what to do in clinic. And on the other hand, we're developing new software to make sense out of different data sets. And I really am a strong believer, you know, that nothing in biology is random. And the moment you bring data together, you can see patterns which enable, you know, to do inferences on function outcomes. But just needs to bring things together here. So, and here just like two tools I use. to look a little bit at some variants. I have no research program on NALCN, so it's, you know, and I don't plan to, but I would love to help people working together. So this is typical mandate what I have. And what you see here with patient variances could also be stratified by phenotypes or readouts from certain functional experiments. And then at the end, you know, it requires a certain level of, you know, domain expertise to bring it together, understand strengths and weaknesses. So when I come to this a little bit later. So, but one problem what we're already facing, I would say in disorders which are a little bit more ahead in the therapeutic pipeline, other channelopathies, is that we're really running into the problem of matching the right patient to the right compound or drug, and also assessing for every type of new therapeutic the level of risk. You know, nothing comes for free, right? Especially when we talk about variants which can strongly modify development. They typically require also stronger drugs, and stronger drugs, if not perfectly calibrated, may lead to things which we don't want. So it really requires a thought-through process. Just to reiterate a little bit the same topic. So typically when one gives a compound and one wants to achieve a certain therapeutic window, right? So one wants to be in the green zone. So assuming one is not in the, you know, green zone and the biology of that individual is not working how one would like to have it, one wants to bring it back, and that everything is corrected, right? So this is where we want. Traditionally with small molecules or old school therapeutics, you can try, you may overshoot, you might not have enough, and everything, you know, you just calibrate, the washes out, and then you try again until you are in the green zone, right? And you know how much you give, given, you know, the body weight of that person, other factors which may play a role. Not talking about polypharmacy, other factors and so on. However, when we talk about, you know, our precision therapeutics, which we want, or like biologically acting therapeutics, which often are what many people believe are required to really do the modification of the disease and not just dealing with a symptom temporarily, we may, you know, have great opportunity to maybe that have with one or few shots on target have something sustainable, which really corrects lifelong. However, if we have the wrong assumption about the underlying biology, we shoot off maybe lifelong, and that can be very dramatic. And we, you know, we need to have a good hypothesis to figure things out. So, so here's, and now I want to just bring. So often when I talk to basic scientists, many people are really engaged in their research and they're really passionate about their results when they write papers. And, you know, as scientists, we all need to sell also a little bit, you know, find the novelty, the cool thing and so on. However, I have many scientists who feel a little bit doing a step back when they say, hey, should we use this for clinic? Because suddenly you don't want to be responsible if your assay says gain of function, but at the end the child got hurt because your assay was not really calibrated, right? And so, but it's really important to be proactive in integrating science into care decision, because people make decisions every day. So if these research tools, which are not CLIA certified or so, if they are not used, then people make decisions without any information. And this is also a problem, or even bigger problem. And I just want to show you like a real world example what I'm talking about, and how things are done today in a setting where we have a specialty clinic where it happens to be that the leader of that clinic is advisory board member of that of the foundation, has the globally largest data set from patient and functional data at his hand, and has more than 15 years of research on that gene. That's me, right? So I'm directing this clinic, and I'm not a clinician, but I have a lot of history on this gene, and I have seen, whatever, 200, 300 patients. But this is, we have a specialty clinic, and we have to make decisions, right? There are people coming, parents who need help, and they want to get the best type of decision possible. And here's a report. I omitted everything what is, you know, should not be seen. So at the beginning, you summarize the medical history. Then you do all these assessments from behavior to whatever, to movement disorders and so on. But there's this point in time where you want to give prognostic information, and you want to give recommendations on which therapeutics to take, right? So because this is what people need, right? And so this requires this section here, which you typically never see, like a variant reanalysis, which we added here. So this is just one, what has ever been functionally tested and what the variant does on the protein, blah, blah, blah. And then we add some scores in here. And then when we come to the point where at some point we need to make the decision for this voltage-gated sodium channel here. Here's the variant here in purple from that patient. This is, again, it's a clinical report, right? So what does the variant do, right? And so, because for voltage-gated sodium channel, it's like SCN2A, if it's a gain-of-function variant, actually sodium channel blockers are useful. To sometimes, you have to look at it, but anyways, sometimes they can be useful. But if it's a loss-of-function variant, then they can be harmful, right? And this is nothing disease-modifying, but a symptom, relieving in that stage. But you need to have a hypothesis because you don't want to harm that individual, right? But there is no clear certified test, right? So what happens is that you need to do like some kind of detective work. You extract some information which is here and there in the literature. So on the one hand, there are, to some degree, established phenotypic proxies for the underlying disease phenotype. But you have to keep an eye on a lot of circularity. So, and I highlighted here in yellow everything which is pro-gain of function in the summary here. So, and then you can also combine, for example, how the patient responds currently to a drug as a proxy for molecular dysfunction and the medical history. So it has all these layers, even in phenotype space, which you need to somehow synthesize and score to come up at the end with a hypothesis what the patient has. Similarly, you have functional data, which can be a mouse, can be an electrophysiological readout, or could be from two distinct labs which have different readouts and maybe disagree slightly. Someone says mixed function and the other one says loss of function, mixed function. What is it, right? So you have to think about this. And so long story short here, so here it was even inconsistent. So the molecular assay, the in vitro assay suggested loss of function. However, the patient was responding to sodium channel blockers, which is based on an army of data, more in vivo evidence, and we would consider, overall, we consider this a gain of function for a couple of reasons. So, but these kind of decisions from people who have no research programs, like I have a research program in this space, right? So, and I have the largest databases because we are doing this for the foundations. People doing this with less resources, and it's happening every day, every minute, right? So somewhere on this planet. So it's really important that we bring all the information we have, and ideally curated by experts like you, to one place, that these informations can happen in the best way possible. And this is what we call in the world of research grants, let's build a knowledge base. So, and this happens in cancer space quite a bit, where people say, hey, if you have that variant in that type of tissue and with that spatial whatever a niche, and then you get this kind of companion diagnosis. This is almost a companion diagnostic, and then you get that chemotherapy or so. Okay, so but you want to go away from this. Okay, I read this, it feels to me like gain of function. You don't want to do this, right? This is not a good scientific way, it's not a good standardized way, and you cannot design a clinical trial protocol for enrollment based on, intuition, right? The intuition can be wrong. So you need to move towards more best practice guidelines, which incorporate functional data as well as clinical data as proxies for outcomes. So this is a talk which I will go over anyways until I get kicked out. But so I just want to briefly show you how this could look like. The geneticists in this room will already recognize that there is the so-called ACMG guidelines, which are used to classify the pathogenicity of a variant based on iterative design classification guidelines. They have been designed, they were actually revised every 2 weeks almost. But we can also develop similar kind of guidelines for just simple binary categories, and this could be, for example, should get an upregulating ASO, should get a downregulating ASO. So the label gain of function, loss of function is really bad. In my space, there's a lot of epilepsy and autism. People talk more about excitation and slowing down the system, and gain of function, loss of function more on the biophysical space. But there's a lot of terminology which is not really set up and is really readout specific, and then translating it to humans is very complicated. It requires a lot of people bringing data together and having discussions, knowledge base, again, super important. So, but you can start getting going, and you can, for example, generate now a scoring schema for weights for gain, in this case, and weights for loss of function using, for example, clinical data. You can just specific, based on literature evidence, assign points. If it's, for example, a hex cell, you get, and it says gain of function, it gets 2 points. If it's a mouse and the, the whole brain is on fire, it's really pushing, then you get 4 points or so. So you could think about rules. And I'm not an expert, but there are experts who can have discussions. And then you can just test in patient cohorts or in other systems if that's actually how well that scoring works. And you can then iterate also based on new evidence. And so, for example, how it can work even with clinical data is, for example, you can have the hypothesis that everyone who has a protein truncating variant has likely a haploinsufficiency, so less protein. And you can say, hey, everyone with a missense variant might be different. And based on just that, where you're not touching functional data because you want to keep this as a separate level of evidence, which is to avoid circularity, you can ask, do people with, so I assume that everyone with a missense variant, with a truncating variant, is haploinsufficient, so loss of function. Whereas for missense variant, it's a mixed bag. So if I compare people clinically, I should get slight statistical differences, and this could be attributed to that more gain of function group here. So even if this is a mixed group, you statistically, if you have enough people, you should see differences, and this is because here's a mixed group. And then you can, for example, here in this case, look if there's differences. And here's, for example, seizure onset. Here's for SCN2A in this case, we see that people with protein truncating variants, they're typically having seizure onset later, versus those with missense variants have it earlier. And there's some statistics one can borrow from the American College for Medical Genetics and Genomics for pathogenicity classification. You can borrow this type of statistics to generate weights, empirical weights for gain and loss of function. And so we're doing this here. And you can do this also with, so for example, here with seizure onset, but you can also do this, and then you can transfer it then into scorings. And you can do this also for hypotonia, it doesn't matter. So, but you also want to integrate functional data. And for many ion channels, you can even take functional readouts from related proteins. You don't even need to take the ones from the exact same one. For example, if there's enough structural homology or functional homology, make it a inactivation unit or a voltage sensor or something like this, or just being a pore lining residue, or you can develop statistics but research need to back up that it makes sense to do these transfers. Then you can, for example, develop simple statistics where you can identify, hey, some of these regions in that channel are enriched for gain versus loss of function. You can then run 3D statistics about this, and you get the exact same residues which have the highest density of gain versus loss of function variance, and then you can either statistically derive odds ratios, or you can have expert agreements and saying, hey, it would give 4 points versus 2 points for that level of evidence. So this is a mixed bag, and then you can bring everything together. So, but I just be very fast, I know I'm over. But you can also develop, the moment I saw so many in vitro assays today, if there would be one postdoc PhD student or so which would just generate a table and say, and just across all these different readouts would say, hey, Kate, this is variant X, this is gain of function, and then here, this is more loss of function, it would generate a table. We can develop prediction models, and we have done this in the past years, for example, for voltage-gated sodium and calcium channels, where we then learn, okay, variant specific, for example, in the S4 linker helix, they are more enriched for gain of function, versus the pore domain is typically more enriched, or selectivity pivot is more enriched for loss of function. So you can develop these kind of structure-based rules even for variants which you haven't seen because they're just located in a specific functional unit. And based on the data I've seen today, it would be possible. So, and yeah, so you can then predict, validate this and so on with independent data sets. So we developed also scores for example, the NMDA receptor, where they work similar. You see that, for example, population variants are located all over the place, whereas the patient variants, they're really close to the pore and the ligand binding sites. And then if we stratify even patient variants into those which have been functionally tested by Steve Tronellis into gain or loss of function, then we see that the loss of function variants are more clustered around the ligand binding sites, whereas the gain of function variants are more the magnesium binding sites in closed pore domain. And with this, we can then develop also a prediction model for pathogenicity, which is even beating AlphaFold, no surprise because we have a lot of data here, and even for gain of function, some basic funds. And you can even get potency. We're working now on, with a couple of people, trying to get the potency out based on the distance to the substrate. Okay, so, and then we can even, there's other ways to do it. If you combine it even with clinical data, you can even predict outcomes for mild versus severe disease. And this has also been published. Can I have 3 more minutes just to go to a web application? Okay, cool. So So I think the internet breaks always when I go here. Yeah, so just a second, I need to reconnect.
So you can also just go down to the passwords NALCN 25 if you for the promotional code. For that
Okay. Yeah. Yeah, but that's fake. So let's see. Okay, so this is not cool because this is a community effort. So from Steve Tornetti's group from Emory, he's the person who's mainly generating the EFIS readouts, and then there's clinical experts who have the natural history studies and registries, and this is the total number of people included here. So we are just more the bioinformatic and web application developing team. And here we have some just general information, what experts like you could say about the history, about these different NMDA receptor subunits, and then you can explore clinical data based just as for education. And we have built these tools for different ion channels. So this is the NMDA receptor portal. And the cool thing is, this is part of an FDA package. So every patient who is enrolled in the phase 3 for GRIN2B, where they only enroll people with gain-of-function variants in GRIN2B, they need to justify who is gain-of-function. And this needs and for this disease, you need to do this apparently through an in vitro assay, and the data needs to be in public space, and so we're uploading this here every week, or almost every week. So here's now a tool where you can submit a variant, in this case, this one here, and what you get is a lot of information. So you see that the variant I just submitted has been observed in 12 patients, and you have some clinical information about who has autism, who has ataxia, and not. Several of the patients have been published in individual case studies or case series, and you can read about them. So that's one feature. So basically, you just for the green genes, you don't need to go anymore much to the literature. You can just go here because we have all information or a lot of information here aggregated. You can even read about the design of the functional labels. Is it presumed gain of function? Is it is it label or gain of function or other kind of readouts? And here our Steve's team needed to really think about, for in a clinical setting, when do we really want to call it gain of function or loss of function or more presumed gain of function? Because what he would do in research papers would be different compared to what we do in this public-facing application, where also families and genetic counselors and geneticists go to. And so here's also a lot of things where how you can classify if the variant is really pathogenic, and also you can, for the bioinformaticians in this room, you can also then see here, for example, here's all the registry data, because we have 800 patients here, right? And we have here population variants. And you can see, for example, that many of the pathogenicity scores are not very well calibrated, So you want that the green one, so you see that the green one is going with the red patient cohort. But you see, so AlphaFold works quite well. But you see then, for example, also many of the population variants are predicted as pathogenic, which is not true. So you can do a lot of things when you have a lot of data at the same space. We can calculate hotspots on the fly. So here on the red is again the patient, his linear protein sequence space. You can see that here the variant I submitted is in a hotspot. And we can also do this also in 2D and 3D. So you can see the variant now here is in the transmembrane domain. And finally, we also have calculated, I mentioned this before, prediction models for, and this predicted as loss of function, and it was also functionally tested as loss of function. So with this thing, we can now provide people who want to make clinical decisions, or scientists who want to explore the data because they're really interested. Here we have this research interface where you can basically intersect everything with another. We enable now to make most of the data. For example, I can say, hey, show me only people who have seizures, and then all the structural modeling will only show residues in 2D and 3D space with seizures. Or if I'm interested in population variants, and I can also look here now on the functional readouts, because I have a lot of in vitro people here. So I can, for example, look at how the individual readouts give certain results in different domains. And I can even look here at potency. How the individual things change, and I say, okay, show me only people who have profound intransmissibility. How does magnesium change? There's apparently not too many. Okay, so I think I will stop here because the point I wanted to make is in a world where no individual data point has 100% certainty, which is 100% the case for care in neurodevelopmental disorders, we need to bring a lot of information together that we can make the best joint inference at any point in time. And this requires data sharing and working together with a lot of experts to develop a resource. And then at the same time, it gives also this typically generates new hypotheses and knows what to focus on and avoids redundancy. Okay, with this I stop. Thank you so much.
Thank you so much, Dr. Law. I don't think we'll have time for questions, but maybe if anyone has any questions afterwards to come ask Dr. Law. And I think that's a really wonderful strategy to help us think about ways to integrate the genetic data and genomic data, the functional data, as well as the clinical data, and then how to visualize it to the community. So I'm very excited to introduce Julia Castro, who's a genetic counselor at UT Health San Antonio, as well as a wonderful person. And so Julia and I work closely together, and she's kind enough to present. And this is largely a presentation just to help families and others understand how to read genetic reports. I know each of us with an affected family member in the room have all received these reports at one point in time, but not always with an explanation of what it means. So we're very thankful for Julia for talking with us about this. And then afterwards, we'll have closing, and then we'll go to barbecue from there. So thank you all.
Yeah, guys, thanks so much for having me. It's a privilege to be here. I am aware I'm the last thing between you and barbecue, so we'll keep that in mind. But just so I have an idea too of who is in the room, can I just get like a quick raise of hands of people who are here who don't necessarily have a science background, but are here because one of their loved ones has an NALCN related condition? Okay. You are the people that this talk is mostly geared towards, so we're glad you're here. I will also say for everyone else in the room, I could not echo loudly enough what was just spoken in the prior presentation. As a genetic counselor, part of my role is looking at these reports and determining what to make of them and what to tell families. And I don't work with a geneticist. So having a database where I can look at what we know about this gene, about hotspots, about gain of function versus loss of function mutations. I mean, this is the bread and butter of what helps me tell families information that is, critical to their understanding of what's going on and key decisions that they make from there. So couldn't echo it loudly enough. So this again, for everyone else in the room, this is kind of basics on what is on a genetic test report. What do these words mean? How do we read this? How do we make sense of it? Just a little bit of background about genetics in general, okay? So our genes are kind of like our body's instruction manual. Each gene is its own set of instructions. And like any other instructions, it's written out using a genetic code, which only has 4 letters in it, which is why you see those 4 letters repeated over and over and over. Side note, if you've ever seen the movie Gattaca, now there's an explanation for why the only letters in that title are G, A, T, and C, okay? But so each gene is its own set of instructions, and the body is able to read those instructions, and that informs the body on how to build a protein. And the protein is what's actually floating around in the body and making things happen. Each protein has a really specific role to play. Each protein is important for our health. Okay, so you can imagine, as with any set of instructions, if you modify them, what ends up getting built can also therefore be built differently. And again, you can kind of carry this metaphor out. Some pieces of the instruction might be really, really critical for the overall function of the protein, and some pieces might be slightly less critical. So it really matters what piece of the genetic instructions is being changed. Where in the gene is that genetic difference located? What predicted impact does this have on the protein, which then impacts health? I'm going to say everyone, but I work in genetics, so I'm going to say most everyone, I would expect, has 2 copies of the NALCN gene. Okay, so one from mom, one from dad. And when we see this, specifically with the NALCN gene, it kind of matters. First of all, is there a genetic difference present on just one copy of the gene, or is it present on both? Okay, that's a little bit simplified. It also still matters what that genetic difference is. But for someone to have an NALCN-related condition, if they have that condition and there's only a variant on one copy of the gene, that produces one condition, Cliff HDD, right? If it's present on both copies of the gene, we'll get something that might have some overlapping symptoms, but might also look a little different, okay? So when you get an actual genetic test report, it's long, there's a lot of things on it, and a lot of it probably looks like total medical jargon to most people, which is totally fair. These are the things or the categories that you might see listed on the report, and I'm Earth is written on this piece of paper. Okay, so this is actually a fairly well-known genetic variant in the NALCN gene, but again, to the untrained eye, it just looks confusing. I am well aware of this. So just to break this down a little bit, that C dot, which is what we call it, C dot notation, that's just saying, okay, where in the genetic code in this gene is the specific difference that that testing lab flagged? Okay, so for this example, at the 3,542nd letter in the genetic code, most people have the letter G at that position. But for someone whose report says this, that specific individual actually has the letter A at that position. That is what was changed in the genetic instructions. P dot just means, well, okay, so what happens to the protein that's built if that's the changes in the genetic instructions? So again, this is where there's different names for the kind of building blocks that are used to build a protein, but you can see it still gets swapped. So at position 1181 in the protein, there's a swap. That might have functional effects on how the protein is able to go and do the functions it's supposed to be able to perform in the body, which explains why someone might have an NALCN-related condition, okay? Zygosity is another term that probably is not super familiar, but it's actually pretty simple to understand. If it says heterozygous on the report, what that means is that specific variant that was just described, it's only present in one of that individual's 2 copies. If it says homozygous, it means that specific variant that was described, it's actually present in both copies, one from mom and from dad. I will say just because there's a genetic variant present, that does not always mean it was inherited from parents. Sometimes things happen spontaneously, but that's what the heterozygous and homozygous terms mean. You'll probably see this on pretty much any genetic test report. Mode of inheritance, this is going to be on some of them, and again, we kind of went over this a little bit earlier, but if we've only got a variant on one copy of the gene, for the most part, that's mostly going to correlate with one condition. If it's on both copies of the gene, so maybe you have two heterozygous variants, or you have a homozygous variant, so it's the same genetic difference on both copies of the gene, it's going to correlate with something slightly different. Okay, and then classification, that's going to be one of the last things written on the report. This is the way that the genetic testing lab says, this is how likely we think it is that this genetic difference can cause disease. If it says pathogenic, that is the most confident that the lab can be. They say, yeah, we know for a fact this is associated with NALCN related conditions. If it says likely pathogenic, the standard for that is essentially we're over 90% confident, okay? The lab is pretty sure that this is able to contribute to NALCN related conditions. But at least in my personal experience, when I get something back that's likely pathogenic instead of pathogenic, what that means is maybe we haven't seen this in somebody else with the condition before, but everything that we know about the genetic difference feels like it would make sense to be able to cause that condition. Whereas pathogenic is typically, everything seems like it would make sense to cause the condition, and we've seen it in lots of affected individuals. That's personal experience, but that seems to be how it kind of shakes out. And then again, uncertain, meaning we're not really sure. And for all the scientists in the room, this is where I absolutely need all of the data we can get to at least give our best guess of, okay, where in the gene is this located? Is it located in a part of the gene that tends to cause problems? What effects does it seem to have on the protein? So on and so forth, to try and make some sense to families of what an uncertain result means and how likely we think it is that this gene could potentially be the explanation for what's going on. And that is pretty much everything you'll see on a genetic test report. So that is oftentimes what I walk through with patients, and I know there will be some tomorrow that I'm meeting with too. But if there are any questions about that, I'm happy to take them now. And if not, I know we're all excited for barbecue. But yeah.
Am I loud enough or okay? Okay. Am I loud enough now? So I remember on my daughter's report it saying that her specific genetic code had not been found, like the missense she had not been found yet. Like they had something similar, but not her specific. Does she then get put into something to explain that? Or is there a way that like that information gets shared for like the next person down the road? Because I don't know how I would get into that, and that's what I'm curious about, is how can I help the next mother that does, or father?
Yeah, absolutely. So I'm going to assume, right, that it's saying on the test, okay, this is the first time we've ever seen this, at least to our knowledge. Like it's not reported anywhere that the lab has seen before. The answer to that is probably twofold. One is that there's one database, it's called ClinVar, and it often, the labs themselves will send the information to that database, and that database will essentially say, has this ever been seen before? And what did the lab think the classification was? Did they think it was related to disease or not? What that doesn't do is it doesn't tell me anything about the patient behind that report. It just tells me if it's been on a report before, which is why I think conferences like these, databases, things like that, are also helpful repositories for, has this been seen before? So when it shows up again, can we make a little more sense of this for someone than we were able to the first time around?
Other questions?
I think does that relate to a variant being reclassified? Yeah, great question. So those uncertain variants don't have to stay uncertain forever. And this is what I tell people. In theory, over time, as we learn more about a specific gene or even a specific variant, we're going to be able to say with confidence at some point, yes, this can cause an NALCN-related condition, or no, this seems to be random harmless genetic variation. In order for a variant to be reclassified, meaning we're going to either bump it up to pathogenic or down to benign if it's in that uncertain category, it has to meet specific criteria. And this was, if anyone was in the room, what was talked about a little bit before, ACMG criteria, essentially meaning there's a set of pieces of evidence that all labs use as a standard to decide, what do we think this variant is? Do we think it's harmful? Do we think it's harmless? When variants are shared in ClinVar, it can maybe contribute a little bit to reclassification, but oftentimes the most helpful things in being able to go from this is an uncertain variant to now we know what this means are things like research studies that look at the impact on the protein. So they study the protein that's been made from that specific variant set of instructions. Research reports that describe yes, this has been seen in individuals who have symptoms that look just like what we would experience or not. So I would say research studies and data repositories and communities coming together maybe have a little more power in reclassifying variants than some of those more agnostic databases.
I just want to make a comment to the families. Someone in our group had their variant reclassified, but her impetus for doing that is she was going to be excluded from a study, and she wanted to be included. And so.
There's a comment to this. So according to guidelines, every 2 years, people are supposed to reclassify or look at genetic test results again in clinical settings. Just also to make sure that what maybe was classified as pathogenic is true from today's perspective still pathogenic, which is typically the case, but also these uncertain ones that you either seek more genetic testing, there might be better technology aware, but also to maybe turn around some of those. And but the unfortunate thing is the reimbursement structure and the digital systems to make this routinely are not in place. So, so that's why you need to be, unfortunately, as parents, also very proactive in this space. But typically the clinical team does it, but you need to push them to, unfortunately.
Yeah, I'll just corroborate and say in calls that I've made with labs before, it sounds a lot more like if we happen to see this again on a report, we'll take a look at it again, but there's not a super standardized procedure for we're going to be able to look at every uncertain variant every year and restudy it. So it does require more push, I think, to have that happen.
In general, with de novo mutations, do they, when I say ever, I mean, is it a frequent thing that they reoccur like exact genetic mutations, or is it often that de novo mutations are different because the code is so long, so many things are so specific? Is that what most of us are facing, that there'll likely never be another child discovered, or very few, with the exact mutation, so thus making it harder to know exactly what exact symptoms they're going to get, like some sort of bio-individuality thing? Is that what we are really looking at with most other diseases, rare diseases? Using them as your guide. Is that how it works?
Yeah, no, great question. And by the way, I'm going to assume that people are familiar with the word de novo mutation, but essentially that means it's one of those ones that was not inherited. Neither parent had that mutation, but it happened to arise spontaneously in the child that's affected. The answer to that is it can kind of depend from gene to gene. And in a lot of genes, there's an idea that there's these, what we call mutational hotspots, which essentially means this seems to be a region of the gene where a lot of the mutations, either these are where all the harmful changes seem to occur, or these are where the changes seem to occur a lot in general.
So in the case with the Clifford thing, it seems that it occurs in the same domain, like the same section. That I get, but it's slightly different in each one. But the symptoms seem rather similar, but the severity, varies and things like that. So, like I said, in this particular case, when you said that they're hotspots, in those diseases, are they like the frequency with which you see something happening, like a de novo mutation? Like, why would it occur over and over and over spontaneously in people? And at that point, isn't there some sort of inheritance factor maybe that they're missing, like tied to other genes, or I don't know.
Yeah. Here, I'll answer that as best I can, maybe twofold. One is sometimes there are certain regions of the gene that, for lack of a better word, are like more slippery than others. And what I mean by that is you think about the fact that every time your body produces a new cell, it has to copy down the genetic code again to put it in the new cell. There are going to be some regions where it's more likely to make a slip up than others. So for some genes, that's an explanation why that specific region of the gene seems to gather de novo mutations more often than other regions of the gene. It's just easier for the body to make a mistake when it's copying down the genetic code to put it in a new cell. In terms of the question, does that mean that all de novo mutations are hopeless in terms of prognostic information and right, like where are we looking at with this? That's also where there's other things that help us give prognostic information. For example, right, maybe this is a brand new de novo mutation. I've never seen this before in anyone else, at least that's been reported. But it seems to be in the region of the gene where all of the other disease-causing mutations are. And we know for a fact that that then impacts a critical function of the protein. So for NALCN, maybe it's in that poor region where those ions are going in and out, and it's more likely to cause problems. So there's other pieces of evidence that help your genetics experts decide, what do we make of this and what do we tell families with confidence, even if this is something we've never seen before? Yeah, no problem. Okay, well, I will be around tomorrow, so if there are more questions, please come find me, but I am going to take this as my cue that we're good to go, so.
If you want to have a one-on-one with Julia tomorrow, send me an email, emily@channelinghope.org, and I can set it up, unless I've already set it up for you.
Hi, so we'll be quick, I promise, because I know it's just us and then Ruby's barbecue. But I'm a parent. I have a almost 21-year-old son who, when I was pregnant, we knew there was something going on. We had all the genetic testing done at the time that was available, and he was diagnosed with syndrome, but not the correct one, because at that point, Clifford was not discovered yet. When he was about 10, we participated in a study out in Seattle, Washington with a doctor. He was one of the patients in that study in which we learned what his true diagnosis was. So even for the second half of his life, taking him to the best hospitals, the best specialists, people ask me, what? How do you spell it? What's it called? I direct them to what I know and the papers that are out there. But when I walked in to get a water before, and I saw all of you up at that board with your Post-its collaborating, I cannot tell you how excited I was. Just having you all together means so much to the families of the kids and the adults that are affected by these syndromes. And I thank you all for coming. I know some of you have traveled far, some of you have not, but you have taken away from very valuable time you all have, and it just means so much to me, and I know the other parents, because the more you're together and learning, the more we learn, and the more we can really help our children and our adults. So I would just really want to thank you all.
Hi, everyone. My name's Diana Dugan, and I'm the mom of Amanda, who's a 9-year-old with an NALCN genetic disorder. I traveled here from southern New Hampshire with my husband, Mark, my eldest daughter, Michaela, who's 11, and then, of course, Amanda. So I'm sure you'll see her around this evening. Our journey started, I would say, probably as Amanda was close to 1-year-old. We knew something was going on. She was very developmentally delayed, and we got referred to a neurologist down in Boston. We met with that neurologist. We did very high-level genetic testing at the time, and everything came back negative. And so I think I've heard from a few families talking that this is very common, and Amanda was diagnosed with a severe developmental delay. As Amanda started getting older, she definitely was still severely delayed. She's still nonverbal. So at the end of 2019, I had requested our neurologist to do an MRI just to cross everything off the list. Did the MRI, everything came back clear. It was then we were offered the whole exome sequencing, which we were ended up being able to get her diagnosis through that testing. So that was in September of 2020, and I've been able to meet so many amazing people in this community today, and working with so many people over the last few years, and learning so much about this disease that is impacting Amanda and all of the amazing families and everyone who's impacted that's here today. So I personally want to thank you from the bottom of my heart, my family's heart, for everything that you do. It is truly fascinating to meet so many of you and be able to interact and learn and really hope for a future with a cure to help these children and young adults that we've been meeting this week. So very much thank you. Lastly, just so I don't forget, we do want your help to nominate an advocate of hope this weekend. We want a person who's looking to raise awareness, provide advocacy, and offer support to individuals and families affected by NALCN and related conditions. The Advocate of Hope embodies compassion, leadership, and a deep commitment to improving the lives of these living with this disorder, making a lasting impact on both individuals and the broader scientific and healthcare communities. So at that, I think we're going out for barbecue. So again, thank you all. I'm looking forward to talking to you all more this evening, and hope you have a great night.
Okay, time to go eat. Thank you all. So the Rudy's is about a 10-minute walk. For anyone who wants to walk, there'll be some little carpooling for anyone who wants to ride, and we'll go from there. And if anyone doesn't want to wait and wants to call an Uber, feel free.